आ गए मैम, आ गए। Good morning, eminent guests, esteemed speakers, and delegates. As we approach the final day of this exciting and insightful conference, I am delighted to welcome you all to the first technical session of today's proceedings. It is my privilege to introduce the theme of this session, nanopharmaceuticals and cutting-edge nanodiagnostics, a subject that is at the forefront of medical innovation. Now, to guide us through this fascinating session, it is my honor to welcome the moderator, Dr. Saman Fatima Ma'am, Assistant Professor, Amity Institute of Pharmacy, Amity University. Please join me in extending a warm welcome to Dr. Saman Fatima as she takes the dais.
Good morning, everyone. It is my pleasure to welcome you all to today's session. To commence, I am honored to invite our esteemed chairperson, Professor Farhan Ahmed and Dr. Mohammad Shamim to the stage to lead us through the proceedings. Professor Farhan, a renowned expert in nanomedicine, serving as the Dean at the School of Pharmaceutical Education and Research, Jamia Hamdard, New Delhi. With over 27 years of experience in research and teaching, he has made significant contributions to the field of nanotechnology-driven drug delivery. His research has led to the development of several DCGI-approved nanoformulations, some of which have been successfully transferred to the defense services. His pioneering work includes the development of nano-dry powder inhalers for COPD patients and iron oxide magnetic nanoparticles for enhanced tumor treatment. Professor Ahmed has received numerous prestigious awards, including the Young Scientist Fast Track from DST, Scientist of the Year 2005 from NISA, UGC Research Award 2011, Bharat Jyoti Award, Pharma Ratan, and the ABAP Senior Scientist Award 2017. He played a key role in establishing Jamia Hamdard's Nanomedicine Lab and the Central Instrumentation Facility. Dr. Mohammad Shamim is Associate Professor at Jamia Hamdard and a leading researcher in nanomedicine, specializing in the synthesis and characterization of nanoparticles for targeted drug delivery and imaging systems. His work integrates nanotechnology with molecular therapeutics to develop innovative solutions for various diseases. With a strong focus on precision medicine, he explores nanoparticle conjugation with small molecules to enhance therapeutic efficacy. Please join me in welcoming Professor Farhan and Dr. Mohammad Shamim. I now request Honorable Chairpersons to kindly begin the session.
Scholars, speakers, I think this is the beginning of the session. Today's first session is about nanopharmaceuticals and nanodiagnostic. So we have learned speakers, Dr. Sandra, Professor Alexander, Mr. Shukla, Dr. Fatima, Dr. Nanika Gunja, Professor Ishita Pali. I request my co-chair, Professor Shani, to introduce the first speaker.
So the first speaker of this session is Dr. Saif Hamid. And Dr. Saif Hamid is an associate professor at the Amity Institute of Biotechnology, Amity University, Haryana, specializing in infectious disease, diagnostic, and antimicrobial resistance. He has conducted postdoctoral research as a CSR research associate and visiting scholars at Henry Kenyon University, Germany. He is a recipient of the Young Scientist Award, SERV 2012, and also a lifetime member of International Society of Infectious Disease. So I formally welcome Professor Saif Rahmi to start his lecture and enlighten all of us in this.
This is the counter. स्पेलिंग करना होगा नहीं हॉट इज चेंज ओके ये चेंजिंग का आ गया चैन पॉइंटर दिस इज द पॉइंटर ओके very good morning to one and all. So first of all, I'd like to thank Professor MZ Abdeen for remembering me and inviting me for this conference. As you have already heard about my biography, that I am an alumnus of Jamia Hamdard itself from the Department of Biotechnology, way back in 2005 pass out batch. So it's been exactly 20 years. I am standing here in front of my teachers. I could see some faces. Dr. Humaira is here, and Dr. Abdeen and Dr. Deepshika Pandey is also another speaker. So good to remnants of the old times, and it's a moment of heartfelt moment for me. So I'm currently working at Amity University. This is the campus situated in the suburbs of district Gurgaon. So any of you are welcome to visit anytime for any training or any collaboration, any sort of help. We are open to provide any help. So to start with, this is my current area of research in which we are working at the moment. So mainly we work on infectious diseases to combat antimicrobial resistance. Antimicrobial resistance is a very major issue nowadays towards efficient therapeutics for any pathogen. So one of the areas in which we are working is I am working is stress biology, where we see how the availability of micronutrients have an impact on drug resistance. So that is one area from my PhD work that I am still continuing to work. Another area is how the natural inhibitors from natural sources, Ayush medicines, how they are working against the microbes. So combating the antimicrobial resistance, we are working on those. That is another area. Then another area in which we are working is the lipidomics. Lipidomics means the mass spectrometry techniques utilized to distinguish the differences in the cell envelope structures about drug sensitive and resistant strains. And recently we have started in the disease diagnostics from the corona pandemic times, the work which I am going to present today. So tuberculosis need not require any introduction. It's a disease which is present with the mankind since antiquity. Okay. The causative agent is Mycobacterium tuberculosis. It is a very notorious pathogen, and we are fighting, entire world is fighting against it. The seriousness of the disease is exemplified by the fact that millions of people die every year from tuberculosis. So, bearing the 3 years of corona, it is still the leading cause of the death from the single infectious agent throughout the world. Okay, you can say that globally almost one third population is affected with tuberculosis. So these are the current anti-TB drugs which are in the market. First line TB drugs, second line TB drugs, some new drugs are also there. Some drugs are in the clinical trials process. But the main problem is the emergence of drug resistance that creates the hurdle against the efficient therapeutics. Now, the current diagnostic methods, we have the culture-based methods, which is the gold standard, but the problem is it is less specific, time consuming. Then we have classical smear microscopy, that is the Zn staining, Zell-Nielsen staining, from where you can see the smears and we say positive or negative, but again it is less sensitive. Then we move on to PCR based methods like DNA PCR or more sophisticated gene expert nowadays, which is doing on. But problem again is that they are although very sensitive, specific, but they require equipment dependency, trained manpower, money also. And we all know that tuberculosis is a disease of poor. So you need we should have devised method that poor people can also do. So let us understand the research gap. What is the research gap? The research gap is if there is delay in tuberculosis diagnosis or we misdiagnose it with some other strains, then it will lead to onset of drug resistance. And because of the onset of drug resistance, the treatment becomes complicated. It will fail. So on other hand, if somehow we do early diagnosis and accurate diagnosis of tuberculosis, then we can initiate the correct therapeutics timely, so the drug resistance can be forgotten. So the need of the hour is that we have to somehow develop some rapid, sensitive, specific assay that is easy to detect tuberculosis so that the correct therapeutics can be initiated to prevent the onset of drug resistance. So isothermal techniques have tried to bridge in the gap between this infectious disease diagnosis because it works on a thermocycler independent amplification. So this chemistry of independent isothermal amplification means that the amplification will take place at a certain constant temperature. So this is very useful in the point of care diagnostic nowadays, and the isothermal techniques are utilized. So one of the most common isothermal technique is very famously called as loop mediated isothermal amplification, which is called as LAMP. So this is the isothermal technique that is used for disease diagnostics. So it amplifies DNA with high efficiency and utilizes 6 different primers. BST polymerizes the enzyme, and the amplification takes place at only one constant temperature between 60 to 60, just one temperature. So it can be done very easily. It can be done with in a water bath, even in a kettle of hot water we can do it. And results can be visualized with the naked eye. And RT-LAMP is the RNA version. For example, if you use RNA template, then it will be called as RT-LAMP. So why it is better than PCR? Because in PCR you use 2 primers, there are 6 primers. Temperature is variable in PCR, because of which you require a thermocycler. It is a constant temperature. Duration takes 2 to 3 hours, but it is very efficient and takes hardly 30 to 40 minutes. Strand displacement is a single elongation reaction, but here we have multiple elongation reaction. The enzymes is different. This is tag polymerase, this is BST polymerase, and the major hurdle, that is thermocycler. You require a PCR machine. You require a PCR machine that you can do this in water bath. And detection is also very simple. It is visual detection we can do, or by gel electrophoresis or fluorescent method. This is just a schematic representation just to understand what is LAMP. So basically what happens is you have 3 primers, forward, inner and outer primer, and a loop primer. So when the inner primer binds and start elongating, then simultaneously this second primer bind and it will displace the elongated strand. This displaced strand forms the loop, and then you have a loop primer. So basically 3 pairs of primers very efficiently in just 20-30 minutes form a dumbbell shaped structure, which is very good in amplification. And we can see it in a by turbidometry, by eye estimation it can be seen. Okay. But that technique, because of its high sensitivity, has a disadvantage also, that sometimes we get false positive reaction. So to prevent that, nowadays what we are doing is you can use the CRISPR technology. CRISPR is a new age genomic uh, diagnostic tools that is used by utilizing the Cas proteins, and it can be integrated with the isothermal amplification techniques so that the false positivity issues can be detected. So you have a very specific guide RNA, CRISPR RNA, guide RNA, with the help of the enzyme, when it goes and bind to the target nucleic acid, then it can be detected with the help of any labeled probe. So these are the two systems which are currently very useful in diagnostics, Cas12 and Cas13. They work on the principle that in Cas12 system you have a single stranded DNA reporter, and in 13 you have a single stranded RNA reporter. And in both the cases, you can use a labeled probe. So when the Cas enzyme binds to the target, it will have the collateral cleavage of the probe, and you can detect by the fluorescent methods. So if you combine both the techniques, what happens is the DNA or RNA is amplified by the isothermal techniques like LAMP or RPA, and then the amplified product with the help of this CRISPR enzyme can be specifically detected upon the cleavage. So if the target sequence is amplified, then only the CRISPR enzyme will go and bind, and then the enzyme will be activated, leading to the collateral cleavage of the labeled probe which is present in the vicinity. So in pursuit of such developing of the protocol, in our lab, we developed a protocol which we named as CRISPR-Cas coupled RT-LAMP amplifying protocol for tuberculosis, and we named it as CLAP-TB. So in the first step, we are isolating the RNA. Then in the second step, we are doing the RT-LAMP reaction, and by the color change, we can detect. So at this stage only, we can detect whether the sample is positive or negative. Simultaneously, we are doing the nucleoprotein complex where the guide RNA and the enzyme is incubated. And the fourth step, we are mixing this nucleoprotein complex and the amplified product here for 60 minutes. And finally, we use to test when we load in just 3 minutes based on the band pattern in the lateral flow, we can say that the sample is positive or not. In the control band, we get only one band, and in the test band, we can get both the bands. So this is our results. First, we standardize the protocol on the RNA, MTB RNA. So in MTB RNA, this was the RNA from MTB. This is non-template control. Non-template control means it will don't have any template. So after 40 minutes, we see that the color changes to yellow, and in the negative control, it remains pink. So this means that in the 40 minutes time, the amplification was done. This was confirmed by gel electrophoresis. This is a typical pattern that we observe for the LAMP reaction. And NTC, there is no smear. And then after CRISPR reaction, when you see, you can see that there is two bands and there is only one band here. So that confirms that the test was positive for this. After optimizing the protocol in our RNA, then we started to see how sensitive is our protocol. So we started serial dilution. So in all the serial dilution, you see from here, 1000 femtograms, 100 femtograms, you see, till AG we go. And the beauty is that even till AG, that is attogram, the reaction is turning out positive. As you can see in the 6th lane, you are getting positive result. And here you can see the bands. Negative control, there is no band. So this is very sensitive protocol when RNA is using. Then we ask the question that why RNA? Why can't we use DNA? So we started comparing the sensitivity, and we see that till lane 4 only, when DNA is used, till 4 lane only you are getting positive, and from 5 and 6 onwards we are not getting. But when RNA is used, again till lane number 6, we are getting the positive results. So RNA was more sensitive. These are the band pattern. You can see till 4 lane the bands are coming, but not in the 5 and 6. So for DNA, we could see the sensitivity only till 100 femtograms, but in RNA, we could get till AG. So RNA was more sensitive than DNA. Then we wanted to check the specificity of the protocol, that how specific it is detecting. So we used RNA from 6 different species: Mycobacterium tuberculosis, Mycobacterium marinum, Smegmatis, E. coli, one fungal strain also, Candida, and of course the NTC. And you can see that only lane number 1 turns out to be positive, and rest all were negative. From the gel pattern only, the lane number 1 is positive, rest is negative, and in the CRISPR also it is simple. So the protocol was very specific also. So after seeing the sensitivity and specificity, we wanted to validate the protocol clinically. So we took the sputum samples from the collaboration of AIMS, we get samples. The sputum samples were tested, and you can see that the positive samples, PS, were the positive samples, NS are the negative samples. So positive samples are turning yellow, negative samples are remain pink. These are the gel patterns. So negative ones are not coming here. Same are the band patterns. Then also we took some drug-resistant samples, drug-resistant TB, and we get similar results. The pink count turns out to be blue, the NTC is pink, the band patterns also suggest same as the gel thing. So after sputum, we thought, what about other samples? Because sputum is a difficult samples to get. Particularly the children and older people cannot give sputum easily. It's an invasive procedure. So we want to see if from the blood sample it can be done. So we took serum samples of the same patients, and luckily serum samples were also giving positive results. You can see the positive samples are positive, the negative samples are pink. These are the gel images, and these are also giving similar results. Further, we also test on urine samples because whether it is sputum or it is blood, it's an invasive procedure. So we wanted to see whether if we took any non-invasive sample like urine, which is easy to give, so whether our assay is tested positive on urine samples or not. And interestingly, we could see that even on urine samples, the test worked. So positive samples are tested positive and negative samples are tested negative. From the LAMP assay, this is the gel images, and these are the lateral flow images. So in all the three categories, we could see. We also took some extra pulmonary tuberculosis samples because usually 80% cases is pulmonary tuberculosis, but TB also affects other organs also. So we took some, yeah. So we took some APTB samples also, and the tests were positive for APTB samples as well. These are the APTB images. These are the healthy controls. Wanted to see whether the negative patients are turned out to be negative or not. So these are the healthy patients. Nothing is there. It turned out to be negative. See, only the positive control is coming and rest all are negative. Then this is a control gene. Whenever we are making a protocol, then we have to also have some internal control gene. So POP7 is an RNase P gene, and you can see that all the positive and all the negative, it should come positive because it is a constitutive express gene. So even in the negative sample and positive sample, it is coming. So this is the statistical validation. We have done about 232 samples. So giving the clinical sensitivity of 98% because just 4 samples we could not test positive. They were positive, but giving false negative. So it was 98%. Now we are also currently doing, apart from lateral flow assay, we are also doing fluorescence based assay because the strip is also a little costly thing. So we are working on fluorescence. So now we have developed the protocol on fluorescence also. I'm not showing the data. This is the graphical summary of the protocol. You have to take the samples, RNA is isolated, the genes are, the primers are there, and you can do the LAMP reaction even in a kettle. Then you do the CRISPR followed by this lateral flow, and you can propose a diagnostic kit. So in that direction, we have to work now. This is the conclusion that this assay is rapid because it takes less than 2 hours, sensitive also because we are integrating 2 techniques, specific because of the Cas enzyme, and easy to use because the calorimetric reaction coupled to lateral flow readout will facilitate the visual inspection of the results, and adaptability also because this method can be easily adjusted for the detection of the any pathogen. We are working for tuberculosis. These are the outcome of our study. We have filed 2 patents for the fluorescence based and the lateral flow based assay, and the paper is also about to be published in Drug Target Insights. Just came for some minor revision. This is one last slide I want to show the chairperson allow that shows the genesis of the study from where actually we get the idea of such. So this was during pandemic, Corona time. We got a project from BRNS BARC, and this was which is 1 year project. So there also in the same idea we developed assay for SARS-CoV-2. So we named as Clever assay, CRISPR integrated RT-LAMP easy visual extraction free RNA. The assay was clever in the sense because we bypassed the RNA extraction step. Because at that time, if you remember, that RNA extraction was a hurdle during Corona time. It requires RT-PCR machine. So we bypassed this step. So that was the uniqueness in this essay. We filed a patent also, and this project was completed. So from there, this idea originated. Now we are doing it for tuberculosis, and currently we are also working for fungal pathogens also. So this, I like to thank the ICMR for the project which we are working currently, MIT University for all the facilities. The co-investigators of this project, Dr. Zishan Frasna from MIT University itself, Dr. Jiten Singh from AIMS, Bhopal, and Dr. Sarman Singh, Dr. Mandira Basil, Pramod Mehta, Dr. Devashish for the generous gifts of strains they provided from timely. This is our group. Nothing is possible without the researchers. So he is Ankush. His work I have presented right in the center. So thank you so much for the time, and I will be happy to answer any question if there is any doubt.
So if audience have any question, they can ask. Otherwise, we will move to the next speaker. I would like to ask.
Yes.
Morning, sir. I really enjoyed the session. Really about the degree that's now. Sir, I would like to ask. Sir, I would like to ask. Like we all get MTP vaccines for with microbial in our childhood. So for Mount test, it gets called positive. So is there any relation which we can find latent or active TB, or it will just give the result that this bacteria in it? Because if we all are vaccinated, it is a very sensitive like NG or AG level.
Yes, yes.
So is there any chance?
No, the idea of developing this assay was only to do a qualitative test only, so that in quick time you tell the person whether it is positive or not. So we cannot tell about Montauk's test and all.
Sir, for the very high
Yes.
because the vaccine was
If he's healthy, then
we got vaccinated. So every person so positive with false positive.
false positive for the tuberculosis. No, so that's why we have also integrated CRISPR technology only, not for that. We cannot get the false positive result.
Thank you.
We can discuss in the. Yes, yes. Sorry, no, APTB samples were different.
Sample, from where you're collecting the sample?
AIMS.
No, no. Site, like you are diagnosing from the urine sample or the blood sample or
No,
from
we did from urine also. We did from sputum also.
which organ is affected? The what-
This is the
the major organ is affected, no?
yes.
Which organ is affected by the
No, organ we cannot test to tell. We can only test whether the person is positive or not. Because the usual culture methods will take at least 3 to 4 weeks to tell a person is positive or not. So the idea was just to in just 2 hours we can say that the person is positive. Then you can do further test which organ is affected or not, and you know.
there, no?
Huh.
So we look for it where which organ is affected.
Yes,
That's the
yes.
major question is-
Yes.
Okay. Stability of RNA.
Mm-hmm.
When you are taking out the RNA from the urine, how stable it is, the urine in comparison to the blood. So it's better to take the sample from the blood from taking the sample from the urine, which will be better for you taking.
This we are still doing. The sensitivity of urine sample is more or blood samples is more. This we have not done. The sensitivity data that I have shown today was from the culture RNA. So this we are still doing. This project we got last year only, 2024. So
It's micro normal RNA.
normal total RNA we get from yes.
So our next speaker is so the next speaker is Dr. Hitesh Kumar Shukla. And Dr. Hitesh Kumar Shukla is the center head at the Center for Industrial Policy and Drug Regulatory Affairs at University, Noida. With expertise in reformulation, profiling, polymorphism, and crystalline solid dispersion and drug regulatory affairs, he has contributed significantly to oral viability, enhancement, and formulation development. He is a recipient of Sir INSA DICT grants and Best Teacher Award and also a lifetime member of PCL, API, IPGA, CERS, USA. So with this introduction, I welcome Dr. Hitesh Kumar Shukla and request him to go for this lecture.
Thank you very much, sir. So, a very good morning to Jamia. I would like to be more focus on regulatory structure, how you can convert your research into the pharmaceutical product, or you can say, as per FDA regulations, each and everything which is used for diagnostic, identification, cure, prevention, or any medico-legal purpose that is going to be considered as a drug. So little bit, these biosimilars are very much fall under this particular class. So today we'll discuss how we can convert our research into the pharmaceutical product, a full branch product to the marketing product. Before that, I would like to thank Professor Abdin sir, Dr. Fatima ma'am, and team of organizing team of this Jamia Hamdard for inviting me for this beautiful gathering. I am honored. Thank you very much. Okay. This is the flow of my presentation. So as we know very well that anything which is obtained from biological source, that is known as a biologics. And today my topic of discussion is biosimilars. So as far as today is concerned, a biological drug, the biologics are the product that are produced from living organisms composed of proteins, serve as a protein treatment for the diverse medical conditions ranging from rheumatoid arthritis to the cancer. So biosimilar products are very similar to the reference product. What is this reference product? Reference product is the already established product, which is already approved by the regulators. And if we are going to develop a similar product like that, which is already approved by the regulators, this is known as biosimilar product. So basically, this is something like twins. They are having same identity with different characters. Sometimes they are more potent as compared to the reference product. And biosimilars are proteins that are similar to the innovator biologics, but have some same or differ slightly with respect to their activity and their pharmacological response. So this is the you can easily evident that this is the picture of a prototype with respect to reference product as well as the biosimilar. So unique cell line produce a similar therapy. A biosimilar is a biologic medicine that is similar but not identical to the original biologics that it is based on. This means the biosimilars are clinically indistinguishable from the reference product, even though it is not the exact copy. So have you saw means they are just like twins. They are having same identity, but they are different in their activity. Today my topic is regulatory aspects, so I'm not going more deep in the biotechnological aspects. There is always a confusion between biosimilars and generics. This is a little bit, most of the persons are confused that biosimilars are generics only. No, they are entirely different. Generics are synthetic products mostly, and this is something related to the Intellectual Property Rights Patent Act. If your pharmaceutical product is approved by the regulators and it has completed 20 years, its patents period, then it becomes generic. So all generic products can be, your all biosimilars can be converted into the generic products, but all generic products cannot be converted into the biosimilars. This is the major difference. Biosimilars, basically, they are composed of from biological source, but generics may be from biologics as well from the chemical entity as well. Biopharmaceuticals are the substance with the high molecular weight. Proteins molecular weight is quite high. But in the case of synthetic, molecular weight or structure are more simpler as compared to the biological products. They can be extremely sensitive to the physical environment in the case of biological products, but in the case of synthetic product, they are typically more stable and more predictable. Biotechnological products or biosimilars are more potentially immunogenic, whereas generic products, if they are from synthetic environment, they are immunologically inactive. And finally, extensive clinical research is required to prove that the similar efficiency and safety profile with respect to and as compared to innovative product with respect to biosimilars. But in the case of generic products, they are already established. We have already utilized them from last 20 years and they are in the market. So today my focus will be more on approval pathways instead of technical details with respect to biosimilars. So as we know, drug discovery, everyone knows it's just like drug discovery is nothing like your biotechnological product development, what you are doing in the lab. If anything which is having therapeutic activity, we are going to consider as a drug. So in that case, this is the okay. It's not good. No, not to talk now. I want to highlight something. This one. So I'm going to give my little bit discussion here and here. How you can approve your product at investigation level, and when your product is already completed the research pathway, how you can convert your product into the full-fledged marketing product. So today, this is the overall drug discovery pathway that what we are doing in our labs. Phase 1, phase 2, phase 3 clinical trials are going on. So I am going to focus more on what are the regulatory requirement, documentary requirement with respect to approval of your product. So this is the pathway flow chart with respect to your product approval in Europe. As per the regulatory structure, India is the federation of states. Europe is the federation of independent nations. So each and every nation have their own regulations, but they are commonly regulated by European Union. Apart from this, that's why they are going to follow four system of approvals: centralized, decentralized, mutual, and individual. So basically, whenever your product is there, it pass your this basic ECDT preparation. It is having 5 parts: part 1, module 1, module 2, module 3, and module 4, and module 5. When it comes, then when you have completed all these things, your product is getting approved. So in the initial stage, you are going to have only module 1, module 2, and module 3 data. That is infrastructure requirement, quality requirement, and the product quality requirements. In 4, you are going to give the specifications with respect to preclinical data. In fifth module, you are going to give the data with respect to the clinical aspects or human subjects for any approval. So once you are going to apply for INDA, you must have data up to level 4, module 4. It means infrastructure, quality requirement, product related quality requirement, as well as your preclinical data with you for the approval of this particular any biosimilar product in European Union or anywhere. Nowadays, each and every regulatory agency is looking for electronic common technical document for their submission process. Only some Asian countries, some ASEAN countries, and some African countries are asking for hard copy data for their approval process. Apart from this, all like TGA, USFDA, MHRA, everyone, even CEDESCO, is asking for soft copy data or electronic submission for the approval process with respect to their products. So this is the common pathway with respect to the product approval process with respect to in European Union. Yes, one more thing I would like to add here, a note that in few European countries, they are looking for eCDT format in their own language, like French, German, or something. So this is a note whenever you're going for approval in the European countries, you must keep these things in your mind that English is not an universal language. They may ask any data in their own language for better understanding. So there are the few common things which are I'm going a little bit fast. So in Europe, European agency, EMA, that is European Medicine Agency, is responsible for the approval of all kind of product, and they are going to give you the regulatory pathway for the centralized approval with respect to any biosimilar product or any pharmaceutical product. Omnitrope is the first product that was approved by this Europe EMA as a biosimilar in 2006. This is the first product. One more thing I would like to highlight here, reference product or the innovator product. That is separate for each and every regulator. This is not mandatory that any product is approved by US FDA that is going to be approved, that is going to be considered by EMA as well. For the regulatory submission, you must take approval from the respective regulatory agency as a standard or as a reference product for your biosimilars. That is mandatory. These are the common things as per the EMA guidelines. It will take some time, but there are quality aspects, EMA standards with respect to your product in European Union. Few more things that I would like to highlight. Reference products must be active molecule, dose and administration must be approved by the regulators. Approval is very much important in this particular case, and EU authorization is required to be considered as a reference product for your biosimilars. You cannot take any biosimilar as a standard. Your standard or your innovator product must be approved by the regulators. That is must. Individually, this is important. If anything is approved by CDSCO or US FDA, that is not applicable on EMA. Quality and all those things are very common, so I'm skipping that part. That is assessment of marketing authorization application. In the case of biologicals, BLA is very much important. That is going to be considered in the US FDA. I will consider it separately. So basically, in the European Union, applications submit their biosimilar and centralized assessment with the Committee for Human Medicinal Product, that is CHMP, and CHMP designate the reporter and post authorization risk assessment committee, that is PRAC, for risk assessment, forming assessment committee for the comprehensive evaluation of your product. So that is, you are going to submit all your paper, all your dossiers, what I have explained in your ECDT format. That is, you are going to prepare a dossier. In that dossier, module 3 and module 4 is going to be very important. On that basis only, you are going to get an approval from the regulatory agencies. Once you get the approval for IND, that is investigation new drug application, then only you are allowed to conduct your research on human subject. And after that, once you collected your data of module 5, that is clinical data, then only you are allowed to file a new drug application with the regulators. So this is the overall process. Without that, you cannot convert your lab product into the industrial product or any marketed product. This is the common flow chart which they are following in Europe. I'm just taking the main points only. So now I'm coming to the little bit with respect to US FDA, and then I'm going to compare all this data, all these standards all together. So in 2009, the US Congress established a Biological Price Competitive and Innovation Act that is that is responsible for the approval of US biosimilars. In 2015, on March 2020, some of the products were deemed to the biological licensing application, that is known as BLA, and have been removed by the FDA. So basically in USA, as India, we are going to publish pharmacopoeias. They are going to publish orange book and purple books. And these books contains list of approved products in their country. So this is the totality of evidence that they require. And you can see this is the difference. In the original biological pathways, they are more focusing on clinical studies. But here they are focusing on analytical aspects. This is the major shift. This is the purple way. Already I have explained approval process. This is the common approval process. United States PHSA Act is responsible for the approval of biosimilars in United States. This is the common pathway. Common pathway for the drug approval process in India. Yes, I'm now going to focus more on Indian approval process because, as like other countries, India is having multiple agencies which are going to give you the guidelines with respect to biotechnological products. India In India, we are going to consider each and everything as a new product. If you refer the overall organization structure of SEDESCO, they are having 5 sections. First one is your drug, second one is your medical device, third one is your biologics, and fourth one is the new drug. So if you are going for the going to apply with the India, you need to file an application as a new drug, because biologics in India, in everywhere, they are going to consider as a drug. So if you are looking for approval in India, you need to file your application as a new drug, and they are going to refer your application to the concerned section. So when it comes to biological products in India, we are having multiple agencies for guidelines like RCGM, GEC. They are going to give you their guidelines according to their specific products. And finally, SEDESCO is responsible for overall approval. In India, only one agency is there which is responsible for the approval of any kind of pharmaceutical products, that is SEDESCO. If you want to approval or any license or anything in India, you need to take approval from the SEDESCO only. So, and yes, with respect to clinical trials, earlier Schedule Y was there of DNC Act 1940 Rules 45, but now they have replaced with the NDCT Regulation 2019. This is applicable for the approval of for the you can say for the conduction of clinical trials in India. Apart from this, these guidelines are based on WHO and ICMR guidelines, which is applicable for trial of any medical substance on human subjects. So these are the few common things, few common formats which we are using for the approval process, for the application process with respect to biosimilars in Indian, according to our Indian regulations. And Government of India has recently given a time limit for the approval process. If you are going to apply with any IND application with the Indian regulations, and if they are not going to respond to you within 30 days, it is automatically considered that they have given their consent. I'm a little bit fast. So now other like preclinical aspects, 45 days protocol in human approved by 45 days, clinical data and response analysis by DCGI within 90 days. So it's all time now, it's in within the time frame. That's why you can see your product are getting approved little bit fast as you're going to compare with last 10 years. This is the reason, because they have given a time frame for the approval process. Little bit apart from this, yes, one. This is the means USFDA product authorized product is not applicable in India. You need to take approval from SEDESCO separately. And few things are not available in India, like interchangeability. This is not permitted in India. Apart from this, all other things are there. CDER is there, that is responsible for the post-marketing license and other things that is already there. And they are very common things. I'm not going. They are the common and current trends in the biosimilar development. This is new approvals. Already they have received in 2024. There are the few things, few possible recommendation inclusions with respect to regulatory aspects with respect to approval of biosimilars by the different regulators. Yes, there is a difference in the documentary requirement in ECDT format in Module 4 and Module 5. If you are going to compare with MHRA, ASEAN countries and India, and as well as EMA. So there is a need of harmonization, because a person he may belongs to individual country, he is not able to understand, because we are investigators, we are not law agencies. So basically there is a need of harmonization in the process, that common requirements must be there. Enhanced pharmacovigilance system, that is very much important. It is reported around 8 to 10% only cases are reported. This is official data. Mostly 1 or 2% data is there which is in action. So that is very much important to focus on that. Clear guidelines on interchangeability in US and EU, this is there. But in India, this is not there. Comprehensive biosimilar biodata already there, and there are few manufacturing aspects, applications are there. I think these are the few future prospects. And herewith, thank you very much. Thank you, sir. If you have any questions, I'm happy to answer you. You can meet me after during tea break. I'm available.
Any questions? Yeah. Okay. That's it. No problem.
Good morning, sir. My name is Rashid, and I have two questions for you. For some products like medicines and to certain diseases or some vaccines, we hear that they get accelerated approval or fast track approval. So my question is, how is that different from normal approval, and what criteria do the approving agencies look at to decide if they have to do that? And to narrow it down, we can strictly CDSCO approval or approval from the FDA.
Very good question. Give the approval. You can see
Then from the other side, where the name brands might not be economically accessible for many people. So what is this tested about, and can we have it?
No. I have never mentioned that generic drugs are not good in quality. Never. They are equally good as your innovator product, provided they have completed their patent period. If you develop a product by today and if you get your drug patented by the patent office, you have a monopoly for 20 years for that particular product. After 20 years, that product becomes generic. Anyone can produce your product without paying you royalty. So during patent period, that person need to take permission from the patentee, or that second generic manufacturer need to wait for 20 years until that patent period is getting over. Because during patent period, that is the monopoly of the innovator or the patentee. You need to take the permission or you need to pay some royalty against that permission. I'm not never made any comment on the quality with respect to generic products. They're equally good as your original product when it was. Now, Government of India is promoting more on generic products only. This Jan Aushadhi Kendra and all, they are distributing and giving you the generic products only.
So my question is that is the marketing for like a kind of tablet capsule?
Pardon? Marketed formulation, that is, I think that is decided during R&D and F&D. Once the product is approved, you need to formulate the same product. If you want to make any change in your product, like a strength, dosage form, or the different, maybe tablet to the syrup, you again need to file ANDA and get it approved by the regulators. Before that, you are not. Once it is approved by the government, you need to produce the manufacturer need to produce the same product in the approved form. And that dosage form is decided on the basis of physicochemical properties of the drug. That in which form this is best available and it will give the best viability to the patient. And it is more stable. Stability is also a consideration. Nanoparticle is something there are two common as per our better understanding. We are having two therapies, two delivery system. First one is the conventional, second is the targeted. When like tablet, capsule, syrup, these are the conventional drug delivery system. Nanoparticles, transferosomes, nanosomes, they are all targeted drug delivery systems. So with this, we are trying to target and we are trying to improve the bioavailability. That's all. If you take tablet, it will take 30 minutes to reach in your blood. If you take injection, it will take 90 seconds to reach in your blood. If you take nanoparticle, it will penetrate deep in your intracellular part as well, and the overall dose will reduce. So each and every drug delivery system has their own merits and demerits. We cannot generalize.
I think I sort of different here because we can have such kind of discussion and now we will move to the next speaker.
Thank you very much, sir. Thank you, sir. Thank you, sir. Thank you so much, sir.
So our next speaker is Dr. Saman Fatima. And Dr. Saman Fatima is the assistant professor at the American Institute of Pharmacy, Amity University, Noida. And she is a passionate researcher specializing in open nano formulation within chemical characterization and pharmacokinetics and pharmacodynamics. She was awarded with Reservoir Award 2018 for the best paper from Asia. With expertise in medical writing, she has significant experience in preparing laboratory documents, publication, manuscripts. With such introduction, I would like to invite Dr. Saman Fatima on the dais and deliver her lecture.
Okay, good morning to one and all present here. First of all, I would like to thank organizing committee for inviting me here and delivering a lecture. And since I'm a complete humble product, so I really feel elated standing here on the podium and presenting in front of my mentors. Now, quickly moving on to the presentation. First of all, the title of my presentation is Designing Gamma-Glutaminal Cysteine-Coated Human Serum Albumin Nanoparticles for Brain-Targeted Delivery of Ropinirole, a three-year approach to combat cerebral ischemia and reperfusion injury. Now, quickly moving on to the introduction. First of all, it is important for us to understand how lethal stroke can be. So, due to arteriovenous malformations or because of brain aneurysms. Now, out of all stroke cases, since 87% of the cases are ischemic in nature, therefore, we had decided to work on ischemic stroke in our proposed study. Now, there is only one therapy that has been approved by US FDA for the treatment of ischemic stroke, which is recombinant tissue plasminogen activator therapy. And one of the major drawbacks of this therapy is associated with its primary objective, that is restoration of the blood supply in ischemic part of brain. This further exacerbates the ischemic injury, which is also called as reperfusion injury, due to increased accumulation of reactive oxygen species in calcium ions in mitochondria, that further favors the opening of mitochondrial permeability transition 4, that allows subsequent leakage of cytochrome C in cytosol, which further activates downstream caspases and finally results into mitochondrial-induced apoptotic cell death. So we are not only treating ischemic stroke, but also reperfusion injury along with it in our study. Now, I would like to point out here that there are two regions that are marked by ischemic stroke. One is ischemic core zone and the another one is ischemic penumbra. Now, cells in the ischemic core zone die extremely rapidly, that is within few minutes as a result of cell necrosis, but cells in the penumbra region die more slowly as a result of active cell death mechanism because of residual and collateral blood supply to this region for a period of 6 hours. So the primary objective of our therapy is to salvage this penumbra region as much as possible and as early as possible in order to inhibit the continued growth of ischemic core zone. Therefore, we had decided to repurpose ropinirole for the treatment of ischemic stroke and reperfusion injury by means of three-year approach. Now, this drug is already available in the market for the treatment of Parkinson's and restless leg syndrome. Coming back to its three-year approach, the drug, apart from being antioxidant and anti-inflammatory nature, was also found to possess anti-apoptotic potential that aims at inhibiting the opening of mitochondrial permeability for transition 4 and subsequently leading to inhibition of mitochondrial-induced apoptotic cell death. So since we know that cerebral ischemia is an interplay of cascade of events, so with the help of this selected drug, multiple pathways could be targeted. Now, ropinirole also has its certain shortcoming, the major being low oral bioavailability, which is only 50% because of first-pass hepatic effect. Also, I would like to stress here on the fact that various neuroprotectants that have already been utilized by researchers in the past for the treatment of IR injury have all shown promising results in preclinical investigations, but they all failed in clinical trials. And the reason being lack of safety and lack of efficacy. So this lack of efficacy could be attributed to the insurmountable blood-brain barrier that restricts the movement of drugs into the brain. Therefore, we had hypothesized to first entrap ropinirole into an FDA-approved protein carrier, that is HSA, which was also selected by us because of its myriad benefits, and then its surface functionalization with gamma-glutaminal cysteine, which was also selected as a putative ligand by means of molecular docking approach, and the result is already published in Journal of Biomolecular Structure and Dynamics. So with the help of this targeted drug delivery system, we would be able to achieve targeted delivery of ropinirole in the ischemic brain, which would help in boosting its therapeutic index and reducing overall systemic toxicity. So on the basis of these observations, our study was designed with the following objectives. One was fabrication and characterization of nanoformulation using HSA for the delivery of ropinirole to ischemic brain. Second is optimization of developed formulation for particle size and drug loading efficiency and the assessment of this formulation for its in vitro release. And lastly, comparing the efficacy of optimized with conventional therapy in male albino vista rats by using T-Macau model, which is transient middle cerebral artery occlusion model that mimics ischemia and reperfusion injury. So at first we had entrapped ropinirole in HSA by using simple dissolution technique. Later it was subjected to statistical optimization by using Box-Bington design of experiment. And post that it was coated with gamma-glutaminal cysteine by simple physical adsorption technique. So the particle size, as per DLS, for CRP-MPs of coated nanoparticles or gamma-glutamyl cysteine coated HSA nanoparticles entrapping ropinirole came out to be 74.74 nanometer. And this was later validated with TEM, which indicated sub 100 nanometer particles, and it was quite analogous to our DLS result. The surface morphology was analyzed by FESEM, which depicted spherical shaped particles with smooth surfaces. The zeta potential of CRP-MPs was also evaluated by DLS, that came out to be minus 25.7 millivolt. Next, we also evaluated the release kinetics of propionirole from RP-MPs and CRP-MPs by means of dialysis bag method, and we had compared it with free propionirole solution. The pH that was taken was 4 and 7.4 in order to simulate the physiological pH of acidic brain during stroke progression and plasma respectively. So as you can see in the graph, with respect to free ropinirole solution, where around 91% of the drug was completely released within 1 hour, in case of RPMPs and CRPMPs, a burst effect was observed within 1 hour, and this was followed by sustained release profile up till 5 hours. We then moved on to the in vitro cytotoxicity assay in order to evaluate the cytotoxicity of RPMPs and CRPMPs. So this MTT assay was performed in neuro 2 cell lines for a period of 24 hours within the concentration range of 5 to 25 microgram per ml. So as you can see, for the delivery of propionirole to the non-coated nanoparticles depicted by A2 and free rhodomine B dye solution as depicted by A1. Quantitatively, also the fluorescence intensity in case of coated nanoparticles was found to be 1.4-fold higher as compared to non-coated nanoparticles and 3.34-fold higher as compared to free rhodomine B dye solution. So this result could be accredited to the selective targeting of gamma-glutamyl cysteine towards glutamate receptors, that is NMD or AMPA, which are profusely expressed at blood-brain barrier junction, that might would have facilitated the enhanced uptake of rhodomine B dye in the brain by means of receptor-mediated endocytosis. We then moved on to pharmacokinetic study, where the rats were randomly divided into 3 groups. The first group was intraperitoneally administered with free ropinirole solution at a dose of 10 mg per kg body weight, and the second and third group were intraperitoneally administered with the non-coated and coated nano formulation at a dose equivalent to 10 mg per kg body weight intraperitoneally. So the plasma concentration versus time curve for all the 3 formulations were plotted, from which various pharmacokinetic parameters were calculated. So as you can see in the graph, the plasma circulation half-life of propionirole from RPMPs and CRPMPs was found to be protracted, which was even detected even after 24 hours. But in case of propionirole, free propionirole solution, it was not detected beyond 12 hours. So this could be attributed to the likeliness of intraperitoneal route to that of oral route, where therapeutic agents may have to undergo hepatic metabolism first before passing into systemic circulation. Besides, a minor IP injected also passes into thoracic level. Now, the entrapment of propionirole in HSA nanoparticles has substantially increased its bioavailability, which came out to be 2.8-fold higher as compared to free propionirole solution. Half-life was increased to 2.2-fold, and clearance was reduced to 2.4-fold. However, in case of CRP-MPs, the bioavailability was even increased by 4.4-fold, and the clearance was reduced by 3.8-fold. So this could be attributed to long circulation half-life of albumin in blood that might would have increased plasma AUC values, which by purely endorsing a therapeutic high concentration of propionirole in the blood for an extended period of time. And it could also be attributed to the sustained release profile of propionirole from RPMPs and CRPMPs. Now, the further enhanced bioavailability and reduced clearance in CRPMPs could be attributed to gamma-glutamyl cysteine modification on its surface that might would have deterred its recognition by reticular endothelial system. Coming back to brain, now next was our brain distribution study. So here we had observed that in comparison to the maximum concentration, which was 24.45 nanogram per ml, that was attained in case of free propionirole solution at 3 hours, 449.57 and 2141.97 nanogram per ml brain concentration of propionirole was attained in case of RPMPs and CRPMPs at 6 hours respectively. So this result is in corroboration to our confocal microscopy result, and it again could be attributed to the selective targeting of gamma-glutamyl cysteine towards glutamate receptor that might would have enhanced or facilitated the increased uptake of propionirole in the brain by means of receptor-mediated endocytosis. We then moved on to the pharmacodynamic study. Now, since we know on the basis of the observation that we have gathered from a pharmacokinetic and brain distribution study, we had observed that CRP-NPs might would enhance the clinical potential of the drug because of its increased brain accumulation and increased plasma circulatory half-life. So further studies in vivo model or T-Macau model was conducted with CRP-NPs in order to evaluate its therapeutic efficacy in a dose-dependent manner. So here we had, in T-Macau model, we had taken 6 groups. The first group was sham control group. Second was T-Macau group where the occlusion was performed for 1 hour and it was followed by reperfusion for remaining 23 hours. The third group was a standard treatment group where free roperidol solution was given at a dose of 20 mg per kg intraperitoneally at 6, 12 and 18 hours during reperfusion. And the deficit score, PNPs significantly ameliorated neurological recovery at a dose equivalent to 10 and 20 microgram per kg body weight. We have also evaluated the impact volume by a classic TTC staining method, which is used to distinguish between metabolically active cells that are given in red color and metabolically inactive cells which are given in white color. So as you can see in the graph, significantly higher impact volume was observed in T-Macau group as compared to the control group, but treatment with CRP-NP significantly reduced the impact volume in T-Macau subjected rats. Now, coming back to 3As, first of all, we had comprehensively investigated the antioxidant potential of CRP-NPs by determining various oxidative stress biomarkers. So, at first, we had determined the defensive antioxidant level in damaged penumbra tissue of T-Macau subjected rats. And as you can see in the graph, depleted levels of GSH was observed in T-Macau group as compared to the control group. But post-treatment with CRP-NPs significantly elevated GSH levels in T-Macau subjected rats. Similarly, an enhanced level of lipid peroxidation was observed in case of T-Macau group as compared to the control group, but treatment with CRP-NP significantly depleted the lipid peroxidation post ischemic stroke and reperfusion injury. We have also determined the mitochondrial ROS production by using flow cytometry. So an increased mitochondrial ROS production was observed in T-Macau group compared to control group, but CRP-NP scavenged reactive oxygen species production post-IR injury in a dose-dependent manner in T-Macau subjected rats. We have also evaluated mitochondrial membrane potential by using flow cytometry. So as you know, in asthenia, a derailed mitochondrial membrane potential is observed. So similar results was observed in our experiment as well. In T-Macau group, a derailed mitochondrial membrane potential was observed as compared to the control group. But treatment with CRP-NP significantly reinstated the mitochondrial membrane potential in T-Macau subjected rats in a dose-dependent manner. Now we know that the first and the foremost event that happened during ischemic stroke and reperfusion injury is the disruption of electron transport chain that results into truncated ATP production, which is again depicted here in our graph. A truncated ATP production was observed in T-Macau group as compared to the control group, but treatment with CRP-NP significantly restored the ATP production. Now coming back to the next A, which was to determine the anti-inflammatory potential of CRP-NPs. We had determined the pro-inflammatory cytokines like TNF-alpha and IL-1 beta that play an important role in neuroinflammation. So as you can see in the graph, an increased expression of TNF-alpha and IL-1 beta was observed in case of T-Macau group as compared to the control group, but treatment with CRP-NP significantly decreased their expression at a dose equivalent to 10 and 20 microgram per kg body weight in T-Macau subjected rats. We had also, because we know that the roperidol is a dopamine agonist, so it also induces dopamine-related neuroprotection. So we had also determined dopamine level. So a significant drop in dopamine was observed in T-Macau subjected rats. Now we had, during the ischemic injury, it is a well-known fact that BAX is translocated from cytosol to mitochondria and is responsible for attaching to the outer mitochondrial membrane and its permeabilization. That causes subsequent leakage of cytochrome C from the mitochondria into cytosol, which further activates apoptotic cascade and results into the mitochondrial-induced apoptotic cell death. So we had also studied the expression of BAX and cytochrome C in both the mitochondrial and cytosolic fractions. So as you can see in the graph, an increased BAX expression was observed in mitochondria and a reduced BAX expression was observed in cytochrome C in case of T-Macau group as compared to the control group. Conversely, an increased cytochrome C expression was observed in cytoplasm and a decreased cytochrome C expression was observed in mitochondria in T-Macau group as compared to the control group. But post-treatment with CRP-NPs significantly abrogated the BAX translocation into mitochondria and subsequent leakage of cytochrome C. Now, it is also worthwhile to mention here that our nano formulation or our optimized CRP-NPs significantly attenuated the progression of ischemic stroke and reperfusion injury with a boosted therapeutic index. That is, it showed similar efficacy at 1,000 times less dose as compared to free ropinirole solution in T-MACA subjected drugs. So at last, I would like to conclude by saying that our research work has reported sub-100 nanometer particles of gamma-determinant cysteine-coated HSA nanoparticles encapsulating ropinirole for active targeting in ischemic brain to encourage in situ activity and reduce unwanted toxicities. In vitro cytotoxicity assay established its safety profile. Confocal microscopy and brain distribution studies validated its targeting potential. Pharmacokinetic studies elucidated that it might would enhance the bioavailability of CRPMPs and also increase its plasma circulation half-life. The therapeutic assessment in T-MACA model suggested that CRPMPs could attenuate the progression of IR injury with a boosted therapeutic index at 1,000 times less dose as compared to that of free ropinirole solution by means of 3-year approach. So in future, we would evaluate the safety and efficacy of a nano formulation in clinical studies. So the data is already published in Journal of Biomaterials with an impact factor of 12.8, and we also patented our formulation. Thank you.
Any questions?
Goodbye.
My name is Jayati, and I wish to ask. I hold a lot of interest in biotechnology, and I wish to ask if this technology could be incorporated for Alzheimer's as well, because it also makes it a problem for blood-brain barrier and those for issues, and like creating biosensors specifically for that is like a long-term issue and with a lot of complications. So can this technology be modified respectively and incorporated?
Actually, if you study all the pathways of all neurodegenerative disease, so it is kind of similar only. So every neurodegenerative disease undergoes oxidative stress, there is neuroinflammation and apoptosis. So similar technology could be easily translated into Alzheimer's as well. And I'm sure that the promising results there also will be obtained.
Okay, this is Atul from JNU. My question is, I think that TNF-alpha has dual role. It involves in the damage of the brain as well as in the repair. So what is your say about it? Can we not use the stimulation of the brain when it is involved in the repair mechanism using some antibody?
So actually, the papers that we had undergone while we were performing the study, so the role of TNF alpha was mainly damaging in case of ischemic stroke and reperfusion injury. So it was mainly involved in neuroinflammation. So that is why we had taken it as a pro-inflammatory cytokine where we could assess the impact of anti-inflammatory potential of CRP.
So, thank you, sir. Our next speaker is Professor Deekshika Pandit. Ma'am. Okay. So next speaker is Dr. Vanika Gupta. Dr. Vanika Gupta is an assistant professor at the Center for Neuroscience and Nanotechnology, Jamia New Delhi. She has a specialization in nanotechnology-based energy solutions and environmental remediation. A former guest scientist at the University of Nuremberg is in Germany. She has authored 4 books, 10 chapters, and nearly 50 research articles. Actively involved in physics, BTC SUR, and nanomission funding projects. Her work advances nanoscience, energy storage, and environment sustainability. Welcome, ma'am, here. I request you to please deliver your lecture.
So thank you very much for the kind introduction, and I'm very much thankful to the conference organizing members Dr. Bharat Pankhi, Dr. Jagati, and Organizing Secretary in the Rajni, and all the dedicated and hardworking because the conference is organized being in regular basis, every 2 year or every year. So always a deep honor to be here to present the work. So with this first slide, it's basically the Center for Nanoscience and Nanotechnology at Jamia Millia Islamia. Yes, not coming. So basically this graph, that pie chart indicates the work that we have published in different areas, physics, chemistry, biotechnology, biochemistry. So based upon the nanomaterials and the funding agencies we have marked on the left side. So our work is mainly on the nanomaterials. So this whole work is basically comprising some publications and the research scholars PhD work is also compiled here. So here we have this techniques which we use for the characterization of nanomaterials like X-ray diffraction, FESAM, with respect to their morphology, crystal structure, band gap, surface charge, so as to optimize them for the given application. We used to work on the advanced nanomaterials, and we have metal oxides, conducting polymer, and the composites for the application. So application includes environmental remediation, energy application, sensors, agriculture, and quantum technology. And the work has already been presented at these platforms. And keeping in view of the sustainable development goals also, like clean water, clean energy, and then life below water also. And one was the most important, that sustainable goal for the healthcare sector. So these materials are used for given sustainable development goal achievements. So materials. So these are the 2D layer materials that we used to work, and these are the advanced nanomaterials, advanced than the graphene, which is well known. So here we have the WS2, Amexine, which is the latest known material, MOS2, MOSC2, and WSC2. So these are the images that we use to characterize them either by field emission scanning electron microscope or transmission, available at our nano center. Then we have the metal organic frameworks with ZIF 67, ZIF L, ZIF 8, ZIF 9. So basically these three-dimensional networks have cage-like structures which are used for catching the pollutants also. So whenever we need the high storage. Their composites 2D with 1D basically helps in reducing the recombination time and helps in removing the problem of agglomeration. So we used to make their composites with the metal oxides as well as the conducting polymers. Once we have the polymer with the conducting polymer, then it results in the increased conductivity that overcomes the issue of poor agglomeration and poor resistivity also. So here we have three-dimensional nanostructure also. When we have decorated these nanomaterials with the carbon quantum dots or silver or gold nanoparticles also. Basically, this will give plasmonic effects. So ultimately, from this we have exciton plus plasmon, which is the new science called plexitonic physics or plexitonic nanostructures, which are very common for the solar light harvesting. Further, this basically chlorophyll-derived carbon quantum dots because many of the applications involve efficient solar light harvesting, whether it's the photo supercapacitor or artificial photosynthesis, in which we want to have solar light absorption as much as possible. So advancements in nanomaterials are done in order to keep in view that in nanomaterials, we have a band gap in the visible range or in the IR range because if we talk about the solar spectrum, it's 4% UV light and remaining 46 to 47% is are infrared and visible. So our aim is to achieve these structures where maximum light harvesting is possible for carrying out solar light harvesting applications also. Here we have one more advanced nanostructure which is MXG. Yuri Gabrosti recently in 2011. Then we have lots of publications based upon this. So this is a new kind of structures where MX are different metals of the periodic table. and this is basically derived from the max phase. So we used to heat and then by top down and bottom up approaches. So if you look at this table that from 2011 to 22, that this MXGene has achieved so much of efficient applications. Either it's a photo detector, water purification, or MXGene nanomaterial for the biosensing applications. Even if you look at the publication details from this 2015 to 22, publication has also outnumbered and for the multiple applications also. Here in our case, we have used it for the sustainable development goal number 6 and for the healthcare sector goal. And it's popular because of the properties like hydrophilic nature, high electrical conductivity, large surface area, good electrochemical properties. So these nanostructure and review articles based upon this we have also contributed in the area. So using top down as well as bottom up approaches, hydrothermal is one of the bottom up approach which is used for the synthesis of this new MXene nanomaterial. So it basically involves the 36 hours of hydrothermal synthesis approach to prepare this. And once it is done, how to verification by doing the X-ray diffraction and the FE-cell fluctuation. That peak at particular position that 002, that is particular mirror indices that confirms the presence of MXene nanostructures. So these large interlayer spacings are important whenever we have the exchange of ions, especially in this say this new application of EV electrical vehicles where we have batteries where continuous exchange of ions used to take place. So we need structures which remains intact without any mechanical embrittlement when exchange of ions used to happen. So here we have the composites of imaging with the metal organic frameworks with conducting polymers as well as oxides to make it perfect for the given application. So here this iron-based nanocomposite of imaging because whenever we have iron-based composites, it's basically easy to do the recycling because we can use it for a number of cycles. Here this is for the vanadium pentaoxide, which is well-known material for the energy storage application. Here this is prepared by the top-down approach, which is the ball milling. So usually we prefer top-down approach under this ball milling and liquid phase exfoliation for preparing the nanostructures. Imagine zinc ferrite-based nanocomposite, and this is the EDEX analysis to check the purity of the material, that what are its constituents. Now, not only about preparation of the nanomaterial, we have to engineer it also for the particular. Say this is one of the example of zeta potential studies where this surface charge has varied from minus 12.5 to 24.9. As per zeta potential studies, the materials with the surface charge above plus 225 and below minus 25 are stable. They avoid agglomeration issues. Secondly, this is one of the point of zero charge studies which shows that, say, if we take the example of pollutants present in the Yamuna River or different water polluted bodies, pollutants vary in surface charge from negative to the positive and different pH also. So whether our material is going to affect or is sufficient effectively for the given number of pollutants is confirmed through the given studies. That this graph shows that given nanomaterial is quite good for the positive charge pollutants, but not for the negatively charged because point of zero charge is not supporting this fact. Second example of this is MOS2 polyaniline nanocomposite, where surface charge is carried from this negative to the positive, and both are present on the given composite in MP21 sample. So what happens that given nanocomposite of different proportions, that MP11, MP12, 13, and MP21, although you are preparing the nanocomposite, but what composition is best suited for the given? So it's basically determined through this surface charge studies. This MP21 is having both positive and negative charges, and further, this IPC measurements confirm that incident photon to electron conversion efficiency, that how much light is incident and efficiently conversion to the this electron. So this IPC measurements, this BW0, BW2, BW4, basically with reference to the pure pristine composite and W2 and W4, further their composites in different ratios. So it is clear that 2 to 20%. So almost 10 times increase in the efficiency of conversion once we have their composites. Next advantage is this through this HR time studies, which is quite evident that in pure MOS2 interlayer spacing is of the order 0.67 nanometers. But once we have its composite, has increased to 0.75. Basically, these layers, as you have studied in graphite or graphene also, there are van der Waals forces. So once you have increased this interlayer gap, the van der Waals interaction reduces and the scope for the agglomerate to form is also reduces. Hence, the given photocatalyst or given material is actively available for the diffusion or actively available for the degradation of pollutants. HR time studies and this schematic confirms the above mentioned facts. This TRPL, that is time resolved photoluminescence studies, basically that when light falls on any material, electrons and holes are generated. Similar to that of the photoelectric effect, for this Einstein got the Nobel Prize. So whether these electrons and holes are available for the formation of reactive oxide species, ROS species. So this is confirmed through the TRPL studies, which confirms that longer the time for which these electrons and holes are available, effective formation of RS species will take place. So for this MP21 sample as mentioned, here this TRPN time, average time is almost 16.71 as compared to the pristine samples. Now coming to some goal number 6, so these are the different target pollutants that are present in the water, either textile dyes or this mercury, chromium based copper heavy metal ions, this whole Ganga Brahmaputra belt. Then we have antibiotics also, which we used to throw them after their expiry, but this also lead to a one global challenge of AMR, which is antimicrobial resistance. So to overcome or to address this issue, again, the given materials are tested for antibiotic degradation. So these are some urinary infection drugs, some are kidney infection and rest also. So further, this terephthalic acid also, which is plastic. So through this, we are targeting the life below water. So this is also one of the issue that in Pacific Ocean, that whole plastic films are available. So through this, you can see the color slide that how change in the color for the MB, methylene blue, methyl orange, and then for chromium. The given photocatalytic materials is quite efficient for almost 90 to 100% degradation. Terephthalic acid, one of the plastic precursor. So through this material, we are targeting plastic pollutants also, which are non-biodegradable, finally leading to the biomagnification part. Health hazards, all of us are familiar with this issue. And using a magazine-based or nanocomposite, we are able to degrade it up to 97%, and with this optimized nanocomposite. Further, this some materials are not only work in the daytime, they are 24 to 7 photocatalysts, also called round-the-clock photocatalysts, because in this ROS species generation is throughout the day as well as night. So this EPR studies, this is which confirm the formation of ROS species. This is also observed in MOSC2 polypyrrole nanocomposite, which used to have lone pair of electrons that are present even in the night time to prepare RO species and to carry out the degradation process. Finally, with our work, we are planning executed this lab to industry that collecting the textile wastewater and finally analyzing the different pollutants present in it. And accordingly, once identification is done through the database, then specific or customized nanocomposite is prepared for the degradation of pollutants. Colorimetric detection is one of the easy ways to detect it. Here also we have tried to detect the mercury. So mercury, if you compare with the rest of the heavy metal impurities that exist near this mines area, this mercury even 100 times lesser is still the same toxic. One of the highest priority means topmost pollutants. So we have done is selectivity test and interference test also using a magazine-based nanocomposite for the colorimetric detection of mercury. So it has been tested for the real samples also. With this, the work that has been presented in these slides are summarized in these journals published by us. in photocatalytic wastewater treatment for using given materials. And then we have this energy sustainability and biosensing used materials for these applications. These are the projects and fellowships that we have got to carry out the given work. These are some books as mentioned. And this is our BRICS project. We have the project with the Russia and the China to carry out the given work. All right. So with this, I thanks the organizing committee, all the students, scholars, and eminent
which has property. The character ones, how they are actually different from the anti-microbial. And as your grandparents were also using for agriculture, how they can be used.
All right. Thank you.
And to later nanoparticles, later nanoparticles, layer one. How these layer nanoparticles are different from these and the advantage.
All right. So for the agriculture part, usually we have preferred say zinc oxide, which is a micronutrient required. Here also because of this large surface area, or but still we need to uh, check the toxicity part also, because once the diameter is below the some critical value, it becomes toxic. Yeah, usually below this 20 to 30 nanometer below diameter, nanorods are not good for the agriculture application. So we have one book, this nanoscience and plant soil system, in which we have mentioned the details of this, and one paper in Frontiers in Microbial Technology, where this zinc oxide interaction with broccoli plant is discussed. And further in crops, this has been implemented at large scale for 3 to 4 cycles, and then it has been observed that increase in the for first at the root level, see how much is increase in the germination rate, root length, shoot length, and this other activities related with this. Second point is this, although you people also study this silver and copper also, but silver being very expensive, and zinc oxide large scale production is also feasible simply by doing the thermal decomposition of zinc acetate dihydrate at different temperatures. One can obtain zinc oxide nanorods of different diameters. Point here is that for most of these applications, we require large scale production. So we need the nanomaterials, like whether this agriculture application is or is environment, this ocean cleaning or seawater cleaning. So for this agriculture part, zinc oxide and toxicity part is important. Then also these four more factors, this ROS stress, oxidative stress, and other also be considered. As far as this antimicrobial and 2D layer material concept is concerned, this 2D layer materials is basically one basic concept is the classification of nanomaterials in different categories. 1D, that quantum dots, nanorods, or this carbon nanotubes under this wall, or 2D materials, which is graphene is well known, but WH is a newly known material which is quite biocompatible. So for antimicrobial application, we used to go for the biocompatibility. Even from the earlier times, this washroom handles are used to be made up of copper, or we used to drink from the silver glass whenever we want this germ-free water. So, but in the nanoparticle form, they are much more effective. So, antimicrobial activity, we used to go for the oxide copper doped nanostructures. Again, the work has been published and for multiple applications, and further tested by MTT test or this count test. And further one FSM micrographs also we have put in, where this cell lysis used to happen, because these nanomaterials are very sharp, so they are able to pierce the membranes of this E. coli and S. aureus bacteria. So active good for both gram positive as well as gram negative bacteria. So this antimicrobial part is done. Then this agriculture part and 2D nanomaterial application is here. The confinement is in the along the thickness dimension, provided further that these nanomaterials like graphene, MOS2, WS2, and this latest material, amexine, although quite expensive, that's why not good for these applications, but have the potential. So say 80,000 rupees for preparing one sample. Used to, we have to spend so much. good for the lab part, but not for the actual application. So here, because of this high conductivity and ability to disperse more, poor agglomeration means issue is also resolved by having this 2D materials composite. And further surface charge optimization is also achievable with the variation in the number of layers. So how this 2D materials provide us the handle to control the band gap also as well as the agglomeration issue. Third is the surface charge optimization as per the pollutant present in the water.
Now when we move to our next speaker, Sigma, she is a renowned researcher in biomedical sciences, focusing on innovative diagnostic tools. Her groundbreaking work has led to the
Kindly request the chairpersons and our present secretary to please felicitate our moderator, Dr. Saman Fatima. I now request the chairpersons, all the esteemed speakers, and our present secretary, Professor M.Z. Abdin sir, to please join the dais for a group photograph. Ask those presenting posters to please head upstairs for their presentation. Also, I now warmly invite all the delegates and participants to join us for a short tea break. We will resume after 10 minutes. You can also support the poster session upstairs. Thank you so much.
international scientific bodies, including the New York Academy of Sciences and the American Society for Microbiology. So, and I would like to invite our second chairperson, Dr. Saima Wajid. Dr. Saima Wajid is Associate Professor at the Department of Biotechnology. She is an elected member of the National Academy of the Medical Sciences and made remarkable studies in the cancer biomarker studies, discovery and herbal-based nano formulation for cancer therapy. Dr. Wajid has published many research papers in international report journal. And one thing I would like to mention, which is not written, that she is the lifeline of our department. And
Ah, so, good afternoon, everyone. And let me congratulate Professor Ranjay Walin sir, who is the organizer secretary of this international conference, and Dr. Saima, and Dr. Who is the other person? Yes, Dr. Omara, and the convener, Dr. Javed, for conducting this conference, and the volunteers and other teachers, staff members, those who are involved in this organization of this conference. I am basically from Jawaharlal University. If you see this sign, this signifies the sharing of the knowledge. This also indicates the formation of a bond actually, very dear to our former Prime Minister Pandit Nehru. Our lab is located on the fourth floor, and JNU is the home of Farm Caviar, a national bird peacock, and the Nilga. I'm going to speak on vaccine development from journal to genomics. If you think of first experiment, probably the known first experiment, the vaccine development, English physician, Edward Jenner, which he conducted in 1796. He uses this small protein bomb named FIP. For this purpose, he has used copper snowflake and inoculated into that bomb. That bomb. Exactly after 200 years of first experiment 1796, then in 1995, the first genome of one bacteria, that is Haemophilus influenza, was out. That was published in Science. The size was around 1.5 MB, 1.8 MB, typically 5% of human genome. And the second was again published simultaneously in the same year, same journal in Science of Microplasma. So till 1995, till today, 200. You know, so what is the main purpose of the genome sequence? We will also know when you look at genome sequence that how many genes are basically involved in the metabolism. How many genes are actually involved in the virus vector, which are helping the bacteria to do the infections? How many genes are involved in the attachment process, helping the bacteria in the attachment? How many genes are helping the bacteria in the invasion process? Means tissue level. Suddenly we are doing a lot of coughing. So the bacteria which was sitting here silently, group Streptococcus, or Staph aureus, or Haemophilus, why they become so much infective in nature? They were sitting silently during the winter time. So maybe some genes, some protein are helping identify what inside the cell. So when you go to a doctor during this time to say that you are having this topic, doctor will ask you to see character. May the bacteria chew a particular area, it means this is the mammalian cell, the bacteria is coming, so their live protein comes out, the surface protein, other protein again have them to attach. If bacteria is not attaching, they are not going to invade. That's for sure. They can also do the infection by releasing the chemical which is called the toxins. So in a way, using the serum analysis, you immediately narrow down the search from 1000 genes to 200, and out of 200, any surface protein like this is the bacteria, this is the surface, some are exposed by this, some are embedded in the membrane. So you immediately discard them. So from 3000 you can come down to 200, from 200 you can come down to 50. 50 genes you can clone with the help of PCR techniques, and then you express the protein, inject it into the mouse or into the rabbit, get that anti-serum reaction, monoclonal antibodies, you do that utilization experiment, and then you will have the vaccine candidate. So you think of this COVID era from 2019 to till today again, they were about and they were chasing the genome sequence of the virus. Why? They want to use the pan-genome approach. They want to see the surface protein which are helping the virus in the attachment process. So as soon as you identify using either this technique, proteomics, or you use in silico, you do the DNA microarray, you do the high throughput screening means, if you do the identification of the environment of the protein or the gene, you do the expression pattern, checking the expression pattern in the if you want high throughput screening, and then you have a vaccine development. So the ideal search of a vaccine candidate in a database is known as reverse engineering. When I say database, means genome sequence database. 10 years back, 20 years back, we used to do the search against the whole NCBI. Nowadays, lot of genome sequences are out. You can see the search against the particular organism, how much bacteria, virus, and then you will identify in a very rapid manner time. So this is, the conventional approaches which we were using in order to develop the vaccines. You culture the bacteria, collect the sera, identify the genes involved. By the time you do all these processes, it will take 5 to 15 years. Long time. If you think of a COVID vaccine process, that will take 1 year time because they have used this reverse vexillology approach. Systematic search after the genome sequence of bacteria, and this also we call pan-genome. What is pan-genome approach means? If some virus or bacteria is spreading either in Delhi, Lahore, Bhutan, wherever it is spreading or prevalent, I have to consider a particular conserved vaccine candidate across the region. Whether in India, Pakistan, Nepal, Bhutan, Germany, Japan, wherever it is, then only I will say that my vaccine is universally applicable. I want to make a vaccine which is applicable throughout the continents or across the continents. So this approach we call pan-genome approach, and every bacteria, just for the result say, the sitting here, every bacteria has a core genome which is 95% similar within the same serotype, and 5% that differ, or 1 to 5% we call variable gene. So if you have one particular vaccine candidate which you have identified in a prevalent bacteria, prevalent in Delhi region or NCR region, you can do the hybridization experiment, dot block, and the same vaccine candidate in a prevalent bacteria circulated in another region or another country. Therefore, you can conclude that same vaccine can be applicable in another patient in case of another country. So this is the bacteria. I'll give you a very good example of this. It's not going back. This is not. Which one? Okay. This is the bacteria gram positive beta-amyloid. Group B is a facultative amount. Basically, it lives inside the mother colorectal. So when mom is pregnant,
third trimester, okay, 34 to 37 weeks. Every doctor in USA, I'm not talking about India, Pakistan, Nepal, Bhutan, ask you to identify whether mother is colonized with this bacteria or not. Reason being, when the baby is about to come out from the birth canal, okay, these bacteria moves from the colon to the newborn baby where it can cause pneumonia, sepsis, meningitis, and the baby can be able to we call them means late onset disease. The babies are 7 to 9 days old. That is we call it late onset disease. So if you're not going to detect this bacteria in a pregnant woman, when the third trimester, that is 34 to 37 weeks, chances are very much there that your baby will get the infection when they are coming out from the birth canal during the birth process. So this test is mandatory in USA, in Germany, in Japan, in UK, many parts of developed Europe, basically. Economically, health advice, even there this bacteria exists and causing problem. But during the surveillance process, this was implemented in the 1970s, the birth rate, 1,000 birth rate, the death rate of this due to this bacteria was 0.71. When the surveillance happened, when the government started to the hospital to detect this bacteria, thus the rate of infection which has come down from 7.7 has come down to 0.13. But this test is yet to be conducted in Indian subcontinent. So what we have done, we have taken this bacteria which moved from a colonized mother to the newborn babies. We have identified this bacteria. Just for your information, this bacteria basically it causes 20 lakh deaths per year actually. And out of 20 lakh deaths, 0 to 4 years, 3 lakhs that happen because of group B streptococcus. Pneumonia also caused by strep pneumoniae, that is primary. Secondary pneumonia is caused by group A and group B also. And this death we highlighted this problem in our article in vaccine which was published 10 years back actually. And less data or no data is available from the Indian population because there and this test is not mandatory in India. All the doctors, I don't know how many doctors are sitting from Jamia Medical College. They said that in India, this bacteria does not colonize the pregnant woman. But when I did the survey actually, along with my PhD done, from 2004 to 2015, we found out up to 12 to 15% pregnant women were having this bacteria. But in mother, it is doing nothing. When it moves from mother to the newborn baby, it causes disease. And when others said this is the when they get the opportunity, mother is lying down on the bed, about to deliver the baby, and doctor says push, push, 1, 2, 3. The baby gets, when it's coming out, the bacteria gets formed from stimulus oxygen and then become infected and comes into the fluid of the mother and transferred from fertilized mother to the newborn baby, where it causes pneumonia, sepsis, and meningitis. So we hypothesize that during the process, the same bacteria will not utilize oxygen, but during the birth process, they become infected. So we correlate this by using a system called DIVAS. I told you already about this. This vaccine, which is made actually in USA, is not be applicable or will not be good to be used in case of Indian population because we do not know whether the vaccine formulation includes the prevalent group which is circulating in Indian population or not. Basically, this vaccine was made by my former boss, which was based on a type 3, group B. Type 3 is not present in population. Even I have collected samples from Karachi. It is also the type 7 actually or type 1A. So even if I am not considering the vaccine candidate which is common between here Parasingulum part. So they are not included that we have pointed out in our article that was published. And moreover, most of the area always something happened outside the medical settings. So we don't know if the mid-gestation abortion happened because of this bacteria, because it was mentioned in the medical books that this bacteria, when infected or colonized the mother, it can chew the uterus cell line, and which results in the mid-gestation abortion, which we call from pre-rupture of membrane. So if this is the uterus, for example, and the baby is attaching itself, like in the fetus, bacteria move from colonized mother, which is there in the vagina, cervix, uterine cell wall, and it start eating the mid-rupture, the pre-rupture membrane happen, and this results in the gestation abortion. So we don't know this, whether it happened because of some physical problem or it happened because of bacteria. So this we have highlighted problem in our article. Whether me, you or anyone, everybody wants beautiful and healthy babies. In order to save the babies from this bacteria, every woman should ask her doctor who will take the presence of this bacteria. Whether or not it is colonizing mother or not. But it is true that we found out it is present in 15% of Indian population. So what happens if someone having the strep B? Spread of this bacteria can be stopped using penicillin shot 4 hours before the delivery. Why 4 hours? It will take almost 2.5 hours to grow that particular area in the stomach. Penicillin wash in the vaginal or the colorectal region so that it can be killed. Secondly, what can I do to protect the baby? Go to a doctor, OBGYN, obstetrician, gynecologist. Ask your doctor, please, and your husband should also go along. And tell them, I want to do testing for the presence of this bacteria. Then only you can stop the spreading of this bacteria, which will cause havoc in the newborn baby. Babies cry because it's very hard to take the food fast because their lungs are infected with bacteria. It's very hard for them actually. They can't. And in the market, the Q-tip is available. Q-tip is nothing, it's a tip which is getting the cervix sample and then detect the test is very, very simple. It's not painful at all. It can be done within 500 rupees. In any normal medical setting, this can be happen. As I said, presence of the GPS bacteria during the pregnancy is available in USA, England, Ireland, or Italy, but no test in Indian or Indian support. So these are some of the pictures we have taken. If you see this bacteria infection caused by this bacteria, this baby, this is a rupture, PRM, pre-rupture of membrane. This one, and if you see this particular, this one, these two babies are not healthy. Spilled of water actually, pneumonia, lungs, in the lungs, the water is there. So this causes pneumonia, sepsis, pre-rupture of membrane, premature birth, and babies are sick in the pot. Very hard. Actually, if some babies, they are 0 to 7 days old, 0 to 30 days old, when you put the baby on the belly of the mother, it's a beautiful relation. Baby can move from a belly and start to. But if it is infected, they become the they start crying, they get fever. Ideally, baby should not get fever at least initially for 30 days because of the transfer of good antibodies with gut microbiome, let's say lactobacterium, from a mother milk to the newborn babies. But if the baby gets some fever, it means something is wrong and this bacteria is there which is causing problem in the newborn babies. So what we did basically, we have designed this particular instrument, we call it, We have simulated the condition. Oops. We have simulated the condition in here, and if you see in this, we have pumped the oxygen 0%, considering the bacterial non-invasive nature inside the mother. And one time we grow the bacteria 0% and start infecting the mammalian cell line from the vagina, from the cervix region, and then we find out how many bacteria 0% oxygen whether they are invading. Again, we grow the bacteria 5% oxygen and we allow them to insert within mammalian cell. Here they are sitting, and we see the difference between the two. As I said, when the baby is coming out, they get the 5%. They become infective in nature. So we find out that at 0% oxygen, these bacteria were invading very less as compared to the 5%. And this pattern was similar in case of cell line from either cervix or from vagina, from uterus, from lung, or either from the breast. A similar pattern was observed. You see this. Now we also found out that this bacteria at 0% are not invading. This is mammalian cell, and these are the bacteria at 0% are not inside, but at 5% oxygen they are inside the cells. That proves that bacteria are invading because of the oxygen. Some genes go after they are having the invasion process. The in vitro studies, when you know, once published to journal, you perform fuse or in vitro studies. So pups were taken and we perform the survival and we found out that 5% oxygen, 10 to the power 6 bacteria all died. 10 to the power 6 died, 10 to the power 1 died. But when we took out 0% growing bacteria, 50 to 60% survive, pups survive basically. That shows there is a correlation between 0% and 5% and in the infection process of the bacteria. This was published a long back. Then again we found out there is a myth actually. They said that group B does not. We found out that type 5 of group B streptococcus, they infect the adults also and results in the death of the patient older than 60 years, 65, when there is a bypass surgery, along with the group A streptococcus or strep pneumoniae. Or multi-organ failure happen due to the fact that they come into the bloodstream, which we call septicemia. So we found out using a proteomics approach, if you see this particular proteins, they are appearing if one speaker is good in proteomics. So if you see, this was done long actually using all the top as well as ESI and SCLS. You see these protein bands are not in this condition. This is 0%, this is 5%. Maybe these proteins are helping in the infection process. So what we did, we cloned all these genes, we injected into the mice, collected the serum and performed the neutralization. For example, if this is mammalian cell and this is the surface protein, block this, neutralize this, this is the antibody, polyclonal which are collected and injected into the mice. This should neutralize the surface protein of bacteria, should not invade. Means it is not going to attack because I have blocked this. If it is not attached, it is not going to invade. So we found out that up to 95% inhibition was stopped, means the adherence was stopped when we use this particular protein called phytide proteins. I will show you. And this work was done by my PhD student, and she was actually raised down to but did a lot of work with me. And we published this paper in Journal of Biological Chemistry, 2013. And a company actually approached us, Novartis, and they wanted us to have the data from Brazil, China, and India. The reason being money, honestly speaking, along with the caring of again this particular organism because these three countries have the largest population. So we collected the data from China, Brazil, and India, and we found out that these spike protein, as I was talking about, having the bacterial attachment process. We actually chop this out. This so bacteria actually stop immunity. The mammalian cells. So this we have used the dot hybridization. We found out the similar protein candidate used by Harvard people to make this vaccine. The same fly was also present in case of Indian population, prevent bacteria. Therefore, we concluded using the DNA microarray. So if you see this particular arrow showing the presence and conservation of this particular fly protein in case of GBS circulating or are present in Indian population. And we concluded that the fly protein used as a vaccine candidate in case of can also work in case of Indian population. This vaccine is approved by the FDA in 2013 and now it is in the market. Another thing is Mucus streptococcus. Again, a streptococcal. The problem with streptococcal is it has to the vaccine so far again this particular bacteria is not licensed. Reason being the circulation of particular serotype is different from Delhi to Chennai or from one another in the local. Same problem. But the vaccine made by Jim Day, University of Utah, or another group, Michael Loeb from US, from Australia, Brisbane, QIMR, Queensland Institute of Medical Research, they have not considered any circulating or prevalent serotypes either circulating in Indian subcontinent or for that matter SAARC countries. Even they have not taken under consideration circulating serotype from Japan or Korea. Both are developed countries in terms of medical facilities. So what we did basically, and also let me see, go ahead. Jindal has made a vaccine based on an M protein. M protein, though it is conserved across the GBS serotypes, but it has a homology with our purpose. If I make a vaccine, if I use a protein as a vaccine candidate, if I inject into our body, there will be a cross reactivity with the heart muscles and there will be a problem. As it is, this bacteria causes rheumatic heart disease, rheumatic fever. More frequency rheumatic fever would cause into rheumatic heart disease. Okay, that becomes a problem. 6 lakh deaths happen every year because of this bacteria. 70 crore people are infected with this bacteria. So, a child which is going to the school, they get 13 infections every year, and out of 13, 6.3 happen because which we have test. Whether it is happening with scrapping or happening because of hemophils or because of staph aureus, because group streptococcus. So what we did basically in this case, I talked about this problem with M-protein based vaccine. JAD is also there. But if you think of, forget about US, forget about Sweden, think of Africa or Sub-Saharan countries, Uganda, Ethiopia, Namibia, Nepal. Think of Sub-Saharan countries. Whether this vaccine will work or not, honestly speaking. So we found out a vaccine candidate, okay, which we found that it is conserved across the 98% circulating serotype throughout the region, throughout the world, whether in Middle East, whether South region, whether USA, or whether South America. So that this vaccine can be universally applicable. Okay? After identification, this is just for your knowledge that this bacteria causes both suppurative and non-suppurative infections, pus forming, non-pus forming, and this is important. We have selected this particular protein which is surface exposed. This one I'm talking. This one I'm talking. So what we did actually, we have run a 16,000 protein. So nine sequences were out. We hybridized that and found out how many are common between serotypes circulating on a protein across the regions. And this work was done by former PhD student Avinash Sharma and was published in Journal Proteome Research. And he selected basically using single ID server 147 from one from Timber, and using proteomics we selected 120 in one case, 148 in another case, and something 112 in another case. And from all M1 and M49 circulating serotype, we selected 52. All 52 genes of protein were cloned, expressed, purified, and injected into the mice in order to collect the antibiotic. And this is just a picture showing how dangerous this bacteria is. They're invading the mammalian cell, actually in this state, hypophilic state. Then you get the couple. And we selected a protein involved rational variants, involved in the laser, involved in the adherence, as well as surface exposure. There were many proteins which were helping in the laser, adherence, but to the mice. From 10-fold to 20-fold it can go, and that shows that protein is really stimulating the formation or reducing the hormonal response. And we also did the optional phagocytosis. We collected the serum, well, the antibody, and then we put our bacteria. Invasive in nature, we found 90%. 70% bacteria were killed. Another important, I might take this to one. Another important thing when you make a vaccine is whether, in addition to humoral response, your vaccine should also induce cellular immune responses. So we collected the sera and we determined using the fact analysis was done in Jamia. Ma'am, very important. So we found out that it is producing many more fold of the gamma interferon as compared to controls. 225 fold actually. And the function of gamma interferon that it comes at the site of the infection, ask the other army of cytokines and fight against the bacteria which are prevalent in that particular region. In addition to Th1 responses, IR70 was also found out expressing more of formation was more there, at least 24 there. And we found out that this particular protein when injected into the mice not only stimulating the human responses but also the cellular responses, which were helping the bacteria, helping the mice in order to fight against this. For this purpose, we proved by taking the invasive strains from, I told you, from Pakistan, from Middle East, from England, from US and from India. And this is the survival. As a Kaplan map, we found out 70 to 90% survival of the bat in the mice when injected against this particular infection. This work was done by my PhD student, Uday Sandula, and was published in the Journal Nature Communications. And this is just a mechanism when antibody are there, they are going to block gamma interferon, and then when they block it, bacteria are going to attach the mice survive. And the fellowship was awarded to my former PhD student, now I postdoc in very famous lab, University of California, San Francisco, IUPAC Fellowship, Paris. Pooja recently completed using this vaccine formulation. Another trial we are doing, just completed that using LM actually. Rupesh is working in the development of inhibitors. I just had a talk with Dr. who has done pharmacokinetics studies. And this is me. This is our publication in Nature, PNAS, Plant Cell, and Nature Communications, and Science Medicine, where we have developed inhibitors against this Metropolitan society means skin causing bacteria. So skin cause means if it is infected, this is is going to go inside and it will take at least 3 weeks for the treatment process. It is to start getting the flesh. And lot of funding, lot of acknowledgement from Larry Balati, Dennis Caspar, the whole universe, the had actually was made the adjuvant for COVID vaccine, safety. And Philip Glaser, G.H. Chatwal, Professor Stout, Professor Prasad, Rakesh Bhatnagar sir, lot of funding, Indo-Israel, UGC, DBT, Indo-German, ASM, Tata Innovation Fellowship. And this is my group. When bacteria was interacting with the mammalian cell, mice were also interacting between them. He interacted with this girl and got married. He interacted with this girl and they got married. This is when Pooja was awarded PhD. And when we published in Nature Communication as well as in Science Astronauts in Holy was celebrated in big way. And this is not my idea, this is the same that was published the first program. And I always thank my two twins, twin daughters. who have
Memory B cells. If you look for the memory B cells, because this is the extracellular pathogen and mostly the animal response will be working. So what is the status of memory B cells?
he recently
T cells.
long
Long-term memory in T cells. So since it's an extracellular pathogen, human will be working more.
that is both. So I know why you are creation of
Right.
because
Right. One more talk I have, so you have looked for the oxygen, like impact of oxygen on the growth of this bacteria in the lung and because oxygen is less in the vaginal canal. So what about the pH? Have you looked into pH also? Because there is a big difference in the pH. pH is very low in vaginal canal compared to the lung.
CDA.
Yes.
So we can try the pH of yeah. So we have the pH 7.3 so that if you change the pH actually, it circulates pH requirement. And then you take the first media in addition to the you found out that the bacteria dividing both. When you yeah, the rate, the CDM. Bacteria were divided at every 1.8 hours. Fast growth actually. And they were invading more. When we have actually used 0.6 moles of the media, they were coming every 8 hours. And they were invading something like that. So when I showed you that, dividing fast. They have
No, sir. Thank you, sir. But actually, is there any proteomic study on this, like difference in the pH or not? I want to know. Okay, thank you, sir. Thank you very much.
Yes, please. Yes. During during when? Because inside the mother, this bacteria is doing nothing. Okay, that's what I said. It is a opportunistic pathogen or a frank pathogen. In mother, it is doing nothing because it is a facultative arrow. Whenever there is a powerful stimulus of oxygen, it start infecting the newborn babies. So the caution is well taken in the sense when baby start feeding actually, along with the mother milk, there are some gut microbiota transfer from mother to the newborn babies. IgG transfer along with the microRNA actually, and which also encoding the presence of many gut microbiome helping bacteria. So along with the IgG antibody, gut microbiota transferring from a colonized mother to the newborn babies would help against this. So maternal antibody, yes, it does help. Very good question. So question arises actually when we were doing this vaccine treatment testing in USA when I was a postdoc. So question was whether we can treat the mother with the vaccine or with the newborn babies. The idea was because from a colonized mother, when we inject the vaccine, ideally during the third trimester, because the birth would happen during the third trimester immediately after 36 weeks till 40th week. So we were expecting if you do the vaccination in the mother, the antibody would transfer from mother to the newborn babies, but not by transferring the blood. It never goes like that.
Thank you, sir.
Oh, thank you very much for your time.
Speaker is Dr. Mohammad Irfan Qureshi. He is a distinguished scientist specializing in plant-based vaccine development, plant genome editing for disease, and molecular responses to abiotic stress. He is a governing council member of the Lead Science Foundation India and has led pioneering work in botanical survey of Jamia Millia Islam, identifying 200 trees with QR coded information. A recipient of prestigious awards like the ISA PMT Visiting Facts Program, BSc Fast Track and Science Commission, and Maulana Azad Award for Excellence in Leadership. He continues to drive innovation and plant biotechnology. Stay right, Professor Fresh, for his talk. So we have
Hello. Right? So this is for forward and then steps. This is the statistic को आ रहा है इसमें डिस्टेंस होता है वहाँ से क्या नहीं कोई भी क्या यूज करें ये वाला भी क्या ये तो ठीक है हैंड माइक बस हैंड माइक दे दो कोई बात नहीं इसमें एक आ रहा है इसमें डिस्टेंस बिल्कुल तीन का अब मैं तीसरा कहाँ से लाऊं आप कर लीजिए इसका यूज हाँ सर Well, good afternoon to all of you, and I'm really grateful to the organizers for inviting me to deliver a talk. And this is always good to be here in Jamia Ahmed because I'm a child of Jamia. This is
matter of pleasure for me. And Professor Abhi has been my co-supervisor, PhD co-supervisor, and Professor Muhammad Iqbal, he was my PhD supervisor. Although as Sir Johari, I really enjoyed his talk, and he mentioned that I am from the field of proteomics, so I am not much old in this field. So I did not ask any question from Professor Johari, and I expect the same from him. Anyways, I'm going to talk on the some important aspect, and that is related to the vaccines, mainly vaccines against the viruses. So, uh, the topic is harnessing the potential of the plants to to produce antigens, which we call as the vaccines, to treat or to use them against viruses. So, this is the work which has been done by one of my students. She is Sadia Kamal, sitting over there among you. So she has conducted this. And this is a figure that you have seen in media and at so many places. So this basically demonstrates something very serious because this is associated with a lot of deaths that is occurring on the planet Earth because of different diseases or the pandemics. So, if I go to the specific each and everyone, it will take a lot of time, but this is just to show you the gravity of the deaths that they are causing because of number of infections. And if you go to the black Black Death, which is called as bubonic plague, and you will find that it has got 200 million deaths, more than that. And then I will directly come to the HIV/AIDS, which has been talk of the town because it is recently occurred disease which was caused by HIV. And then the latest one, the most drastic one that you have gone through, and which has impacted your studies also, and this is none other than the COVID-19 virus, which we call as SARS-CoV-2. So all this is here, but if you go a bit back, there was some other viruses were there, which were making the life difficult, although they were not that means lethal, but they make the life difficult. So we have done some work, and I will be presenting some of the slides. So this is easy to show you here the history of the pandemics starting being recorded 1890, 94 onwards, and now we are here somewhere 2019 and above, where there are more than 27 million people have been killed because of this disease. So how to deal with the pandemics? And this is very simple, as Professor Jordan told you, that this is vaccination. And just by the vaccination, you get the immunity, which is being either by the introduction of the viruses or their proteins, and then vaccines, which are one of the most powerful tools preventing infectious diseases. They work by stimulating the immune system, you know the mechanism, and then through supportive care or by using antiviral medications. In fact, if you don't have antibodies against any viral infections, you need to get some medicines. And then we believe that they are going to work. If they are going to work, you are being protected against that infection. But this is always good. You have your own immunity against the viral diseases. So antibiotics are not much effective against the viruses. We know that also. So this is a powerful protection they are providing to us, and they are also adding to the herd immunity. Not much, but yes, because they help us not spreading the disease further. So this is vaccination is a crucial public health measure that protects individuals and communities from preventable infectious diseases. And you see the importance of the vaccines through this slide, which basically this is the data which was collected by Johns Hopkins, and you see now they have stopped collecting this data October 23 onward, but still you see they have shown that at least 13.3 billion doses of COVID-19 vaccine have been administered. So this is the global vaccine timeline stretched beyond 2023. So this was basically a projection which was given before 23. Now we have achieved this, and you see that with the passing time, what is the frequency of the vaccination throughout the globe. So this is basically the coverage target for some specific disease that is a very common combo of disease with all the vaccines that we are using. This is called as DPP. So this is basically the coverage throughout the world. So just giving some glimpse that how important vaccines are. This is the perspective that people are having. That should we take the vaccine or are they safe? So you will see that a large number of people they hesitate for the vaccines, but there are a much bigger group of people. It is basically you will see that some developed countries they are having some better opinion about that and they would like to have the vaccines. So the types of the vaccines you are much aware, and here you will see that live attenuated vaccines or the organism is being used to just induce the immunity. Inactivated one are also used, but beside that you have some other viral vectors are there, mRNA is there, and conjugate vaccines are there, toxoids are there. But there is one more approach, and this is what as the sub-unit vaccine. You just target any specific protein. Let's say this is there on the surface. You just clone it. You just introduce. You are multiplying it, and to multiply it, you can have some eukaryotic expression systems. You can have prokaryotic expression system, including the plants. So here we are talking about the plants that how these subunits can be expressed in the plants. Are they successful, or if there is any success story, what is that? So plants can be genetically modified to produce vaccine, that is antigens. These are the key components and trigger immunity. Then these are cost effective. That is one of the reasons because this is easy to grow the plants. You just genetically transform them. Then you are producing the protein of your choice. You just isolate them, and then this is ready. But it has to go throughout. It has to pass all the stages to become a qualified vaccine. Then this is large scale production is possible, and because of that, this will be accessible to the people because of the bulkiness and also that this will be a cheap candidate economically. So genetic modification genes that code specific vaccine antigen, we have to pick that, we have to clone that, and using the plant as a bio factory. This is some of the stories with the expression of some antigens. Here you see that the Newcastle disease, COVID-19, that is SARS-CoV-2, against that also vaccine that has been basically approved and it has been administered to a lot of people of the population, mainly in the Canada. Then against influenza, norovirus, hepatitis B, and so on. So there are a lot of people who are working on this aspect, but the cases of failures are also there. I will tell you what is the reason behind it. So I'm going to talk now on production of chikungunya vaccine in tobacco, and that work has been done by Dr. Sadia Kamar, who is sitting among you. I told you already, and also we are working on very important aspect for last 2-3 years. That is production of monkeypox vaccine in tobacco. And this is again one student who is just clicking my photograph is working on this aspect. He is Musafir Rahim Khan. So you must be aware that I was talking that not all the viral diseases they are that much dead. There are some diseases which make your life difficult. In fact, you will hear from some old persons they were still be saying that since I had chikungunya, my joints are paining. I am in trouble. So any sort of trouble they would be having. So this is transmitted, this disease or this virus basically is transmitted by a mosquito of Aedes species. And chikungunya virus was first discovered in Tanzania in 1952, but there are usual sporadic outbreaks in different countries have been reported, mainly in India and nearby countries. So this is a positive single stranded RNA virus. And here we have targeted E2 because of a reason that although I should tell you that glycoproteins E1 and E2 are therefore the key targets because they are the surface proteins and they are interacting with the host when they are coming in the infection mode. So this is the prevalence of chikungunya. Almost every part of the world has been covered, but this green one is highly impacted, and you will see that always on the media and the newspaper when chikungunya was there just after the monsoon season few years ago in 2016 perhaps somewhere there. So you had a lot of news, and it shows the gravity of this viral disease. So this is something to just reflect that how this virus is propagating, and this is of course the application of chikungunya virus in mosquito. The mosquito feeds on virus-infected blood and occurs intrinsic incubation period in the vector, and then it replicates where mosquito saliva is stored. So then the saliva, which is having a lot of virus, then this is transferred to the host cell, the next, that is the human. And then this is immunomodulation that our body is offering against chikungunya virus. This is the part of the genome where you can see E1 and E2 are also there. So they are receptor binding proteins. And just coming directly to the E2 protein, which because of the time paucity, and just coming to the protein which we have selected to produce a vaccine in the tobacco plant. So here you will see that the E2 protein is the second envelope glycoprotein of chikungunya virus, and this is responsible for viral attachment to the host receptor, which is the initial step in the viral entry when it comes in contact with the human cell. The E2 protein forms a spike on the viral surface, and then it is facilitating the entry for the virus. The E2 protein is also target of host immune response leading to the production of antibodies. It has been proven in some of the studies. So both E1 and E2 proteins are important targets for antiviral drugs, and we have selected E2 for this reason. And then we have cloned it. I am going to just describe about that. And we have used here just some idea that why we use plants, because you can express the antigen of your choice in either of the organ in the plant. It could be fruit. You can take fruit specific promoters. It can be roots. It can be tubers or the leaf, and wherever you want, you can express that system. And this is again with some references, some studies which have been, except this last one, bacteria, all are the viruses against which some success has been obtained, and the vaccine have been developed. So, because of the gravity of the disease, because there was no chikungunya vaccine when we started to work on that, but there is some strain of some weak strain of chikungunya against the original one has been used to raise the vaccines, and this was recently approved just before 9 months. So there was no vaccine available. After that, this is only our work which is there in this field. So because of this urgent condition, we started this work. No reports were available at the time, and she delivered her Viva Voci, PhD Viva Voci, except her own. Then this is something about the tobacco that why we selected. Otherwise, the question should be coming from the audience. But this is just to tell you that this is easy to grow. It has a lot of biomass production in a very little time. This is easy to transform, and one of the good thing is that you can perform short-term expression without without being integrated with the genome. So you can go for that also. So these were the objectives in silico analysis to understand E2, because we are selecting E2 as a candidate protein. So this is important. We just prove that it is going to induce immunity. So we have to perform some in silico experiment, and we did that. Later on, construction of suitable expression system, genetic transformation of Escherichia tobacco, then immunoanalysis of the antibodies which have been raised, then molecular analysis to prove that what we got, this is immunogen and the antibodies have been raised against that. So this is what we chose. We chose S27 African strain of chikungunya virus because this is the most prevalent strain. And then this is the sequence, And translation product is also there, which is there, which will be coming in the form of the protein after being synthesized. So this has been displayed here. And we perform the B cell peptide prediction. This is something we found that B cell, perhaps better is way, we found B cell linear epitopes in the surface glycoprotein of S27 African stem virus, which may be capable of inducing the desired immune response. And we found that yes, it can induce immune response because of the presence of specific sequences which are present in this protein, which we see in the E2, that is on the surface of the virus. So after performing multiple tests which are available in silico, we found that yes, this is a good candidate, and we can move forward to use it as a candidate which can be sometime proved to be a vaccine candidate against chikungunya virus. So you see that so many peptides which are basically qualifying this protein to be a vaccine candidate. So we constructed, you see that these are the vectors that we got. We express this protein not only in the tobacco but also in the E. coli. And we found when we got the expression from the both of the organisms, that is from the prokaryotic source and eukaryotic, and they proved equally good, which you will see ahead. So we cloned it in PU C57, then we confirmed it, then subcloning in PAT28A, then chikungunya virus gene expression was confirmed using RT are also back. Then purification of recombinant protein because we use his tag and thus using the nickel anti column, we purified that protein. So before that, we had so many molecular analysis to just confirm whether we are having the gene in that construct or not. So we used PCR, we used restriction, and we found yes, our band is there. We move forward. Then the recombinant PAT28 chickpea E2, we are calling it because our gene chick E2 is there, so that's why this name has been placed over here. Plasmid was used to transform the expression, strain, and that we found. We basically on the antibiotic we did screening, and this is basically the colonies that we have got. These were antibiotic resistance. So the resistant product that was carried forward. And this is important to mention that the induction of the protein expression was there, and ultimately what the protein we got, we run it on the SGS page, and we found that there was a protein of the 49 kDa which would be a recombinant protein which was confirmed by the Western blot. Then this is total protein from E. coli was isolated, of course, and then this was run on the nickel anti column, and using the imidazole, we isolated the purified E2 protein which we expressed. Here, this is purified on the nickel anti affinity purified fractions on 12% SDS phase has been run, and this has been displayed over here. Then on the Western blot, we got it positive. So this was these are the conclusion of the objective, right? Okay. So genetic transformation of Nicotiana was performed, and we put those. This is the construct that we use here, and you can see that hygromycin resistance and kanamycin resistance were used, and this was selected. Later on, the protein that we got, you can see here on the column that we collected it, and then it was injected into mice just to see whether it is producing any sort of immune response or not. So ultimately, just coming to the, this is tobacco that we transformed, and agro infiltration you can see here, sorry, agro infiltration followed by the expression which is easily visible on the confocal photographs that you see here, that the expression was confirmed. Ultimately, we were confirmed that we have got the protein which we were working with. The protein which was extracted from the E. coli, this was also injected to the mice, and we found yes, both of the proteins, either coming from the bacteria or the plants, they were genic. Later on, all the assays from the blood of mice, including the immunoassays, were performed. This is all the group that we have used of the mice. So we found that yes, we got protein, and that is the protein which induce immune response in the mice. So of course we have not gone to the human. In fact, we purchased the kit from the market against E2, and that was also positive. So all the molecular analysis of the plant was also performed. RT-PCR results were positive. Ultimately, we filed a patent few months ago, and hopefully moving forward with this to have first Indian vaccine against chikungunya virus. So these are some limitations, and I would like to acknowledge the help of Professor Abidi, who is one of my collaborators, also Professor Lando Zola from Italia, Monica Dadal, Dr. Altaf Ahmad from AMU, Professor Saeed Ahmad from Jamia Hamdard, Professor Suhail Parvez from Jamia Hamdard. These are my present lab members, and these are the past lab members that you can see on the right side. There are some around 20 PhD students who have been produced from my laboratory. And the last two that you see in the list, last three, say Saida Amna, she recently got a postdoc in Oak Laboratories in Tennessee. And then the last one here in Muhammad Irfan Dar, he has got postdoc recently in New York University. So this is some success from my lab that you see here. Dr. Hina Tabasu, she did a postdoc from my lab, and now she is one of the senior scientists in ICMR. Dr. Javed Ahmad is among you. He is assistant professor in your department. And then Sobia Munir, she is associate professor in PIT, in Department of Biotechnology, and so many others at good positions. If you have any questions, then you may ask, but I
So open for discussion and we can take two questions. Yes, please.
So using plant as biofactories is a very enticing concept. So my question lies is there a scope of expressing vaccines in food so as to develop edible vaccines? What challenges do you foresee in that?
Yeah, this is of course. Actually, thing is that when you are going to consume the vaccines which are oral vaccines, so let's say in carrot you are producing some antigen. So when you eat it, then what will happen? The protein is digested in digestive tract. For example, if you eat the snake venom, that will be digested and no harm to you. But if we have some protein or some system which can help in the absorption of that protein, and if it can induce immune response, then definitely that will be successful. But people, you know that people are against GMO, so that is something which they will not trust that much, except if you have something purified and proven with the clinical trials.
Okay, sir.
जो बीएससी एमएससी के स्टूडेंट निकल रहा है एग्जाम में राहुल बदला लेने का अच्छा मौका है
Assalamualaikum, sir. Good afternoon. Sir, very nice talk. Thank you for bringing the new paradigm of vaccine development, plant-based vaccine. So first, actually concern I had, how we are going to control the dose of the vaccine? If it is edible vaccine, how we are going to control the dose of the vaccine? Like if it is a banana, how many bananas we have to eat? So that is a concern for the vaccination.
So the good name
Mirage.
so Dr. Mehraj, he's just like deep sea, you know, deep sea, the latest AI tool. So he has so many good questions as he also asked. So this is again a good question. He is taking all your responsibility alone. So of course, yeah. This is a good question. Basically, he want to know that at what level of dose of the antigen this is sufficient for us. So let me tell you that for that, in fact, a small dose is sufficient. And if you just induce immune response against that, then the matter which is more important is that what would be the half life of those antibodies like that, and how the human body is going to treat it in the future, the infection. So of course, this study has not been done in human, but in rat we have estimated the half life with the quantity. So if you know the quantity, and you can predict that how much response this would be. Yes.
Thank you, sir. Sir, another concern about the neutralization assay. If you have an
Please don't disturb me in the lunchtime.
thank you.
You can.
Yes, sir. So, in the sense that, let's say we check that astrology industry 2 milligram of protein is coming in the one banana. So we see the amount of the responses generating in our body, basically first in the minds. If the responses are low, it may cause autoimmune response.
Right.
So really we have to check that those dependent assays that's what happened in case of COVID. There was a suddenly stop that was filling our organs. So we really have to optimize that particular role to check.
Thank you, sir.
Any question? If not, then please you can discuss during the lunch time and let us summarize this session. I talked on vaccine development for vaccines against pneumonia.
Thank you, sir.
And he talked about on using the plant to produce things. It's a wonderful idea, and he has worked on chicken awareness. And he has expressed the protein in the different factors, protein expression factors. So hopefully, as you rightly said, that the vaccine will be the first one in India to be produced against the chikungunya virus. Before this took out into the market, it needs really a lot of clinical trials. We have just completed the animal model. One person was asked from the backside of the audience, it's a speaker. He asked me whether we can do the blood transfusion. He has one very good point. He has talked about from the cost effectiveness of the vaccine. Yes, we can do that. That's not a problem. I've done one technology there. We can make the antibody, and we can go ahead with that. But your vaccine production should be cost effective. So initially, when no vaccine was available for COVID, if you are talking about the replacement of the plasma vaccine or something like that, they can do that. So I think the question was just if you can answer the question. The young man actually, I was looking forward to share some.
Thank you, sir. My question is, प्लांट को अगर वैक्सीनेशन करते हैं तो सर उसका जो साइकोलॉजिकल कंडीशन उस पर इफेक्ट होगा या नहीं, अगर होगा तो किस पे होगा सर, सेंसिटिविटी पे होगा, ड्रग स्ट्रेस पे?
The first thing is that हम प्लांट को वैक्सीनेट नहीं कर रहे, क्योंकि हम किसी डिजीज के अगेंस्ट प्लांट को वैक्सीनेट नहीं कर रहे हैं. हम लोग प्लांट को यूज कर रहे हैं एज ए बायोफैक्ट्री, अपना प्रोडक्ट उसमें एक्सप्रेस करने के लिए. सो डेफिनेटली साइकोलॉजिकल प्रॉब्लम तो उसको भी जिसमें नर्वस सिस्टम होगा, प्लांट के अंदर नर्वस सिस्टम नहीं होता है. सो उसका सेल था एक जीन को रीड करते हुए पूरा का पूरा जो उसका ट्रांसक्रिप्शन सिस्टम है, ट्रांसलेशन सिस्टम है, हमारे प्रोटीन को प्रोड्यूस करते हैं. ये हमारा ऑब्जेक्टिव है. और उसके बाद प्लांट का फेट होता है जो सैलेड का होता है, मैंगो का होता है, जो वाटरमेलन का होता है. ठीक है ना?
सर मैं आपके यहाँ पर थोड़ा सा मेरे को पता है, प्लांट डज हैव immune response to our body को बनाते हैं, antibodies तो plant के अंदर भी होते हैं. Plant does also have सभी को लेक्चर नहीं बोला है, किसी को भी बोला and people know we are working on plant. But what we are doing actually, वो यूज कर रहा प्लांट को बायोफैक्ट्री. SCLS सीएम इंस्टीट्यूट को मेरे को 50 ml करके 10 mg मिला, वो कॉस्ट इफेक्टिव नहीं है, बनाना दे सकते हैं, इजीली अवेलेबल है, उसको वो प्रोडक्शन भी है। टारगेट ये है कि वो प्लांट को एज ए बायोप्रोडक्ट यूज करना चाहे तो कॉस्ट इफेक्टिव प्रॉपर मैनेजमेंट के कुछ पर केले निकलना इजी है, चाहे वो गाँव हो ब्रांड में हो, कोई दिल्ली हो गए कि केला मिल जाए तो वो वैसे एकदम अच्छी है, बहुत टिकने
थैंक यू सर।
आप क्वेश्चन है कि इम्पैक्ट आएगा शायद डेफिनेटली ये भी दिस इज श्योर मैं कह रहा है तो रेडॉक्स पोटेंशियल में क्यों नहीं आता है शायद कहता बहुत अच्छा
क्वेश्चन आपके जो कैंडल उसको एक्सप्रेस करना है उसमें क्या होता है आई थिंक
थैंक यू सर।
एनी क्वेश्चन प्लीज वन प्लीज वन प्लीज so that everybody can
Is there a correlation between the types of vaccine and the plant species you are using? या कोई भी प्लांट स्पीशीज ले सकते हैं वैक्सीन बनाने के लिए?
और एक वाइड सिलेक्शन करना जरूरी है क्यों इतने सारे प्लांट लेट्स से लेटेस है हम लोग वो भी यूज करते हैं टोबैको उससे बेटर प्लेयर है ना आसानी से बायोमास ग्रो करता है स्पीड से ग्रो करता है लीफ ब्रॉड है इजीली आप इसमें एग्रोफिल्ट्रेशन कर सकते हो आसानी से इसका कल्चर सब कल्चरिंग कर सकते हो सो हमने इसलिए टोबैको
आपने लेकिन आपने लिखा है टाइप्स ऑफ वैक्सीन में आपने दूसरे प्लांट्स या कोई और दूसरे प्लांट्स लिए हुए जैसे आपने बताया लेट्यूस लिया या टोबैको, तो उसके पीछे क्या रैशनल था, क्या तो क्या थिंकिंग था कि ये पर्टिकुलर वैक्सीन इसमें एक्सप्रेस होनी चाहिए?
कोई भी एंटीजन जिसका आप जीन साइज कर रहे हो उसको आप उस प्लांट में एक्सेस कर सकते हो तो जरूरी नहीं आप तो जब वहां प्लांट आ वो इन्फेक्ट कर सकती है ना मैंगो भी हो सकता है कोकोआ भी हो सकता है जो इजी एक इजी गोइंग कॉस्ट हमें तो जैसे मैंने वैक्सीन के टाइप है वो वो नहीं है वो सारे टाइप वाला अलग टेबल था जो एक्सप्रेस किए गए वो अलग टेबल है तो वायरस लाइक पार्टिकल एक्सप्रेस करना एक अलग बात है और जो जितने जो कंप्लीट ऑर्गेनिज्म वो आप एक्सप्रेस नहीं कर सकते हो क्योंकि आपने उनकी जीन को एक्यूमुलेट करके एक्सप्रेस नहीं किया है
के कंपैरिजन में सिस्टम कितना आई मीन एडवांटेजेस है?
मैं अदर वे राउंड बता रहा हूं तो ह्यूमन के कंपैरिजन में ये दोनों सिस्टम फेल हो सकते हैं या वीक हो सकते हैं उसका रीजन है क्योंकि आपको पता है कि प्रोटीन्स ग्लाइकोप्रोटीन्स अगर हैं तो वो तब भी प्रॉपर्ली ग्लाइकेटेड होंगी जब वो ह्यूमनाइज होंगी तो वैक्सीन का ह्यूमनाइजेशन भी इंपॉर्टेंट है हो सकता है उस तरह का ग्लाइकोसाइलेशन ग्लाइकेशन अंड से ना हो बैक्टीरिया बट इत्तेफाक की बात है इंड्यूस किया गया रोडेंट्स में यानी रैट में उसने इम्यून रिस्पांस शो किया तो यू आर गेट दिस अदरवाइज पीटीएम पोस्ट ट्रांसलेशनल मॉडिफिकेशन का प्रॉब्लम हो सकती है प्लांट्स में बैक
Any question? No, there is no question. I think we thank you for
It was very wonderful and knowledgeable session. Thanks to our speakers. So, to conclude the session, I would like to call upon Dr. Umara Faruqi, HOI Department of Biotechnology, to offer memento to our esteemed guest speakers, Professor Atul Kumar Johri and Dr. Irfan Qureshi. Sir. Now I would would like to call our chairperson, Professor A.K. Jory and Dr. Saima Wajid to accept the memento for chairing the session.
मैं कहीं कॉन्फ्रेंस में जाता था उनके लगता था मैंने ओलंपिक के रेस से आ रहा तो पूछती थी कि पापा क्या जीत के आए तो मैं उनको ये दिखा दूँ और आप लोग बड़े हो गए तो थैंक यू वेरी मच फॉर
Our chairperson, please felicitate the moderator of this session as a token of appreciation. Now I request all the speakers, chairpersons and moderator of this session and the head department of biotechnology for a group photograph. So I invite all of our guests to join us for the lunch. So, and we will resume at 2:30 for our next session.
Should I start? A very warm welcome, everyone. I hope you all are re-energized. Now we have some engaging talks lined up. So here we begin our plenary session 4. The theme is insight into molecular biology for global health security. I consider it as a great honor to invite Dr. Miraj Ahmed Ansari, Assistant Professor, Department of Biotechnology, Jamia Hamdard, to moderate this session.
Good afternoon, everyone. I know it's very difficult to sit post lunch, so thank you all who make it to be here. So welcome to the plenary session 4. And in this plenary session, we have two speakers, Dr. Abdul Mahmood Khan and Dr. Viba Ahuja. But unfortunately, Dr. Abdul Mahmood Khan cannot make it today to be here. So we have one speaker, and Dr. Viba Ahuja is also happen to be chairperson for this session. So Dr. Viba Ahuja is serving as Chief General Manager at Biotech Consortium India Limited, and a leading expert in biosafety, biotechnology regulations, and capacity building. With nearly 27 years of experience, she has worked extensively with regulatory agencies across India and Asia, shaping biosafety policies and guidelines. As lead officer for the UNEP Biosafety Capacity Building Project, she played a pivotal role in strengthening global biosafety governance. Dr. Ahuja has been actively engaged in the policy issues related to gene editing in India through engagement in the development of policy briefs by National Academy of Agriculture Sciences and member of drafting committee constituted by the Department of Biotechnology. A lot to learn from her regarding the biosafety, because most of the people are unaware of the biosafety and do experiments they need. It's very basic, and I hope that it will be very thought-provoking session by you with your expertise. Another chairperson we have our very own Professor M.Z. Abdi. The words will be like showing candle to sun. So we all are aware. He is founder of biotechnology, founder of CTPD, and both of the departments and centers are doing very well under his leadership. So Professor Amjad Abdin, a renowned plant scientist from Department of Biotechnology, Jamia Hamdard, honored with many awards like the Gyan Ratna Award by UPCST, Government of Uttar Pradesh, and the Best Researcher Award by SciFax for his contributions to biotechnology. He has received a mark of honor from His Excellency Governor of Maharashtra, Shri Bhagat Singh Koshyari, for excellence in plant science. With expertise in enzyme and gene regulation, biochemical analysis, and genetic engineering, he continues to advance biotechnology and molecular plant research. Thank you, sir, for being here. And I invite our eminent speaker, Dr. Riva Ahuja, to deliver a lecture.
Fine.
ऐसे ही करना है?
No, ma'am, this is the forward and this is the backward. This is the pointer.
Thank you very much for your kind introduction. And at the outset, I also wish to express my heartfelt thanks to Professor Abdeen and all his colleagues with whom we are trying to work together for taking forward a GM product from your esteemed university. And we are very hopeful that we will succeed. The topic that has been given to me, you all are researchers and do great research here. But then, taking forward this research, what we say from lab to land, there are also certain types of regulations, as the kind moderator introduced to me, that there are certain biosafety issues that we need to deal with, and that is what we will discuss today. But before we move forward, I would like to briefly introduce, maybe some of you are not aware about Biotech Consortium India Limited. We operated way back in 1990. It was an idea by the Department of Biotechnology, exactly the same idea that the topic has been given to me. How do we take the technologies which are developed biotechnologies developed in the laboratories to the institutions, because there is a work beyond the lab which is required, and this may include your intellectual property, as you can see, transfer of technologies to the industry. In some products, there may be some kind of certifications or some kind of, standardizations required, scale up of technologies is required. And then, as we moved around that time itself into modern biotechnology, which is the genetic engineering and now a lot of activity in gene editing, there were another very important area of regulations, and these were the biosafety regulations, which we will talk about. So BCIL works in all these areas, which are required to facilitate commercialization of biotechnologies developed by both public and private sector. So particularly in case of biosafety, we have been working for many years, almost since 1996, and we have done, worked with the concerned ministries in formulation of the guidelines in India, providing the resource support to them, then doing lots and lots of training, international conferences, and so on. Regulations are, although we all have national regulations, but then there is also, because to facilitate trade, there is also required the harmonization with other countries. We cannot have something very separate regulation from what is happening globally. So we do work with other countries and try to understand where they are moving. And then there are lots of international bodies like, FAO, WHO, there's a Cartagena Protocol on Biosafety, these international treaties under which these matters are discussed. So we have to follow them, but at the same time, when the negotiations are on, our government, participates in that to ensure that nothing against our national interest happens. So when those things happen, there's a lot of scientific support required. Consultation with other stakeholders is required. And even to take those inputs, we need to educate sometimes the stakeholders of the development. So that's also another area where we work a lot, not only in India, but in the neighboring countries also. Bangladesh, South Sri Lanka, Bhutan, all these countries also we work, and sometimes in other Asian countries. Professor Ardeen had also participated once in our workshop conference in Bangladesh, where his work was really well received by the people. So we undertake all these kind of capacity building. Now, coming exactly to the topic of today, how do we improve crop improvement, or what we say plant improvement is very important. We have for centuries since, what I would say ancestors, from the gatherers to the selectors to the breeding, and so many things have happened ultimately for crop improvement. And this crop improvements is for multiple objectives: to increase the productivity, to fight against pests, to improve nutritionally. We have so many interests as a humankind, that we need to work on the crop improvement. So traditionally, what was the method was the conventional breeding, which was used for many years, almost 100 years or so. Later came mutation breeding. So breeding was between the close relatives, which can naturally breed with each other. So there was not much of a regulatory concern there. If you're having a maize variety A with maize variety B, the maximum you could do is interchange of genes. And this was happening for hundreds of years, and we have, I don't know what kind of varieties to improve significantly to our use. And then in the 1970s, came the recombinant DNA technology. And this recombinant DNA technology is a powerful tool which empowered us to transfer genes not only from the related species, but from the unrelated species. So technically, recombinant DNA technology could help us take genes from bacteria to plants, plants to animals, or whichever genera, as we know the code is universal. But when this technology came and we were taking genes from different organisms, this area emerged that, some of these genes may not be in our food chain. We are not saying they're not safe, but we don't have experience with them. So it is better that we should have a safety regulations. We should test them before we give it to the society, which any scientist will do. So 1972, this technology came, and in 1975, The group of scientists in United States where this technology was started met at Asilomar. Asilomar is a place, and this conference is fondly called Asilomar Conference, where they met first time to discuss if this technology needs to be treated differently from things like breeding. So there was a uniform, sort of consensus that, we should have some safety measures, but because it could be dual use, we do not know this gene could be harmful. But then the initial focus was only on the laboratory safety. So at plants, and it became one of the very important tools for the crop improvement and because and for such traits which we could not have it in the germplasm of that particular crop. Now I will when we move through, we will see what kind of examples we have. So this was genetic modification, and every country, there are 1986 OECD group of countries. They came up with safety guidelines which were like applicable in all OECD countries, and it is fondly called Blue Book, 1986 Blue Book, and that provided the first context of biosafety. And after that, all the countries, wherever there was some biotech research initiated, started having their own, regulations. And then we have now conventional breeding. You should be well versed with this. You should be well versed with this so that as you proceed with your research, in parallel, you should comply with applicable regulations in the country. It's in the same way that when we do driving, we take license, and then same way for these kind of research, you have to have, there are certain procedures in the country which every research institutions or every researcher has to comply with, and they are same for public and private sector institutions. So as I said, innovation, whatever innovation you do in the lab, it has to go to the field, and one of the important steps for genetic modification and genetic engineering and gene editing is the regulatory part, and this is what is going to be our focus. So as I said, this is a huge field. There's a lot of science. There is also legal aspects and so on. There are country-specific regulations. There are international. But we will, in our limited time of 30-35 minutes, we will try to learn about what is in India and what you as scientists and researchers should be following. So as I said, India's biotech, like around 1986, when the rules came internationally, guidelines, most countries got active. In India, 1987, the Department of Biotechnology was set up, and they started supporting some research. And we have been quite early movers in this area. And 1989 itself, the government of India, under the Environment Protection Act 1986, came up with these rules. And these are the rules for manufacture, use, import, export, storage of hazardous microorganisms and genetically engineered organisms. Now, a question comes in mind of many people that why we are clubbing them with hazardous microorganisms. So that is very important to know, even from the public perception point of view. This doesn't mean that all GMOs are hazardous. See, this is just an evolutionary process that when the Environmental Protection Act came in India, subsequently, the process is there is an act, there are rules, regulations, and guidelines. This is how our legal system proceeds. Act is the most powerful, it is passed in the parliament. Rules are framed by the ministries, they are placed in the parliament, don't need any approval. And subsequently, the process gets, limited to those ministries. So when the Environmental Protection Act came in 1986, and this act came actually after the 4 years after the Bhopal tragedy, we always had a environmental protection in our constitution, but a separate act came in 1986. So the concerned ministry started developing various rules for hazardous chemicals, rules for hazardous solid waste, rules for hazardous, microorganisms. And around that time, as I said, the DBT had come in. So they wrote to the ministry that we need something for genetically engineered organisms. So they added this in that. So that was the reason, and it is not why I have raised this, that we've seen some activist organizations getting into this propaganda that GMOs are clubbed with hazardous and this kind of. So I always take a minute to inform you all. So now this is way back in 1989, and we are having this conference. You would have heard so many good research going on how do we update our rules? It's not easy to update an act or a rule, but what it is done is that the concerned ministries, they come up with the guidelines. So we have a lot of guidance documents which I will elaborate upon. Now these rules 1989, they are implemented because they are under EPA and the Ministry of Environment, Forest and Climate Change. So that is the ministry which will implement it. They are supported by the Department of Biotechnology, jointly implemented, because the subject is very science based and you need a scientific support to that. And then the state governments are involved in the monitoring activities. As in other cases, we have in our country a system that we constitute committees, they're not independent full-time people. So these are the statutory bodies which have been prescribed in the rules and are functioning. So first is the Recombinant DNA Advisory Committee, which is in the Department of Biotechnology, and this committee is actually not a day-to-day functioning committee. It meets when there is some larger issue to be tackled. So 89, when the rules came, 1990, the guidelines came, the first guideline, the committee met. After that, for many years, it was not required. But like an issue like gene editing came, which was a very different technology. So this committee met again and advised the government on how they should deal with this kind of a newer, innovative, like a, path breaking or something like a different thing. The day-to-day committees are three, which are in green here, and they are involved in your approval of applications, and that is where you interact. You as scientists would be interacting with. So the first is your institutional biosafety committee. In your institution, as per our legal provisions, every institution which is doing research in using genetic engineering or recombinant DNA technology, they are supposed to have an institutional biosafety committee. So like your institute also has an institutional biosafety committee, and so are almost 400, 500 institutions in India, both public and private sector. The next is the Review Committee on Genetic Manipulation, which functions in DBT. Most of the contained research or very, very small scale activities, they are the responsibility of the Review Committee on Genetic Manipulation. They are very intensive in terms of scientific evaluation, because they are first time coming at that level. The next comes is the Genetic Engineering Appraisal Committee, which is in the Ministry of Environment. That will come into play when you are doing the field trials or any large scale evaluation or the final commercial release of a product. At the state level, the function has been given to the state level biotechnology coordination committee and district level committee. I mean, I'll just share with you all that, if you look at this SBC and DLC, somebody thinks, why at district, why do we need to go that far? Because they're into monitoring. And imagine 1989, people didn't have experience with what kind of range of products will come. So they have given something similar structures as for chemicals, as for waste and so on. So some of these things have been over the period modified amended by notifications or guidelines and so on. So if we think about the plants, right from, as I said, the recombinant DNA safety research, the focus is on guidelines, focus of the guidelines on contained research, laboratory research basically. That was 1990. The guidelines have now been updated in 2017. Then as the work started on plants more, then the plant guidelines came, then the intensive large number of field trials started, so we needed field trials. Then food safety assessment. These food safety assessment and environmental safety guidelines also are a result of developments at the international level. We did have some brief provisions in our guidelines, but when the WHO, FAO, WHO Codex, they adopted a new guidelines, so we updated our system and came up with a dedicated guidance, and so on. So also with the environmental, environmental impact. So if we look at the rules 1989, they're very broad. So you can think, even if I today ask you to make something on AI or something, you'll try to make it as broad as possible. So all entire spectrum of activities is covered. And if you look at the third bullet in this, it says new gene technologies in addition to cell hybridization and genetic engineering. So in a way, they made it so broad based that anything which will come will be covered. So that was actually something which became very important and how we dealt with when the gene editing came. That's very important to know. I'm not going to go here step by step all the procedures, but there are websites, and any of you have any issues, you can also get in touch with us. We will provide you guidance. But there is a broad procedure which is captured in this flowchart. If you are an applicant, till the time you are doing, laboratory studies, your point of interaction is only your IBSC. The more important things come when you do field trials and environmental release. At that time, you will give your application to IBSC, it will go to RCGM. As I said, there are certain responsibilities of RCGM, GEAC, and in between there will be some other committee visiting, so on and so forth. And finally, you get the approval. Now, in this whole process, this was the more of a process, this committee, that committee, so on and so forth. What is the scientific data which is important and which is generated? So if you think of the safety data, it is very, very comprehensive. Right from the gene that you have taken, what was the source, what was the pathogenicity of your donor, what are the characteristics of the recipient organisms, complete molecular characterization in terms of copy numbers, this, that, all that is required to be done. What protein is it expressing, intervention, whatever it has been made, what it manifests into, the history of safe use of that protein, all these are very important. You see, if I have made an intervention and I'm just expressing something, taken a gene from maize to rice, both are edible. I don't need to go into toxicity and allergenicity, but I need to prove the history of safe use. But if it is a new protein, I need to go into the toxicity part of it. And for doing the toxicity, I need to know the expression levels. So expression levels are not only important from your performance point of view, but from the safety point of view also. Then, food and feed composition. Has it made any change in the composition? And this fourth part is actually very detailed. And the reason is that if there is any unintended effect, it is generally thought that it will manifest itself. That's what the codex negotiations, concluded, agreed, consensus came that detailed composition analysis is a very important indicator if there is any variation or is there any interruption of any other metabolic pathway. Then you have the environmental issues. So you have made a new plant. You were trying to protect it from one insect. It is what happens to other insects, non-target organisms, soil microflora, this, that. But as I said, not everything is applicable to everything. Your crop trait combination will tell you what tests are required and what tests are not required. So all that we'll have to do. But the take-home message is that the process is so comprehensive, the guidelines are so comprehensive that at the end of this evaluation, you are virtually, no scientist will say 100%, but there is a virtually zero doubt that this product can cause any harm. And that's what over a period of 20-25 years, the systems have developed so strongly. So anything which has been approved by the regulatory system, there is actually beyond doubt the safety. Now, where are we in terms of India, in terms of the GM crops which have been approved or not approved? What happened? So if you look at this, BT cotton was the first product which was approved by India way back in 2002. This was against one insect which was having a devastating effect, and it had been, really very, very harmful, Helicoverpa armigera. And globally, it was found that there is a protein from Bacillus thuringiensis which can, protect the plants from this particular insect. Now, Bacillus thuringiensis had a very safe history of use. It was actually used as a formulation. But then the company, this was by a multinational company in collaboration with an Indian company. But as soon as it was approved, within 3 years or 4 or 5 years, there was almost 95% adoption of this particular product. It was so effective and it helped India triple its cotton production. We were an importer and we became an exporter. Tripling the production meant tripling the cotton seed meal availability, tripling the cottonseed oil availability. So it had a really very positive effect. BT cotton has been a very big success in India. However, there are of course challenges because since 22 years, because of various reasons, activist representations and all, more products have not come. So that is why we are really looking forward now to the newer products. The second product which was actually approved in India by the GEAC was BT brinjal. Now, BT brinjal was approved in India, but They were at that time the minister got lots and lots of representations, and in his wisdom he thought that I think this is a debatable issue, and he decided to do some public hearings, and you know how these public hearings are, and finally he decided that unless there is scientists come to an agreement or those kind of issues, this should be held back, a moratorium on that. However, in our neighboring country, Bangladesh, also similar research was going on. They used our safety data and they released Betibrinjal in 2013. So it is grown, consumed, and they have taken benefit of it. But in our country, it is still on hold. The company is now trying to get it released here, but still it is not. But as far as the scientific evaluation is concerned, it has been cleared by GS. The third product which moved this whole ladder of was from a public sector institutions, our own University of Delhi, Professor Deepak Pental, he had developed we are a big importer of oils. India is the world's largest importer of vegetable oils. And we spend some close to 150,000 crores on just importing vegetable oils. So we have to really work to increase the productivity of our vegetable oils. And some of those like mustard, they are self-pollinating, and to make hybrids, the world over, genetic systems are used, which is called Barnage Barstar. So university has developed this, and they, in fact, BCIL worked with Professor Pentel to take this product, generate all this biosafety data, all the trials for 3 years, and so on. And we submitted a dossier to the government. Everything happened. It was about to be released in 2017, but was held back. Finally, because some activists raised this issue of honeybees and some other adverse impacts, apprehensions. In 2022, the government of India reconsidered it and released this product. But by that time, we have a court case going on by the activist organization. So they went to the court, and still the hearings are going on. So, but at the last year, there was a lot of hearings. In last year, there was a judgment by a two-judge bench, and the two judges gave opposite judgments. One judge said these are safe, whatever government procedures are absolutely fine, and this should be taken forward. The other judge had a different view. So it has been referred to a three-judge bench. So it's very, very unfortunate. We have been working with Professor Pentel. Having seen these scientific issues discussed there is very painful, because they won't understand science, what is it? But then that's how it is. But the government of India is very positive, and they are actually, now fighting for it, and hopefully it will be cleared in the court. So as I said, the important developments have been mustard. So like, I told you in 2010, there was a moratorium, and subsequently the research slowed down. But the last 3 years or so, there has been a very positive outcomes. Things like approval of mustard, the herbicide tolerant is under consideration. In fact, we are helping public sector institutions, so several trials are underway. GM banana, late blight resistant potato, pigeon pea, cotton, corn, and so on. Now, the last point is very important. You see, I have not brought in global data, because that's something you will anywhere see that. But I just want to put forth this point that the way we had experience with cotton world over, like United States, Argentina, especially the South American countries who are big agriculture producers, they are exporters. The moment they adopted a technology like GM corn or anything, canola in Canada, which is like mustard, there's a 95% adoption in 2 to 3 years. So that is the benefit of the technology. So in 2021, we had soybean is another largest product. GM soybean is the most cultivated world over. 85% of soybean globally grown is, I think, between 80 to 85 is GM soybean. So in 2021, we had a shortage of soybean in our country. All the soybean oil that you get, it is all imported, and it is derived from GM soybean. But we had a shortage of GM soya meal, which is used by our poultry. So when there was a shortage, if you go to the international market and you want to import, you won't get non-GM soybean. So that is the situation, and that is the level of adoption. So India also had to import some GM soybean meal, although we have not approved in our country. These are some of the challenges which I have already discussed, but I will move on. But long and short of the story is that there are challenges in GM, but in the last 3 years, we have seen very, very positive steps by the government because the agriculture challenges are big and some of these problems cannot be solved without adopting technologies like genetic engineering. The next one which we'll talk about, which is the recent, tool which has come for the plant improvement is the genome editing. It is a new tool for crop improvement. Extensive research efforts underway globally and in India, multiple crops and targets. And if you see the bottom pictures, these are all the products which have been approved somewhere in the world. So first one there was the soybean with high oleic acids. The second was an high GABA tomato. But in Japan, this is a tomato with an enzyme and it relaxes the blood pressure. So it is already in the market and is being widely distributed. We have a reduced browning, what you say, banana. It helps in meeting the some post-harvest losses. Non-browning potato, very much of interest to the chips and the potato processing industry. Then this is a recent one which has been introduced last year. Like, in the Western countries, they like to eat a lot of salads. So mustard has a lot of properties, but it is strongly flavored. So they don't eat. So they have developed through gene editing a milder flavored mustard. And then there are, of course, I have not added here, there are fish, there are other kind of products gene edited, which is going on a big way. But unlike genetic engineering, where most research started in United States and Europe and then moved on to this one, in gene editing, the highest number of papers and highest number of activity you see was by China. So in fact, China has sort of, moved ahead of US in terms of publications and all other things. So this technology, gene editing, it was discovered in 2012. The two ladies who worked on it, in fact, first time by two women together, Nobel Prize in 2022. And one of them is Jennifer Doudna, who wrote a paper in 2023, actually, 2023, and it came. So this paper, because when they discovered the technology, the paper was that, how the gene editing is being used. They wanted to evaluate. So this is a paper from Science, and I have just taken this picture. If you see, the left-hand side shows past 10 years, where all it has been used. In the past 10 years, gene knockouts, sickle cell therapy, knockout mice, and CRISPR modified crops. That's a very important area which emerged, screens and so on and so forth. But if you see in the next 10 years what they have projected with their wisdom, where all it is likely to be applied, and you see the agriculture is getting expanded in a big way. So for the plants and the animals, there are going to be large number of applications, and this is actually evident from the kind of research papers and the products which are coming in. So now we'll come back to our topic of regulations. Genetic engineering, we regulated. Why? Because there was a new gene. Now, when it came the gene editing, there were some key points with respect to as a regulator, what do I look at? So what we realized was that CRISPR is only, or gene editing is just such editing through mutation breeding, but gene editing is allowing it to do it in a much more precise manner. But the traits which are finally coming, they are not novel. So many of them are generally possible to produce through conventional breeding. And what gene editing is allowing us to do it possible to do more easily, more expeditiously, and with a greater precision. Because now we know the genetic code, so we know exactly where to edit. We don't have to do random editing and then select. We know what to do. So some methods of gene, but the important point is some methods of it, the initial part of it will require recombinant DNA technology, and it may be transient, the genetic transformation will be transient, but in the final product, in some of them, not all, no transgenes may remain in the final, this one. So keeping that in view, the categorization of plants, there are called SGN1 and SGN2 and SGN3. SGN means the site-directed nucleases, the cuts that we make. 1 and 2, the final products would not carry any exogenous DNA. 3 will have uninserted DNA because gene editing can also be used to insert genes. So now the final products of any final product produced using SDN1 type of gene editing we can actually get the same through mutation breeding. And if I produced using two methods, you will not be able to distinguish which method has been used. There's one deletion in both. So this is very important for a regulator, because how do I implement the regulation if I can't distinguish between the two? So the fourth important point is the regulatory systems are always risk based. As I mentioned, I gave a long preamble to why we regulate genetic engineering, because there were new genes. Here we don't have new genes. And we were not, if you remember my first picture, we were not regulating breeding. We were regulating genetic engineering. So SDN1 and SDN2 come under category 1, which is like breeding, and 3 comes like a GM. So based on this, the regulatory system has also been worked like that. It is not that, it's very important to understand. I hear many people saying gene editing has been exempted, which is not the way to, as a scientist, as a science students, we have to really understand why it is so. It's not that one fine morning somebody got up and thought, let's deregulate gene editing. There's a basis of that, and that basis is very, very important to understand. So because of all these considerations, there have been many countries which came forward, and they have come out with a clear policy approach wherein they are saying that the products where there is no exogenous DNA is similar to that of conventional breeding. So we need not regulate it like a GM crop, but there is still some countries who are thinking about it, especially the European Union and New Zealand and South Africa and so on. This is a slightly older map because this keeps changing, but you can see the message here is, if you look at the agriculture dominant economies like North America, South America, Australia, even India, we have also said that we won't regulate. So these are the countries which have been more quick in issuing exemptions, because we have to do crop improvement. We want faster crop improvement and faster bringing them to the field for use by our farmers. Our challenges are very great, and we all need to work for that. Now, if we go back to the regulations part, the regulation that we saw, as I said, there was a very broad definition of gene technology. And if you look at this definition, it included deletion and removal. So technically, our gene editing, as per our rules, was to be treated like GM. So what we did, there has been a process in India. As I said, our process of updation is have a new guidance. So Department of Biotechnology started with making a guidelines, and they came up with this regulatory framework and guidelines for risk assessment. And they opened it up for public comments in January 2020. But what happened? This document covered everything, plants, animals, microorganisms, human cells. So there was lot of confusion, because everything has to be dealt with differently as far as gene editing is concerned. So there were lots of comments by the stakeholders, and one of the most important agency which gave the comments and which came forward was the National Academy of Agriculture Sciences. So with all their efforts, the government decided that they will focus on genome editing, and then this whole process went through, and finally we had a notification by the Ministry of Environment wherein the genome edited plants falling under the category of SDN1 and SDN2 are exempted from regulations. I've just taken the key features here. SDN1 and 2 free from transgenes. There was a rule 20 in our rules which empowers the government to issue such exemptions for specific organisms. So they used that, and they said SDN3 will be treated in the same way as GM because there are new genes, and they will be treated on difference. Now the first point is very important. However, the initial work using genome editing requires recombinant DNA technology. So your initial work, until you demonstrate that it is free from exogenous DNA, will have to be regulated by your institutional biosafety committee. So even if you are starting research on genome editing or genetic engineering, keep your IBSC informed. Any release there will be, as the new Seed Act, this, that, all will apply in the same way. The government is really wanting to promote the research on gene editing because this is more easier and regulatory. More work needs to be done. So immediately in March, the notification came, and in 20, they have also issued guidelines where the definitions have been provided. The process has been provided, SDN 1 and 2, how it will be moving 3, how it will be moving. Then even in terms of your research, from guide RNA design to gene delivery, and then demonstrating the segregation, till what time you are regulated, all that has been provided for. Now there was another challenge. We in this regulations, it is your institutional biosafety committee which is empowered to communicate that it is free from exogenous DNA. Now we have 300 IBSCs, so everybody may talk different. So standard operating procedures have been provided, which are to be followed by all IBSCs and also by all researchers. This is to facilitate the regulatory review for SDN1 and SDN2 until they are free from exogenous DNA. The information which is to be seen by IBSC has been provided so that there is no variation. The protocols to show that this is exempt from exogenous DNA have been standardized. Standard protocols have been given. I'm just giving briefly, but I would encourage whosoever is working on starting work on genome editing should look at these SOPs right from beginning, right from the first stage when you have to start your work, what form you have to fill, how you have to follow. This has to happen in parallel. So we have been moving very fast. There are institutions which have already taken approval. So we have in India, Indian Institute of Rice Research, IERI, they have taken already exemption for 2 rice varieties, which are under varietal evaluation. Our own NIPGR has taken a gene edited mustard has been cleared. There is an institute in Cuttack which has even developed a mini genome editor. So there is a lot of activity. It's very important to target these technologies. You see, I talked to some of the experts and they say that our agriculture challenges are huge, so we need lots of tools to work on. And this is just how the international markets are growing. You can see the breeding will continue. A blue one is breeding with minor increase. Then the GM came. Now we have gene editing, rapid increase. So we all have to work for taking care of the crop improvement, plant improvement through this. And we have the enough procedures on how to take it forward. So that's all I wanted to communicate. So just to conclude, agricultural biotechnology adoption is the need of the hour. We have robust framework in place, and we have had a positive progress in the last 2-3 years, which was the approval of mustard by the government and then this notification SDN 1 and 2. In fact, we surprised the world by being the most clear regulatory framework for SDN 1 without any doubts. And with all this, there is an enabling policy environment for researchers and developers to develop the improved crops or plants to deal with the various challenges. Thank you so much. And this is just because I thought there are few students here on GM. There is a portal. Some of you may be familiar is the Swayam portal by the Ministry of Human Resource Development. Earlier under one of the projects with the ministry, there is an e-learning course also on GM crops, and I would encourage you all to look at it. This is just like a walking through, but if you want to know, and it's not very tedious, 15 minutes each, kind of 5-6 modules, which you can always look at it at leisure. Thank you.
Thank you, ma'am, for sharing your lifelong experience and telling us about the regulatory bodies of India. Let me tell in simple words what my want to convey us. If you are proud to celebrate 15th August for the independence, be ready to enjoy celebrating 26th January as well. To learn your constitution, to be a good citizen. All kind of research is invited by the government, but it should be under surveillance by the surveillance under the committees formed by the government. So you do the good research, don't do anything wrong, specifically in the field of genetic engineering, recombinant DNA technology. We also have the RDSC in our institution, and Dr. Saima is looking after it. Thanks to her, she brought it back after 3 years by very hard work. So now I would like to invite questions if anyone is ready. Any doubt for the moment? Okay. So let me invite our attorney, Dr. Umaira Faruqi. She's there. Okay. So I will request Mr. to felicitate our speaker.
तो जो है मैं बोलना चाहूँगा, देखिए ऐसा है, आई थिंक बायो दिस BCIL बायोटेक्नोलॉजी कंसोर्शियम लिमिटेड, ये जो है काफी पहले से ये काम कर रहे हैं। डॉक्टर विवाह आहूजा, एक्चुअली she facilitated our students earlier for their training to different biotechnology institutions with fellowship. I think that program is still going on. अच्छा, it is with RCB, तो जो है RCB में इनको अप्लाई करना पड़ेगा ये जो है। So actually that program is still going on, and RCB is the nodal agency. So if somebody some of our students want to get to have that fellowship and get an opportunity to work in biotechnology institute, you can apply for that. And then actually Dr. Vivai is working with us for this RTPCR to take it to the field. Actually, she has, I calculate, ma'am, you have already given so nice and very informative lecture in so simplified way that I think most of us have understood the regulatory framework that is already there in our country for regulation of genetically modified animals, including crops. So hope that this will help us, the researchers and students, to learn and to then use these this information in their future work. So congratulations, thank you very much.
Felicitate Dr. Vibha. किसको बोला किससे बोला I would like to request Dr. Samawati and Professor Shahryar to felicitate Professor Jadani, the chairperson of this assembly. Professor Shahryar, I request you to felicitate Dr. Vibha Ahuja for what as a chairperson. She also chaired the session.
And I request to the chairperson, Professor N.Z. Abedin and Dr. Viva Ahuja to present the memento to the moderator of this session, Dr. Miraj. Now I invite all the speakers, chairperson and moderator of this session for group photograph.
मैम जो है काफी और PCIL से काफी हमारे स्टूडेंट ने फेलोशिप एक जमाने में जो है बहुत स्टूडेंट जो है RCB में से उनको से कहिए अप्लाई करें।
कुछ करना है, कुछ बोलना है? कौन आएगा? अब कौन आएगा? अच्छा ठीक, हम चले जाए।
हाँ जी, नहीं सभी
लगता है ना तो होगा ना?
बेटा मैं ना हेल्प हेल्प सॉरी
Good evening, everyone. Let's resume the event with full energy. We will now begin with technical session 7. The theme of the session is Green Gold Innovations in Medicinal and Aromatic Plants. I would request Dr. Salik Noorani, Assistant Professor in Department of Botany, Jamia Hamdard, to moderate the session. Please welcome Dr. Salik Noorani.
Good afternoon, everyone. It is my great pleasure to introduce our esteemed chairpersons, Professor Muhammad Shahriar and Professor Viduveri from Jamia Hamdard. Professor Muhammad Shahriar is a professor of pharmaceutical chemistry, has over 25 years of teaching and research experience. He has specialized in synthesis of bioactive heterocyclic compounds derived with the application in anti-cancer and anti-diabetic. He has collaborated with the renowned institutes like National Cancer Institute USA and Riga Institute Belgium and has over 120 publications to his credit. Professor Viduveri, former dean and the head, is a professor of pharmacology and phytochemistry with 35 years of experience in medicinal plant research. She has published over 150 research papers and has significantly contributed to the validation of traditional medicines. With this brief introduction, I would like to request Professor Muhammad Shahriar sir and Professor Viduveri ma'am, please come on the dais and chair the session.
So good evening to all of you and a warm welcome. I think this is the last session, so your attention is really required. And it's a very important session, which very well goes, with the theme of the conference, which has been organized for all of you. I am delighted to introduce Dr. Sharad Vats, the first speaker. Dr. Sharad Vats is an associate professor at the Department of Bioscience and Biotechnology, Vanasthali Vidyapeeth, Rajasthan. With over a decade of experience in teaching and research, his research focuses on plant tissue culture, plant metabolites and antioxidants. He has made valuable contributions to understanding the biochemical properties and applications of plant-based compounds, and topic is phytochemicals, a treasure trove of bioactivities. So I welcome Dr. Sharad Vats.
Slide forward, and this is what I want. Thank you. So at the very outset, I'd like to thank Professor Abdi for providing me the opportunity to share the platform with eminent members of the scientific fraternity. Thanks to conveners, co-conveners, and each and every member associated with the conference. Medicinal plants, phytochemicals, these are all treasure troves of bioactivities. You name one, you name a disease, you talk about anything that is having issues in humans. Plant is something that comes to your mind first and foremost. Since the advent of human civilization, mankind has always aspired to be ever healthy, ever happy, and may God, let me be immortal. We all know that's not very true at every stages of life. But I'll be true to my words if I say that plants have been pretty instrumental in achieving these goals. In fact, to fulfill this quest, and that's why I term them as humans' best friend. You talk about ancient Indian manuscripts, Rig Veda, Ayurveda, Bhagavad, Ashtanga Hridaya, and others. You talk about Samhitas, Shruta, Charak, and all. Every kind of ancient Indian manuscripts has something related to plants and their beneficial properties. In fact, you talk about ancient systems of medicines, including that of ours, Chinese, Siddha, Unani, homeopathy. These are all, we all somewhere or the plant-based. Now, basically the activities have mainly been assigned to the metabolites present in the plants called as secondary metabolites. We can just broadly categorize into two. First, as primary ones needed for the growth and the development of the plant, and secondary, which are not needed for the growth and the development. However, they have ecological role, ensuring the survival of the plants in its ecosystem. And that is good for us as medicinal value. So we all know that nowadays we are all enveloped with diseases, recurrence of diseases. You find new disease cropping up every now and then. You talk about disease in plants and all. So for me, metabolites that help to save you out, metabolites that help you to stay fit is very important. So that's why I think there is nothing secondary about secondary metabolites. Now, this is a kind of data that helps you to understand that how much important this industry is related to plants, herbs, and natural products. In fact, you can see almost close to 80,000 flowering plant species have been used for therapeutic activities globally. And the percentage usage of total plant species, it's good in India, followed by China, Sri Lanka, and US as well. So most of the Asian countries have been up to using plants and their products and other natural products as well for their betterment. We talk about United States. Out of the 150 mostly prescribed drugs, 79% are somewhere natural, and out of those, 74 are plant-based. In fact, in India and China, we all know plants have been used quite significantly. And this is a very good data that shows the export from India, right from 15-16 to, in fact, the latest one, 22-23. You can see the kind of export has almost doubled, right from approximately 300 million USD to 600 million USD. So again, as I mentioned just now, shortly, that it's a good venture that one can go ahead for exploitation, in fact, commercial exploitation. And the path is quite good as well, because the projected, data suggests that in 2050, it will cross somewhere $5 trillion US. So immense potential, in my opinion. In fact, WHO also recognized the importance of medicinal plants and natural products. In 1970, it started with the collection of data related to traditional medicines and all. And 2014 and 2023, they aim to integrate traditional and complementary medicine into healthcare system as well. So in fact, global recognition is there. This is the data, I think, a bit old, but this shows you that varied activities of plants and their products, and the cost is quite hefty. You'll be surprised to know Catharanthus सदाबहार कहते हैं हिंदी में इसको. You grow 1 hectare of सदाबहार and you get mere 1 gram, only 1 gram. And now, if that 1 gram is in your pocket, beware, you can be kidnapped because it costs more than $1 million. So that's the potential of plant metabolites and natural compounds. This is the game changer. The Nobel Prize in Physiology and Medicine 2015. In fact, Campbell, you talk about Omura, they all got Nobel Prize for ivermectin from streptomyces, basically. And my focus will be on Yo-Yo Tube because she is known for 3 no's. No means N-O. At the time she did her work, she was not even PhD. She never actually visited abroad for any studies. And she was also not member of any of the scientific agencies in China. But still, she is now a Nobel laureate. So, there's a short story associated with the kind of Nobel Prize and how she came out with anti-malarial drugs. She was given this task by the government of China, and more than 1,000 formulations she chalked out with her teammates. And finally, she zeroed upon to 640 approximately, if I'm not wrong, and all had the antipyretic effect. So they thought which is the main ingredient, in fact, the common ones. So Artemisia was the main and the common ingredients in that. And then she did work, and mostly, as you all know, kadha or decoction, we call it as in Indian system and Chinese system, it's very common. And she was working on this, but she was not getting a very stable result. Sometimes a very good one, sometimes it was not very good. So she was fond of reading books and all. She read somewhere that put the plant in cold water and then give it to the patient and patient will recover. And that struck her mind. She thought that might be possible. Higher temperature, she used ethanol basically. Higher temperature might be doing some wrong to that metabolite. And when she used, I guess, ether, and she came out with wonders, had the stable kind of activities, and she was very happy. So the point here is that more you get a deep dive in the sea of review, more chances are there that you'll be coming out with expensive pearls. Now, the 2 sides of coin, you have positives, you have negatives. So if they are so very important, they will be exploited by industries, by people, and other communities working on it. So, over-exploitation has also led to the entry of in the red data book, in fact, the 100 to 1,000 times higher than the natural rate of extinction. And they are being harvested at a very rapid rate. You talk about increasing population, urbanization, industrialization. There are several factors which are adding up to this issue. About 15,000 species are on the verge of extinction due to loss of their habitat and over-harvesting. So basically, we need a kind of dual approach almost that serve the purpose simultaneously. In fact, one that conserves the plants, and secondly, knowing the medicinal importance, also provide the raw material for various needs. Tissue culture can be one of the effective approaches. There are others, but I'll be focusing on this. And you can have growth, and you can have the clonal propagation of elite germplasm. You can also manipulate the conditions to have a kind of enhanced yield of metabolite of interest. So that can be taken care. Now coming to a couple of plants, being the constraints of the time, I'll be focusing on Acacia tora, which is called as sickle senna. Senna kai baar purgative ki tarah bhi use hota hai. Pods are sickle shaped, that's why the corn we called as sickle senna. It has multiple traditional activities like it's hepatoprotective. It's used again in dyspepsia, cardiac disorders, and all. And in China, Acacia tora tea is very much relevant, as you can see in the figure. So we went ahead with the callus culture initially, 2,4-D dichlorophenoxic acetic acid auxins were not able to induce callousing. However, when we increased the concentration, it showed a good callousing. And then we also tried some combinations using 3 auxins and kindin, and then 2 auxins. Best result we got in 2,4-D LA and carnitine callousing. So you can see the initiation one over here and the whitish off-white colored callus. Then again, browning of callus was very common. The scene you can see all of the media has turned out to brown together with the callus due to the phenolic secretion. There are multiple ways you can use antioxidants and all, but we tried a kind of easier approach. Basically, we subcultured at a very early stages, approximately 3 to 4 weeks, and we got good results. And the callus, as you can see over here, the media as well, sorry, was not having kind of phenolic secretions. This was the growth index, max at 6 weeks, and then it's got down. And then we focused on protenoids. We saw basically these are isoflavonoids having chromanochromone ring, also used as biopesticides. And FAO also has advised the use of the same. We isolated these compounds, metrol, ceftriaxone, rotenone. It's a very potent kind of insecticidal agent. Decurin, rotenol, and elliptone in different plant parts in callus culture. This is the table for thin layer chromatography, preliminary one, but why I thought of showing this is because normally we say and we assume that 100 degrees are fine enough, or 110 degrees are fine enough to get the spots after spraying the TLC. But here the main point is that unless and until you go to 120 degrees centigrade in oven, you'll not get the spots in the TLC. So that is a kind of peculiar characteristic feature of these groups of compounds. Now this is the GLC data showing again, and you can see the differential content of rotenoids maximally in roots and minimum in stem. Then we thought of doing the larvicidal activity. Basically, we focused on the larva of Anopheles. Malaria, again, we know that it's a common disease. For sure, we have done enough. The government has done enough to have a control on this disease. But still, it's there, and changing climatic conditions, synthetic insecticides being, larva and all mosquito being resistant. We should always be having some kind of good molecules under the belt so that whenever it is needed, we can use it as and when required. So you can see this is the activity. I see LC50 was 110. Pure rotenone had a very good 3.2 approximately. And when we thought of that, it's functioning quite fine. So we thought of eliciting, in fact, in the culture system itself. You can use elicitors as well, and there are precursors that can be used within the medium. Precursors, basically, we know it's the starting compound of a biosynthetic pathway. However, intermediates can also be used for this. And then we saw a good increase. In fact, we used phenylalanine and methionine, 0.05 millimolar concentration. Different concentration we use, but that clipped the maximum, and you can see the growth index got high, in fact, more than the control system. And then again, this is the content also increased almost in a similar manner. We had the sigmoid growth curve right over there. Now overall, the content was also more than the plant parts. It was, I guess, approximately twofold more. Second plant of interest is Tecomena, and actually why I'll be showing in fact a preliminary data, but it's very important because it has not been tried much as I see in the reviews and all. In fact, there's a story associated with it. I was just loitering in my campus and saw this tree, and I just went close to that tree and saw that the bark is totally out from that. Somebody has literally like scraped it out. Thought some infection and all, but it was manually done. So there was a guard passing by. I just asked why this tree is having this kind of scenario. So that person told, sir, ये बहुत important है. सर्दी खांसी होता है. Somebody is having some kind of female disorder, especially leukorrhea and all. सब इसको लेकर चले जाते हैं. Or this is also called as the desert teak or marwar teak in Rajasthan itself. Its flower is the state flower also. So since it is also commercially viable and important furnitures, good furnitures are made using this plant, people are growing it, but they cut it out and they sell it off. So it's again one of the endangered plants you can see, and commonly called as Rohida, desert teak as mentioned, honey tree also. We have several Ayurvedic formulations also in this, and herbal formulation, leaf 52, Levo Plus and all, having the content of this plant. So this is the point, as I mentioned just now. The bark has already totally been scraped out by the people. And wherever you see in Rajasthan, in fact, you will find this kind of same happening in the tree because of this no, medicinal importance. So our team just sat down and just thought to check out the ethnomedicinal importance of this plant. Mostly used in India and Pakistan. I'll be focusing data on India itself. So you can see that you talk about birth control, leukaria, as I mentioned, infections are one of the major remedy. You can say that using this plant. Digestive disorders, hepatoproductive activity is again one of the common that we found in most of the things. And mostly you can see here stem bark that is being used. Talk about other plant parts, you can say flowers and seeds and leaves, almost similar activities are there. And that's why people are taking out the bark mostly. That is just distribution. In fact, mode of administration, you see, mostly, it's oral, talk about in Pakistan. It's again almost 32%. And overall, you can see oral administration of this plant is very much, followed by topical. And this is the distribution in terms of plant part used. As you can see in India, mostly taken by stem bark, more than I guess close to 60%. If you talk about Pakistan, almost halfway down, I guess 25 or 30 as such in Pakistan. And other plants parts, sorry, also used. So again, since it is endangered, we did the tissue culture of this plant. In fact, the callus culture. These are some data which has already been reported. Very scanty report. In fact, I guess you will go to 1980s and all. You'll find some data from again one of the universities of Rajasthan in micropropagation. And thereafter, we did and tried several combinations. Kinetin was not conducive enough to initiate the culture. However, it was reported earlier, but we didn't get a good result in that. Then again, MS, BAP, and NA, and BAP and IA clicked for us. More than 95% we had the growth. So this shows in different combinations, individual and of auxins and cytokinin. This is the initial one, and it goes a bit more, the texture is more hard, you can see in BAP. And then this is BAP with 2,4-D. Again, this is only 2,4-D. Right, different concentration, higher concentration, we had lesser callousing. And then you talk about this BAPNA, 0.01 and 2.5 mg per liter. We use that because we had more fragile callus in this, and we are planning again to go ahead with the suspension culture using this plant. This is again the second hormone of our choice, again BAP and IAA. This was from the nodal X plant. It had more kind of callousing, as you can see over here. Most of the hormones were similar. Again, these two, matter of choice for us to work further. Then we did some quantitative analysis. Total phenolic content, as you can see, was good in plant parts and more in this callus as well, almost. Right, and this antioxidant properties we tried showed good results, but the nitric oxide scavenging activity was not good enough. However, others were quite fine. We thought to analyze a few metabolites from this, in fact, phenolic compounds, and we saw this data. In fact, this hydrobenzoic acid, syringic acid, naringin, vanillic acid, ferulic, salicylic, cinnamic acid. These are all present in different plant parts. Just quickly skip this. This data suggests the content, basically, you can see that in callus we had good content, in fact, more than the plant parts. And ferulic acid, cinnamic acid, and these. So that was good for us, and that's why I just told that we'll be focusing for the suspension culture. This was again DNA damage protection assay. You can see Probably not clear over here. Slight production was there, but here the C1 flower and the callus culture showed good production against the DNA damage. Since it was used for infection, cure infection, in fact, we tried several organisms, E. coli, Bacillus, 2 species, and Pseudomonas, and we got good results. That is shown in this zone of inhibition as well. And now we are just going ahead with the quorum sensing because this Pseudomonas is also for use for positive agent for urinary tract infection and all. So initial results are quite good, and now we are trying to go ahead with some docking activity also using some of the genes. Hopefully, we'll get good results. These are few of the more plants that we have worked on: Moringa, Acacia, Parentalis, Neem, then Gymnema, Asclepidaceae family. There's the Urea picta recently review published. Some bryophytes also we are working on. These are lab members. She's working on this Tecomella. These two, she's working on hepatoprotic activity. She's working on this byproducts of fruits and vegetables to come with formulations having antioxidant activity. Ruban is from University of Kashmir, and he is working on Elsia rosea. It's our ornamental plant. And recently we had the publication in scientific reports. These are some of the collaborations. So in fact, this is not an end, not even beginning of end, just an end of the beginning. So more part to explore and much to research during the course of time. Thank you, everybody. Thank you.
And you finished your talk within a specified time, and that was very nicely presented. And it was no doubt, there is no doubt that the phytochemicals are essential to our country. And recently we had participated in the International Immunity Day, 11th and 12th. Right. So there it was informed that approximately threefold increment. Initially it was $8 billion, but nowadays the people are turning up toward the herbal medicine, right, irrespective of that, either the Unani or the Ayurveda. So definitely there is a good catch, and we should definitely working in from our department. Professor Abdi work is tremendous in the field of biotechnology, because all the time we cannot get all kind of plants. So in that case, the role of biotechnology becomes very important. So the only issue what we are facing as of now, the dosage regime. Most of the time, because in the allopathy we are getting the very reduced form of the medicine, that the patient compliance is there. But when we are talking about the plants, irrespective of that betterment, the dose regimen, the quantity of the dose and duration, that is the major problem. How can we overcome from these problems? If you can enlighten us within a minute.
For sure. I'd like to just comment on this regarding Unani system of medicine. In fact, in talk, there's one doctor, just sharing my experience, in fact. I had a high TSH, right? So since I'm working on plants and all, I thought of curing it because, you need to take Thyronome or Thyroxine, and that is there all through your life without any cure. So I had some kind of dhania preparation over there. So it lowered down, first of all. And then I consulted that doctor to one of my friends, and he gave some formulations. I'm just asking what formulation actually he gave. And you'll be surprised, it dropped from 14 to 4. 14 is quite high, we should talk about. But that is the relevance and importance. And the point again goes with one drug and one target. Here in talk about Unani, Ayurveda, that is multiple drug and multiple target. We are now heading for drug repurposing. Why we are doing that? That is simply because this is all tried and tested clinically. So we'll be getting lesser time. Same goes with plant. We have been eating since years. We know which plant is good, which plant is bad. In fact, you talk about haldi, curcumin and all. We don't have good kind of formulation related to that. But that is stringent. Blood nikal raha to aap laga sakte hain. Infection mein kaam raha, multiple activities we are having. So I think our aim should be to have formulations using those plants or spices which are already there in the domain and we are eating. So that at least we can say that if we are consuming, there is not much of trouble as such. For sure, dosing is there. Anybody can have a higher dose of paracetamol also. That has to be restricted and kind of monitored. That is there for sure. And but the main problem is that the duration of sure. That is the main reason why people are having a kind of backseat. But my call is that if you are having a long duration of drug regime and you have alleviated the disease, what is bad in that? If it recurs after 7 days, is it good? So you need to kind of understand this, in fact, everybody, and formulations should be there. And we are having this manuscripts and all that is also there and tried and tested. So we need to have a kind of holistic view. Then only we'll come out with that. But still, thanks to COVID, everybody says COVID is bad, but I say it's thanks to COVID. We had no drugs. Everybody was on decoction, everybody was on kada, and people who opted that, I guess, they got saved. Because I have several kind of examples. People outrightly went to the hospitals, many times, no, they saw death. So that is the relevance.
I am not asking you, just comment that actually, my sir also worked on many demanding populations.
For sure, for sure.
And I was also a member of during the time of Professor Anand when he was chairman. So actually, I have seen that many of the so-called drugs, they are having many components which are not genuine. And still those foundations are there. We have already surveyed the markets from South India to North India, and in many of the foundations, more than 80% of the foundations, there were better rates. In some of the foundations, we have not found even, they were totally substituted by another. So, where consistency in the therapeutic efficacy of the foundation, if this is the state of affairs. Then the other thing is that when we are collecting the material from, because we are not cultivating them, we don't have good agricultural practices, nor we are not also having any quality parameters. When we are collecting material from the forest resource or from the wild, we are not sure that at what stage we are collecting the material, what plant part we are collecting. Actually, in some of the cases, we found that actually the recommendation is that we have to use leaf, but the whole plant part was there in the market. Even while we are transporting those plant parts from the site to the place where they have been stored and marketed, like during that period, many microbes they infect them, changing the whole life of the
plant metabolites.
So, through transformation of actually, there are a lot of problems. Actually, the AC of the herbal combinations are not only like, you cannot say that even if you have genuine plant material, you are correctly at the right time, but during transportation, there is a lot of change. Right? So, you will not be getting the batch efficacy of the, and that's why many mission mode programs, they failed. The Golden Triangle program involving ICR, ICMR, and the CSIR, that was failed. Dr. Kazi is there. You can ask the story from him.
Yes, sir.
So,
that's so important.
actually, that is my comment.
Yeah.
Actually, we need quality parameters from the initial stage where we are preparing Graha to the foundation development and finished product that we still need, that should be part of our farm. That's all.
That is very much important, sir. Rightly taken. Thank you.
One minute, sir. Now I would like to request to our honorable chairpersons to present memento to the honorable speaker, Dr. Sadwat, as a token of appreciation.
So it's my honor to introduce the next speaker, Professor DC Jain. I think he's a real expert, you can say a lot of experience from the industry. So I want all the students, to listen very carefully because here is a person who is from the industry with a lot of experience. So Professor DC Jain is faculty at Sage University. He served as a scientist at CSIR CMAP from 1981 to 2003, General Manager at IPCA Labs from 4 to 17, and has 36 years of experience, 22 in R&D and 14 in industry in total. So what more you want, you're going to hear right from the person who is having a lot of experience. So he pioneered Artemisia annua commercialization in India, and I hope, and I'm sure that you must be knowing about artemisinin, which is a very important anti-malarial, cultivating 300 acres and producing 10 MT of artemisinin annually, sir. Great. He established advanced extraction methods, developed new anti-malarial drug Artemether, and contributed to India meeting 1/3 of global artemisinin demand. So, this is very huge and big what he has achieved. So he has been honored with awards like DST National Award for Drug Artemether in 1999, IDM Award for Best Patent in Biotechnology, Dr. P.D. Sethi Award for Best Paper in HPTLC, and many more. So his topic is green extraction and purification technology for artemisinin, a sustainable approach to green gold. Thank you. An honor to.
मैं यहाँ पर ऑर्गेनाइजर्स का बहुत बहुत धन्यवाद करना चाहता हूँ और स्पेशली डॉक्टर साहब एमजेड आब्दीन ने मुझे एक्सपीरियंस को जो मेरी 25 साल की जर्नी एक पौधे के ऊपर, ये बहुत बड़ी बात है। और जिस पौधे के साथ में जिया और उसको जीरो से कॉमर्शलाइज किया, सबसे बड़ी बात है कि बहुत सारी बातें हो रही है, बहुत सारा लिटरेचर इस टाइम है। हम काम किसके लिए कर रहे हैं? हम दूसरों के लिए कर रहे हैं, पर हम उनमें से कॉमर्शलाइज कुछ भी नहीं कर रहे हैं। क्यों नहीं कर रहे हैं? क्योंकि हम उन पर ध्यान नहीं दे रहे हैं। हमने बहुत मेडिसिनल प्लांट पर काम किया है, हजारों पेपर हमने छापे हैं, और आउटपुट कॉमर्शियल तो 5% भी नहीं है इस देश में। 60% मार्केट तो चाइना लेके बैठा है। क्या हम इस देश में कमजोर हैं उस देश से? नहीं है। और मैं उसी एक एग्जाम्पल को कि जो पौधा, जिस पौधे को नोबेल प्राइज मिला है, पहला पौधा आर्टेमिसिया एनवा चाइना को, हमने भी 25 साल काम किया। फर्स्ट टाइम बहुत कंट्रोवर्सी हुई थी, वाई नॉट इन इंडिया। पर हमारे देश में हम अपने लोग, अपने साइंटिस्टों की भी कदर नहीं करते कई बार, तो दुर्भाग्य। और चलिए, वो कोई अफसोस नहीं है। और मुझे इस बात का खुशी है कि मैंने 22 साल गवर्नमेंट ऑफ इंडिया सीमैप लखनऊ में काम किया और 15 साल इसी पौधे आर्टिमिशिया पे काम किया। मुझे जिस दिन लगा कि मैंने पब्लिकेशन शो कर दिए, 50-30 पेटेंट कर दिए, फॉर व्हाट? हम ये भी देखते हैं कि हम ड्रोज में रख देते हैं, भूल जाओ। मैंने एक दिन देखा कि इंडिया दुनिया की 45 मोर देन 60% एंटी मलेरियल ड्रग हिंदुस्तान में इफ्का रतलाम बना, और रॉ मटेरियल 100% डिपेंडेंट ऑन चाइना, जबकि दिस मलेरिया इज आवर प्रॉब्लम, नॉट द चाइनीज प्रॉब्लम। तो हमारे लिए क्यों काम करेंगे? काम तो हमें अपने लिए करना है। तो मुझे एक अच्छा इस पूरे प्रोजेक्ट पे काम करने का मौका मिला। व्हाई इट इज ग्रीन गोल्ड? बिकॉज आर्टेमिसिया जो आर्टेमिसिन कंपाउंड है, there is no synthetic route available till now. This is a beauty of this compound. This is a very unstable compound, 5 oxygens present in that compound. And the Europeans working a lot to produce synthetic artemisinin. And billions of dollars they use to produce synthetic, and they're successful, no doubt at all. And they were, this is the bad news to the farmers for the Indians and Chinese. But no, ultimately, they don't reduce the prices of the artemisinin below $300. Where is the natural we produced that time in China also produced $200 in India and China. So a commitment hai, if you feel or if you can do, and that is the real problem. The plant-based drugs are costly. We are thinking about the three things. We cultivate, we develop a good varieties of the medicinal plant. This plant taught me a lot of things. When you harvest in the period of 9 months, 3 times artemisinin content increasing and decreasing. So which is a real harvesting time? Which plant? The plant contains zero of the artemisinin also. Not every plant has a different artemisinin content. And I showed some data of the farmers of 200 or 2000 farmers. Lot of, these are cross-pollinated crop. It's very difficult. So this is a real challenge of this work and this plant. So this is a one good example. A lot of work we have done from the, especially we done the good varieties for the field wise, not for R&D work. We have done a lot of R&D. Now we also work for the commercialization. Then extraction technology. You ask anybody, and I have seen a lot of industries other than IPCA, that everybody is the old methodology. A lot of new extraction technology is coming. Nobody interested in ultrasonic based extraction technology. Nobody interested in microwave. Nobody interested on the solvent based, solvent free technology. So we also use be cost effective. Until unless we drug is not cost effective, we can't challenge the synthetic materials. And that we achieved, no doubt. And I just show you some slides. And at present, I'm working as a just a commercial person, or you can say in the Sai University, as a professor, to teach only students to share my knowledge, to go for entrepreneurship, nothing else. But still, I'm working 2, 3 products. One is the anti-cancer, breast cancer drug, from the herbal, commercialize. We try to commercialize them also. And this is the artemisia. So artemisia annua is suit is a green gold we just already tell. And how we done this a green technology. What is green? It doesn't mean give everything green, but we reduce the solvents, minimize the solvent, cost-effective manners. We also prepare the losses. We've done a lot of losses, conversion cost. So this is also a cost green technology. A lot of solvent. In R&D lab is very 1 liter, 2 liter. But when I am going to commercialize this product, 10 ton per day, 10 metric ton per day extraction plant. And I use 1 lakh of liter of the solvent hexane. How much the loss? So that loss we are reducing by using latest technology, 200 to 20 liters. Not immediately. And that is why we are competitive against the China. We introduced this plant in 1985-86 in India. This is a plant in the cold climate plant, high altitude plant. Everybody is saying how you can do. We also started in Kashmir Valley. First time we introduced in India. But later on, due to terrorism problems, other problems, we shifted to the plain in Lucknow. And we developed this technology comparable in the this plain area of the. For commercialization, you want to cultivate large quantity in the high this field or forest. So in 1986, then this variety is developed, 4 varieties. When we first introduced this plant in India, the drug content is 0.01%. 0.01%. Then first variety 0.1%. After 5 years, next variety is 0.8%, and last variety is 1.28%, 1.2%. So this is a long journey, not a one-day journey. Or the whole department, whole CMAP institute working on this is a national program, and definitely we commercialize this product. Then finally we transport this technology to IPCA. IPCA is a leader, and they approach to us how we commercialize them. Then I take voluntary retirement from the government of India, and I switch over to IPCA. This is the plant, Artemisia annua. We cultivated in Baroda, and Gujarat, more than 1,000 acre land. UP, more than 2,000, Uttaranchal, Bihar, and other places. It's a very good crop. Starting from, this is 4 to 5 months crop, and starting in the in November or December, and in Gujarat, but in UP, you go up to February or March. So these are a lot of differences in the climatical condition. And when you harvest, when we analyze 9 months, the 3 times week, plus, minus, high, low yield. But farmers don't know. When you harvest, he told, then we finally decide you harvest 120 days when you introduce this plant in the field. So this is the maximum. Then in the big level, how you dry, shade dry, how is a shade drying? What is it? So we give simple technology to farmer, not taking the big technology, go to big dryers and this. Tons of material they produce. How is done? We simplify the technology. There is a one tree in your field, you shade dry under the tree. To me. So then it's commercially viable and possible for me. देखिए number of farmers 1,410. Total Artemisia produced 3,000 metric ton in a 1 year. And you just see the content variation from 0.5 to 1%. So first range of the farmers is 8% farmers up to the range of 0.5 to 0.6. Second is 0.6 to 0.69. 0.7. Behind this whole analysis, I think more than 4 to 5 years. Selection of the area and how we improve the average yield. Because some farmers in the 0.5 to 6, where we focus, we remove this area, where we improve, so that we can improve average 0.8 or 0.9. Because this much amount, so continuous improvement in this way, and it is not so simple ki this material, but I use the HPPLC in this. Yes, and it's so simple methodology. Nobody have told you ki how you go. Every day I analyze 60 to 70 samples. Is it possible? Because I payment to the farmers based on the content of the drug. Because industry wants to produce active material. So this is the benefit. Farmers give more biomass. No. So then one sample in SOX lab, you prepare in 3 hours, at least minimum 2 to 3 hours for HPLC analysis. For HPLC is also very costly. Even I have given the payment within month to this much farmers. This is my commitment to that farmers. So I use this microwave. Everybody, Dr. Jain, what you are doing? How? But microwave, we develop the sample preparation in 10 minutes or less than 10 minutes. Yes, that is why we did every day 60 to 70 samples and human. So you can solve. A lot of literature I read that time. Number of persons opposed to me, Dr. Jain, what you are using in microwave, agent sample preparation. No. Then I published this data and in present international form in UK, then acceptable. Okay, go ahead. So in this way, we can solve this problem, how to fast analysis and everything. And that is why we slowly, every year we improve the biomass. We just see this 2004 when I start commercialization after leaving CMAP, and up to 2017 I work for this project in commercial way. So you just start the biomass cultivation. Farmers are not adopting easily. How you convince payment basis? We simply give farmers, they want only the real payment and when you collect the milk. And buyback guarantee. We give the buyback guarantee to the farmers and collection from their farm. So this much amount we have collected, tons of material, tons of payment, average is this. In this way, slowly then we expand the area of cultivation, 7 to 8 state I start, and reach up to 3,000 metric ton. So in this way, or the average of this is 0.7. That I have showed the farmers different levels. In this way, we consistently improving our, this is commercially viable for us. This is the cultivation part we have done easily. Then we establish the cultivation. Then the second, we asked Dr. Jain, China is a challenge for us. China is a major problem to India. How you, so one is the improvement the quality, second farmers level, so biomass, then we start the extraction technology also working to how we reduce the cost. So we work this continuous extraction method and then purification by HFC salts to reduce the cost to be competition against China. Dr. Jain, you can't compete China. Everybody told me in number of seminars. Unnecessarily you are wasting your time. Okay, no problem. If management interested, I am also working, this is my interest. Okay, but we compete. Nothing is impossible, everything is possible. So this is a festoterpene lactone. This is an antimalarial combination therapy. This is the only drug at present available, and we need the high quality. The plant contains more than 170 compounds reported from this plant. And out of them, one compound we isolate, which is required 99%, is not so easy. And plant contains every type of compound. How it is possible? So technology is very difficult. Still we try and we develop. Because that time the literature is not available. Chinese literature is not. So whatever we develop in indigenous in CMAP, and then we commercialize in it. This is a simple methodology. We use traditional method, dry use, we extraction. Then I introduce this liquid extraction technology. This is very useful for the industry also. You reduce lot of impurities. So column cost in a year. Then we go for column chromatography, purification, and final pure artemisinin 99. For 1 ton give 4 kg, 5 kg. So it's still costly affair. And to be recovery in overall 60 to 70%. Every step is lost. And you just see left column is 1 kilo, 2 kg. I use 1 ton of silica gel in each column. And yes, 3 columns every day. Or 1 ton of silica gel is only 3 days I can separate. So this is a real challenge. From lab to scale up is also a big challenge, but we did, and slowly, this LA liquid extraction, we have selected, only we use this one solvent where we transport, and 50% of impurities we retain as a gel in one mobile phase, and we improve. At least silica gel ratio is improved 1:50 to 1:10, then we 1:3 and 4, then commercially wide. Then 1 ton of silica gel separation is really difficult, and silica gel is the major cost involved in this technology. Because when I start work in the CMAP and 40,000 rupees per kg is our cost. Director told me, Dr. Jain, how we compete China? I said, sir, you increase the yield, I am doing chemistry part. But raw material, silica gel, how many times you can reuse this silica gel? This is the best material. But I work in CMAP at the time is 10, 20 kg, and my director told me, Dr. Jain, this is not science. What you are doing? You are not appointing for this purpose. Okay. But I know one day that this is turned the commercialization and change whole scenario of this. And this is the real cost. Okay, I learned, but only this silica gel regeneration and the cost is 40 to 10,000. 42. क्योंकि when we regenerate at that time 250 rupees is the cost per kg of silica gel, and regeneration cost is 10 rupees, 20 rupees. Not only one time, I did 7 times. Negligible cost of the regeneration. That is why we reduce the cost from 40 to 10,000 per kg. In this way, we come into the against the China. You can also produce, you can also produce in the same. And IPCA, क्योंकि major producer है 40 metric ton of artemisinin purchased every year. So उन्होंने कहा Dr. Jain, don't worry, you do a lot of R&D. This is the facilities they given me. But we, India, at least 1/3 of that time, because already farmers, we extended the farmers, lot of confidence to the farmers, because farmers doesn't, we want money and you collect the material, nothing else, and payment at the time. So in this way we can improve. And after 10 years working, lot of competition to China. Column chromatography is a really tedious job. Because 1 ton every day is using regeneration, lot of plants and everything. So then we think how we improve this. Then I use extraction technology, not normal extraction, then I go ultrasound. Yes, from 100 liter to 2 kL reactor, then to 6 kL reactor. That reduce the time side. जो 76 days लग रहे थे in one base side. Thank you. मैं 5-7 मिनट बताऊंगा. 76 से 72 पे आया. So this is one improvement, and then we go finally continuous show you, and let's this HFC technology or CO2 technology is chromatography is a tough job. So I think which technology I can use, and I heard this technology 2010 in UK at one seminar, and in 1 gram, then we convert into 50 kg, and this is really a good technology for herbal extraction and purification. This is a comparison just like existing 6 kL to 2 kL, and this is also 67 hours to 12 hours. So ultrasonic is also very good, best technology, good yield and cost effective. Then this is a continuous extraction solvent that I developed. This is a plant which I developed. Nobody knows about the continuous plant. Everybody knows the soybean extraction plant. In plant material, herbal, I don't think anybody using 12 metric tons per day extraction plant. It is a fluffy plant, fluffy leaves and this. So after working on 1 or 2 years, then we developed. And the solvent losses, really 200 liters per ton losses up to 10 to 15 liters. Then management was really, this is a good work that is reduce the cost. And in 1 lakh liter per day solvent. But the plant has no smell. Very good continuous plant. तो कॉस्ट एक्चुअली काफी इफेक्टिव मुझे इससे मिला. Then I feel that this column chromatography another tedious problem. Then I use this HFC solvent and it get the better recovery and less effective. This is the indigenous plant we have developed and for trial and development and extraction. And I 10 kg I produce this artemisinin. Then we go for this commercial plant. Nobody in India can scale up, help to me to scale up this plant. Then I purchase this is a plant from Italy and use this thing. So HFC is a hydrofluorocarbon solvent, non-toxic, non-flammable, and is better than CO2. CO2 is a high pressure technology. This is a low pressure. This is the importance. And very selective solvent to remove, not extracting the any fat, chlorophyll, and other. So the product is very consistent of more purity. So 70 to 90% recovery is improving and more efficient, more economical. This is 1,1,1,2 tetrafluoroethane, R134 I am using, and not ozone depleting. And so this is easily available at that time. And we use it in cars also these days for it. So HFC say is a high pressure technology, lot of plant in India, but not CO2 plant. But HFC is very 6 to 8 pressure you can use this. Is get best lot of example for the essential oil and fragrance because there is no impurities or the solvents. And I also use in the rosas this technology, food flavors and natural product. So this is the plant. Plant we can also use directly HFC, no doubt. But in commercial level is very difficult because how big plant you need. So UK and other places I visited, they are using directly big plant for HFC for extraction. Okay, you can add. Then I change. No, I use first hexane extract or the extract. 1 ton, 50 kg. Then I can usually small units and purify. So this technology in this way can use only for purification, not for the extraction. So this simple extract and extract the HFC extraction, then crystallize the product and purification. So a lot of this big columns. So this purification technology. So this extract can also purify. This is the oil you get, and this is your product, and then 85% purity directly, and recrystallize to get 100%. So in HFC, I can say more selective than hexane, CO2 to wax and pigment injection, and approved food and pharmaceutical. This is very simple method. And high yield and improved variety, reduce the solvent consumption, environmental impact, safer, high quality artemisinin, and this plant is anti-cancerous. So, आने वाला future, not for malaria, but the cancer. The yield is also improving 80 to 90%. That is why the cost is reduced, and quality is also. In conclusion, we can say that this technology can be utilized to lot of medicinal plants to reduce the cost and good quality, rather than using column and this and that. This is a combination of three: farmers, CMAP, and industry. This is a public-private partnership. और ये एक पहला अवार्ड हमें इसीलिए मिला डॉक्टर अब्दुल जी के साथ, क्योंकि हम तीनों ने मिलकर काम किया था. और दिस फर्स्ट टाइम दिस award is only for public private partnership. Very good example, क्योंकि लोग तीनों को नहीं जोड़ पाते और बिना उसके कि आप हर्बल में इंडस्ट्री में भी कभी भी सक्सेस नहीं होंगे. लोग अपने अपने इसमें काम करते हैं सेल में। यदि आप एग्रीकल्चर को इम्प्रूव नहीं करेंगे, आप इंडस्ट्री को इन्वॉल्व नहीं करेंगे और इंस्टीट्यूशन को इन्वॉल्व नहीं करेंगे, तो आप किसी प्रोडक्ट को कमर्शलाइज नहीं कर। We also use some byproducts to reduce the cost, क्योंकि हर्बल इंडस्ट्री को यही जरूरत है। एक प्रोडक्ट निकाला 170 में से 169 को क्या कर रहे हैं, तब जाके आप एलोपैथी को या किसी को कम कर सकते हैं। हमने इसकी ब्रिक्स बनाई, 10 टन पर डे जो लीव्स एक्सट्रैक्ट कर रहा था उसका 10 टन ब्रिक बनाया एंड रिप्लेस 10 टन कोल पर डे इन द बायोफील्ड। 10 rupees per kg coal, 2 rupees per brick, 8 rupees saving. Overall cost is $30 to $40 per kg reduced the cost of. Then oil is also a lot of oil, essential oil, mosquito repellent, gels and this. So this is the costing of this bricks. Thank you very much.
Thank you, Dr. Jain. It was really a nice journey. You're the pioneer of the Artemis scene, and we have seen since last 20 more than 50 years, Rapid is also working, and we just saw here also working with you. And it's definitely a need that we have to look up for the commercialization of the various things. And since this work is tremendous, as you have mentioned, the future of the Artemis scene is very bright, because this future is going to be used for the anti-cancer treatment as well. I believe there may be many more activities and the pharmacological activities may be lying with you.
Lot of activities. Even the plant material is not available in the country. After 85, plants, a lot of research is going on already. And just like in also UK, they develop, Chinese also doing, we are also working on the improvement of the yield. If one day when the 1.5 or 2%, still we can beat China. No problem.
So it was very insightful for our students, and I believe after this kind of presentation, our students will be motivated and they will definitely work hard to extract out not only from the artistic, from the other plants also. Thank you.
Thank you so much.
Thank you.
She has done. Sir, just one question, that did you also give the technology to the farmers, that what changes should be in the soil and other things?
No. मैम वैसे मैं, क्योंकि देखिए एक तो हमारे साइंटिस्ट लोग भी हैं, टीम में काम नहीं करते हैं देश। दिस इज द रियल प्रॉब्लम। एक डिपार्टमेंट दूसरे डिपार्टमेंट के साथ कोलैबोरेट नहीं करता। आई एम द ओनली पर्सन इन सी में, मैंने 10 के 10 डिपार्टमेंट के साथ काम किया। और वहां से मैं एग्रीकल्चर सीखा, वहां से मैं बायोटेक्नोलॉजी सीखा। मैं तो बेसिकली ऑर्गेनिक केमिस्ट्री, वहां से मैं इंजीनियरिंग सीखा। और उस पूरी टीम में मैं लीडर नहीं रहा कहीं भी, मैं एक वर्कर रहा, पर मैं इस प्रोजेक्ट को कंटिन्यूटी में लाया। तो मुझे लगता है कि असंभव नहीं है। और टेक्नोलॉजी ट्रांसफर हमें मालूम है, इंडस्ट्री को जब टेक्नोलॉजी ट्रांसफर करते हैं तो बहुत गालियां भी खानी पड़ती है साइंटिस्ट को, मिलता कुछ नहीं है।
Do you think जो टेक्नोलॉजी दे तो हम yield को और इंक्रीज कर सकते हैं?
यस, इसलिए कर सकते हैं कि मैम मैंने बहुत से फार्मर्स को जब हमने दिया तो हर एरिया में गए, बिहार में गया, यूपी में गया, गुजरात में गया, साउथ इंडिया आल्सो आई ट्राई, एंड आई सीन कि सम ऑफ द फार्मर इज मोर साइंटिफिक प्रोडक्ट प्रोड्यूस देन आवर्स। दे टोल्ड मी डॉक्टर जैन इसका भी चेंज किया जा सकता है, क्योंकि इस प्लांट ने बहुत कुछ सिखाया है। टिश्यू कल्चर लैब, बायोटेक्नोलॉजी और फार्मर्स, क्योंकि कल्टीवेशन प्रैक्टिसेस आल्सो चेंज लॉट। और हमने ट्रेनिंग दी फार्मर्स को, खड़े होकर ट्रेनिंग दी। हर साल मैं एक विजिट हर फार्मर के फील्ड पे करता हूँ, इसलिए मुझे बहुत डेप्थ नॉलेज भी है, बहुत कुछ किया मैंने इसका। थैंक यू। I would like to invite our third speaker, Professor Abdul Latif Sahab. He is a distinguished expert in Unani pharmacology and is recipient of Sheikh Zayed International TCM Award 2024 and Ajmal Khan Award 2017. Formerly, he was associated with the Aligarh Muslim University. He has led multiple CCRUM projects, pioneering herbal drugs and standardization, fermented dosage forms like Navis, and innovative teaching models. His work integrates traditional Unani medicines and modern science, including a UV lamp and vitiligo as psoriasis treatment, advancing global recognition and validation of Unani pharmacology. I invite Professor Abdul Latif with a request. We have only 15 minutes because we have to, valedictory program is already, please. Valedictory program is also scheduled from 5:15. So I request if you could finish your talk within 15 minutes, will be good. Please.
So this is for forward and this is for backward. This is forward and this is for indicator.
नहीं यहाँ से भी तो है ना बैकवर्ड
यहाँ से भी कर सकते हैं।
ये है ना ये आगे जाएगा
और ये पीछे
ये जी
से बटन से भी कर सकते
यहाँ से ले ऊपर कर बिस्मिल्लाहिर्रहमानिर्रहीम Thank you, Professor MZ Abdin, for inviting me here, giving this opportunity to be here. I am here to share my thoughts on the global crisis of antimicrobial resistance. I strongly believe that scientists must prioritize developing a safer antibiotic from the abundant plant sources surrounding us. तो बढ़ाइए थोड़ा सा मैं आँख पे देखने में तो मुझे यहाँ से देखना पड़ेगा आँख में प्रॉब्लम है पर
चेंज से हो जाएगा इसी से चेंज हो जाएगा सर
ये
नीचे वाला
सर ये बीच वाला
नहीं
सर पढ़ने में इशू हाँ हो जाएगा
नहीं मुझे करीब से
मुझे
देखना
एक मिनट जाएगा पढ़े
भाई
नो सर हो जाएगा कहाँ गया डुप्लीकेट नहीं जूमिंग का नहीं है सॉरी
कोई बात नहीं मैं देखता हूँ उधर से करूँगा थैंक यू Come to the introduction. Rising threat, antimicrobial resistance (AMR) has sparked a global health crisis. Unani medicine, a centuries-old traditional practice, presents a viable solution. This paper explores the necessity and challenges of developing Unani herbal antibiotic as an alternative to conventional treatment. To preserve the bioactive compound in Unani herbs, particularly those sensitive to heat, we emphasize, just already has spoken by Professor Jan, emphasize the importance of low temperature extraction, eco-friendly extraction method, and microwave assisted extraction. Our antimicrobial screening of 80 Unani herbal medicine has yielded promising results. Further research, including clinical trials and biotechnological innovation, is crucial to develop effective alternative antibiotic. This paper concludes by highlighting Unani herbs
antibiotic potential in combating AMR and advocating for interdisciplinary collaboration to further explore this critical area. Antimicrobial resistance of antibiotic is ticking time bomb. The World Health Organization warns that AMR could claim up to 10 million lives annually by 2050. The overuse and misuse of antibiotics have accelerated this crisis. Conventional antibiotics are losing effectiveness and new ones are scarce. We urgently need innovative solution. Unani medicine, with its rich history and emphasizes on natural healing, offers a glimmer of hope. Antimicrobial resistance AMR Week was celebrated in November 2024 to raise awareness about the growing threat of AMR and to promote responsible use of antibiotics. Here are some reasons why AMR Week is observed. Number one, AMR, rising AMR cases with modern antibiotics. India has reported a significant increase in AMR cases, making it a major public health concern. Number two, high antibiotic usage. India is among the largest consumers of antibiotics globally, contributing the AMR problem. Number third, limited awareness. Many Indians are unaware of AMR's consequences, highlighting the need for education and awareness. Number four, inadequate infection control. Healthcare associated infections are common in Indian healthcare setting exhibiting AMR. Number five, need for antimicrobial stewardship, promoting responsible antibiotic use and antimicrobial stewardship is crucial in healthcare. Number six, government initiative. The government of India has launched initiatives like the National Action Plan on AMR in 2017, 2017 and 2021, and the national programs on AR containment. Number seven, global alignment. India has participates in the global efforts to combat AMR, aligning with the World Health Organization and other international organizations. By observing AMR Week, India aims to reduce antibiotic misuse and overuse, improve infection control practices, enhance antimicrobial stewardship, promote research and surveillance, educate the public and healthcare professionals, mitigate the spread of AMR. Unani medicine, a complete science of health. Unani medicine is based on the principles put forward by Hippocrates. He was the first established that disease was a natural process. Its symptoms are reaction to the body, to the diseases, and the chief function of physician is to aid the natural forces of the body. In Unani dictum, health is harmony of humerus is the only valid point of view if one desires to cure the cause of a disease. Unani system is synthesis of Greek and Arab medical tradition. It incorporates concepts from ancient Greek philosophy, but it also absorbed ideas and practices from the Islamic Golden Age when Arab physicians made scientists significant advancement in medicine. This period saw the integration of knowledge from various cultures, including Persian, Egyptian, and Indian medical tradition. The fusion of these influences made Unani medicine a comprehensive system that combined practical treatment with a strong philosophical foundation. Unani medicine's connection to plant and herbal remedies is also deeply rooted in its history. Ancient texts such as Kitab al-Hashad by the Sughrais, Kitab al-Jamil al-Faradat, al-Adbiya wal-Aghziya by Ibn Baitar document the use of medicinal plants. These texts provide valuable information about the identification, properties, and application of over 1,500 drugs derived from plant. Medicinal plants have been central to the Unani medicine tradition, and many of the plants used historically continue to be relevant in modern medicine. Approximately 25% of contemporary drugs are derived from plants, with many of these plant-based compounds forming the basis of the modern pharmaceutical. Medicinal plants are widely used today, especially in developing countries where they are valued for low cost, safety, minimal side effects. Unani medicine is focused on nature-based therapies has contributed to continued relevance. Plant-based drugs such as quinine, morphine, vinblastine, originally identified through Unani and other ancient medical systems, remain essential in modern medical practice. The rise of antimicrobial resistance, AMR, is critical global health challenges. So this addresses the issue by emphasizing the importance of natural Unani antibiotics, which are safer alternative to modern system synthetic antibiotic. In light of AMR, the paper advocates for prudent use of antimicrobial agents and encourages collaboration across sectors to prevent resistance. Challenges in developing even an herbal antibiotic. Despite of despite the potential of Unani herbal antibiotic, several challenges needs to be addressed. These includes number one, standardization of herbal extracts. Ensuring the standardization of herbal extract is crucial to guarantee their efficacy and safety. Number two, extraction technique. Developing efficient and sustainable extraction technique is essential to preserve the bioactive compound of Unani herbs. Number third, thermolabile compound. Many Unani herbs contain thermolabile compounds which can degrade when exposed to high temperature. Now, clinical trial conducting the clinical trials of to evaluate the safety and efficacy of Unani herbal antibiotic in human is essential. Now come to the green detection techniques. Already explained by the Jen Saab. How to get extraction of low temperature to avoid destruction of thermolabile antibiotics in Unani herbs. These are just ultrasound assisted extraction, microwave assisted extraction, critical fluid extraction. Benefits and challenges. Unani herbal antibiotic offer numerous benefits, safety, efficacy, minimal side effect, and cost effective. However, the challenges remain standardization, clinical trial, regulation, education. Addressing these challenges will require collaboration between researchers, policymakers, and healthcare professionals. These slides demonstrate the antimicrobial screening of Unani drug conducted in the microbiology lab of Department of Immunology via AMU. Now come to biotechnological solution. Biotechnology offers several solutions for development of alternative antibiotic from Unani herbs. These include plant tissue culture, using plant tissue culture to produce large quantities of bioactive compound from Unani herbs. Number two, by genetic engineering, using genetic engineering to enhance the production of bioactive compound in Unani herbs. Number three, nanotechnology, using nanotechnology to develop large targeted delivery system for Unani herbal antibiotics. Come to the conclusion. Need for adoption of Unani antibiotic. The adoption of Unani antibiotic is crucial in context of rising antimicrobial resistance (AMR), growing reliance on herbal medicine in Asia. Medicinal plants have long been used in Indian subcontinent for their effectiveness, low cost, and minimal side effects. Unani medicine, which utilize over thousands plant species, offer a rich tradition of treating infectious diseases with natural remedies. These plants, many collected from the wild, have shown promising antimicrobial properties with numerous studies validating their efficacy. I have given the order by the chairman to finish, so I am to
Request.
so thank you very much.
For the insightful talk. The only concern which I would like to ask, many of the medicines as you have mentioned here are being used for the treatment of the bacterial infections. But I personally have seen several medicines kept in the store, the herbs, as a crude form, the bacteria and the fungus develops into it
That
itself.
is an accepted. That is an
And
admission. You are sure.
yes.
But it's not concerned with the paper.
Yes. So how can we
This
eliminate
is
those
a conversation and preservation is another department.
Yes, definitely.
It's a technique.
Agree.
So
My only
that
concern
should be
is
followed.
yes.
You go to the Khauri Bauli.
Everything is
See
available.
that they are what?
Fungus and the bacteria.
What this government not doing? What is
Doing nothing.
why not doing?
They must. They must.
उनको पकड़ के जेल में बंद कर देना चाहिए। हाय मैं गया मैंने कहा क्या कर रहे हो हमारा नहीं हैं?
The fungus is growing
तुम
onto
इतना
the
बोल रहे
herbs.
चूहे इतने बड़े हो रहे दिल्ली के बराबर के? अरे अरे भाई कैसे होता है कैसे देखिए तो देयर इज a community. हम India especially I'm telling you sir to all of you I am a Unani person but I am very sorry to say I use only Hamdard drug not my own Dawkhana drug.
So we are simply asking what is your suggestions the people who are preparing the medicine out of the herbs and if it is rotten, so how can we
Then this if we if we will see the antibiotic properly, we should take fresh medicine. Fresh medicine is available. Go to the cultivating field and get collected. Then apply.
is there any method for the disinfecting of
Yeah.
those medicines also if you have gone through it?
That
Like a separate part. It
That
is
is-
not related to you. Thank you. Thank you, sir. Thank you.
Very good. आपसे डिस्कस करेंगे, हमें कुछ बताइएगा। जी जी। जी, 2 साल पहले आपके यहाँ संसद में काम किया है।
So we have reached to our conclusive part. Our last speaker is Professor Anwar Shehzad Sahab. Professor Anwar Shehzad is a distinguished scientist from Aligarh Muslim University. He has 30 years of extensive research experience in plant tissue culture. He has successfully conducted studies on in vitro regeneration and conservation of more than 80 medicinal plants and 25 endangered species. He has successfully achieved somatic embryogenesis, conducted various synthetic seed production, and collaborated on antimicrobial studies of medicinal plant extract. He has more than 150 publications of high impact journals and 15 awards and fellowship. He has extensive academic contributions and he has served more than 60 administrative role in the AMU and he is serving as a reviewer of more than 50 international journals. I invite him with all respect. He is supposed to deliver a talk on EMS induced in vitro mutagenesis and characterization of high secondary metabolites producing lines of Tylophora indica. I welcome Professor Anwar.
Thank you so much for nice introduction. First of all, I must.
Only request is time is 15 minutes.
I will take about 10 minutes.
Thank you. Thanks.
So, first of all, I will. I'm very thankful to organizing committee and especially Professor Zainul Abdin Sahab. He has given me to share some of my research here in this platform. So, I will be concentrating on the title, that is EMS induced in vitro mutagenesis and characterization of high secondary metabolite producing lines of Tylophora indica. As you know, Tylophora indica is a very medicinal plant and that is being used in Unani system of medicine and Ayurveda since long. And a very well-known plant, it is having so many alkaloids and other things. But problem is that the medicinal plants are not gaining any recognition from the agriculture scientists, breeders, and others, so that improvement could be done properly. And to enhance the secondary metabolites in a proper way, or to increase the chemical compounds in the plants. And in other crafts, scientists are concentrating and they are mixing other genetic material to get multiple products in one frame. But in case of medicinal plant, it is lacking. So, and for micropropagation, as you know, medicinal plants are being cultivated from the wild. They're not getting proper recognition for their agricultural production. So this is one problem that genuine materials are not being collected and efforts are not being done to replenish the wild population. And they are being diminished, they are being rare now. So one of them is Tylophora indica, which is belongs to family Asalpidaceae, and that is now referring as endangered. So for the propagation of this plant through tissue culture, so many workers have done, and one of my lab is there. We have published so many papers on this Tylophora indica, and from here, from Hamdard also, Professor Mujib has done on this micropropagation work of Tylophora indica. But now slightly I changes the direction of this research to use the EMS for the generation of variability in the regenerants of this Tylophora indica. So may I take? यहाँ से मुझे खड़े हुए बोलने नहीं आएगा. Yeah. So this one is Tylophora indica.
As you know, it is known as Nandamur and so many glides are there. It is grown in North East India, Central India, and so on. And it has medicinal property because of several compounds. Some are like your tyloporin, and tyloporidine. These are having the anti-cancerous, anti-inflammatory, antioxidant, and immunomodulatory activities. Okay. Many. So mutagenesis just for your information that I'm going to have some help of this disease. Why mutagenesis? Just I told you that several plants do not undergo segregation and have very slow regeneration potential, which cannot be considered ideal for the selection procedure. For such plant modification in the mutation breeding, mutation breeding technique was needed, which manifested in the form of in vitro mutagenesis. Because if you are exposing the seed to the mutagen, that is a normal process of mutagenesis. But to improve the process, in vitro culture was in vitro technique was taken. That was taken up for mutagenesis. In vitro mutagenesis technique that uses chemical or physical whatever. Using the tissue culture technique, a large number of propagules as explant can be subjected to physical chemical because we have already developed to production of large number of propagules that could be exposed to the mutagen under in vitro condition. So because of that only we have started this study. Why EMS? Ethyl methyl sulfonate is a sulfonoxyalkyl alkane is a clear colorless organic liquid is carcinogenic. ये है इसको जानना जरूरी है. If you are using any chemical, you must know its activity. So this one is highly carcinogenic. You have to use it carefully only. And EMS has wide mutagenic effect, and it is all well known that already mutation scientists have given the data that this is highly mutagenic agent, other than the EMS and your sodium azide and other. So EMS is good. EMS also has the potential to produce single stranded break in chromosome and ethylate chromosome protein, or 90% of the changes caused by EMS are GC to AT transition, 1% AT to GC transition, and 2% are transfers, and 4% are the. So these type of properties are there of this EMS. Because of that, we have adopted. EMS has already been used to induce mutation in modern plants, for example, Arabidopsis, Solanum persicum, and crop like Rhizos fiber, Trichomestivum, and medicinal plant like Caphanthus rosis, and so many. So in our case, we have this following objectives. To study the effect of EMS mutation and morphological parameter, to conduct cell analysis of whatever changes happens in the leaf tissue, and then compare the analysis of phytocompounds that are present in the leaf tissue from the regenerated and mutated population, and then to compare genetic variation between mother and in vitro raised plant and plant obtained from the in vitro. So this is the methodology. First, we have to induce the callus from the leaf explant, and then not nodal, it is leaf explant, inoculated on 2,4-D. 2,4-D is the good hormones auxin for the induction of the callus. So we have taken 2,4-D, and that works on any 5 micromolar concentration. Best option was 5 micromolar concentration, like this. This one is the different concentrations of 2,4-D. And among these 5 micromolar concentration given 100% regeneration in the callus inducing product. And then these calli were transferred on, this callus proliferation was checked on EMS treatment. The different concentration of EMS was given in the medium, and then calli were subcultured, right? So these calli were subcultured on 2,4-D, then it is 5 micromolar, and this is the EMS concentration. So on this particular concentration of EMS, a considerable amount of callus frequency, callus inducing callus survival frequency was there, 68%. And on from 0.4 and 0.5, it drastically reduced. So we have taken this particular concentration that
This is the 80-50 dose. You consider that near to the 80-50 dose, and that's why this concentration was taken for further research. So we have taken all the concentration. This was the best. Then the calli which were obtained on this on various EMS treatment, 0.1, 0.2, 0.3, 0.4, 0.5, those calli were transferred on PGR degeneration medium for the induction of whatever embryos were produced for their differentiation, so that they may come out and germinate on the medium. For that purpose, we have transferred those calli, as we have already considered that 2,4-D has induced the somatic embryogenesis. Because this experiment was already done in previous studies. The 2,4-D is very good for the induction of somatic embryogenesis. So, induction of somatic embryogenesis, formation of somatic embryogenesis was already started, and thus that was needed to take its next morphology and germination. For that purpose, this was transferred on these media on brief 5 micromolar concentration. This one is the percentage of this production and maturation of the somatic embryo. This one is number of mature embryo in this non-mutagenic line. That is control. And then 0.1% is showing 38.4 and so on. And 2% giving 47% and 3% giving 52%. Right? And next to this, at 0.4%, it reduced to only 30%, and then further reduced to 20%. So this was the makeup, and we find that 0.3% EMS treatment was good. Then containing the 0.3% EMS production of embryos were good, and they supported the embryo germination also. Fortunately, you can say, because EMS is there, and that is supporting the germination and maturation of the embryo also on this medium. Then those, whatever embryos were developed, they were transferred for further development on this medium, so that we can get embryo, right? Plant, you can say, because when embryos transferred into plant, that is called embryo. So to get the embryo, those cultures were transferred from EMS1 to this germination medium to this germination medium, and it was found that 62.6% germination was there, and conversion into plantlet was 59%. 59, any number of. So from 0.3% EMS, this was good combination and concentration for the conversion problem. These are the photographs which are showing some embryo initiation. These are the embryos, and this one is the concentration H. H is this one. This one is, which is having the maximum embryo development. And this one is also germination, in embryo. This is photograph which shows germination of embryo on EMS, E and F. These two are 0.3% EMS treated somatic embryo maturation on MS plus BA plus IBA plus GA for after 4 weeks. Here you have this culture is a 4 weeks old culture, right? That's 4 weeks old culture, right? Yeah. So you get 0.3%. This is the 0.3%. Yes. So they are also supporting growth of the embryos properly. And this one is isolated portion. This one is 0.3 is D. So, and germination of this germination on EMS treated somatic half strength MS medium. Various medium, right? Full strength as well as half strength. So on half strength medium, this was the conversion. Then this one is your morphological structure after isolation of the embryo. So this one is the control. A is the control, and 0.1% this one is showing B. 0.1% EMS, and this one is 2%, 3%. So 3% is more heavier. When the plant medium were supplied with 3%, while 0.3% EMS, they were showing more healthier embryo production. So this one is from 0.3%, and this plant is 0.3%. It is having the broad leaves also and healthy growth. Similar is the case here. Similar analysis was done for the morphological study of this leaf tissue because we are extracting the alkalides, metabolites from the leaf. So for that purpose, we must know that what types of anatomical changes or surface morphology is there. So in this case, you will see that this one is the control, which is showing the trichome. Trichome is present and large number of stomata are there in the first case, which is control. And this one is in vitro control. In vitro control, number of stomata is lesser than the control. And they are not showing any trichome. But as soon as 0.1% EMS was given, or 2.2% or 3%, this one is 2%, this one is 1% and 2%. So they are showing any development, further improvement in stomatal presence as well as the trichome. Small trichomes are there as compared to the big trichome in control, and no trichome in vitro. But in 2% they are having more in number, and in case of third, three, more sturdy, more healthy trichomes are there, as well as number of stomata is equivalent to the control one. And immediately after 0.4, this number of stomata reduced drastically, as well as the trichome. Flaccid trichomes are there in here, and no trichome is there at 0.5% percentage of the units. Okay. So these are the conditions in the mutagenized population. Okay. नेक्स्ट में इसी फोटो देख लीजिए तब भी समझ में आ जाएगा. तो दिस ये देखना जरूरी है. In the first case, this one is the control plant, which is having how many 11 alkalides, right? After GCMS analysis, 11 alkalides are there. Next is the control, in vitro control, which is having 12 alkalides, and this one is having 0.1% EMS. The alkali content is 20. And these pink color compounds are common in all, from control to in vitro control to 0.1 to 3, 4, 5. See, this is having the R7 control and 20/20. And in case of 0.2, it is 25. And in case of 0.3, it is 32. And as soon as you are reaching to 0.4, the concentration, the number of the alkali is drastically to this scale. Only 9. And at 0.5, it is only 6. But the major compounds which are present in the control, they are. But in case of 0.3 percent of the EMS, number of compounds are so many, and many compounds are having similar types of activity. So if one compound is there for one activity, and you are providing 3 to 4 compounds of similar activities, so enhancement of the effectivity will be there. To suppress the element, to clear the element, there will be number of compounds, 3, 4, 5 in place of 1. They will be more useful. We will see this character in the next generation, second and third generation. Only then we will come to conclusion that this is a new one. This one is the first one. तो ये इसके हुआ है सारे. This is वो स्कॉट प्राइमर भी किया हमने, पारीमार्फिक बाइंडिंग पैटर्न आया, और इसमें देखें इन केस ऑफ स्कॉट 3 that shows 17% polymers. Right. This is the, and dendrogram is showing that this your T3 is far away from the control and in vitro. So this is confirming this activity that if some changes happen and they have produced so many alkalides, and those alkalides are also, you can correlate with the morphological parameters. Thank you so much.
Thank you, sir. As said, you have completed your talk within a specified time, although it was not 10 minutes, it was 17 minutes. But it was very informative and short and precise. Only one thing, as you said, it is carried, it can be conducted onto the in vitro. Is there any provision in vivo can also in the consoles?
This is being done. But not in this plant, another plant is being done. But the frequency of having 6 less. Very less, as compared to this. In this one, there is no any report, but we are trying to have some good result in this generation. If it will persist there, only then we can come.
Is there any chances of the commercialization?
After getting this new M3 gen, 0.3% EMS plants, so we can have a patent of this plant that can be used for commercial purpose. Yes, yes, suppose.
Can commercialize?
Yes. We can release as well, right? If it will persist up to 3, 4 generations, this is the yes. Thank you so much. Thank you.
Now I would like to request Honorable Chairperson to present memento as a token of appreciation to our esteemed speakers, Professor Jain sir, Professor Latif sir, and Professor Ahmed Shahjaz sir. Please come on the dais and receive the memento. चलेगा इधर से हाँ आप अनाउंस कर रहे हैं
Professor Latif Saab, if he is available here, please come and join on the stage for felicitation.
चलेगा I would like to invite Dr. Saima Wajid ma'am. Please present memento to our honorable chairperson as a token of appreciation. Abidin sir, please Abidin sir.
बहुत ज्यादा लग रहा है आप जो है बहुत शुक्रिया
Now I request the chairpersons to please felicitate the moderator, Dr. Salik Noorani, with the memento. I request all the speakers, chairpersons, and Dr. Salik Noorani for a group photograph.
Yes, very.
Thank you.
Now we are having 15 minutes for the tea break and we will resume with the valedictory function after 15 minutes.
I request, please be seated everyone. Assalamu alaikum and a very good evening to everyone. So we are about to begin the valedictory function. For this I request Dean Academics Professor S. Raisuddin sir. Professor. I request Dean Academics Professor S. Raisuddin sir, Professor M. Dabdi sir, Uniting Secretary, and the Head Department of Biotechnology, Dr. Humaira Faruqi ma'am, to please join us on the dais. Thank you, sir, for taking out time and gracing this event with your presence. Now, I request Professor M. Dabdi sir, the Organizing Secretary, and Head Department of Biotechnology, Dr. Humaira Faruqi ma'am, to please felicitate Dean Academics Professor Raisuddin sir with the band. Simon ma'am, please come up on the dais. Thank you so much. So we stand at the culmination of this remarkable journey of National Conference cum Workshop on Sustainable Biotech Solutions for Global Challenges. It has been a privilege to witness the wealth of knowledge, insightful discussions, and invaluable contributions that have unfolded over the course of this event. It has been a privilege to witness groundbreaking ideas, diverse perspectives, and innovative solutions that hold the potential to address some of the most pressing challenges in biotechnology, sustainable agriculture, healthcare, and environmental sciences. From the inaugural session that set the stage for this intellectual journey to the plenary talks, technical sessions, poster presentations, and the hands-on workshop, each moment has been a step towards scientific progress and collaboration. This conference brought together over 12 plenary speakers and 55 invited speakers from the premier institutions across India, fostering enriching discussions on cutting-edge biotechnological advancements. We were also honored to have over 50 posters, more than 10 oral presentations, and 15 enthusiastic students who actively participated in the workshop. We express our heartfelt gratitude to our eminent speakers whose wisdom and expertise have illuminated our path. Your insights and experiences have inspired us all, leaving a lasting impact on our academic and professional pursuits. A special appreciation goes to all the participants of the hands-on workshop. Your dedication, enthusiasm, and eagerness to learn have been truly commendable. I now request all the 15 students who actively participated in the workshop to kindly come up on the dais for a group photograph. I am calling the names Vansh Vadera, Muzna Naeem, Jyoti Sharma, Musabur Rahim Khan, Ashfi Behra, Nagaraya Raja, Arnav Sansar, Poonam Dev, Chaili Tyagi, Ikra Ali, Swati, Sana Khan, Sadia Kamar, Krishna Kumari, and Tapasyam Kumar. Please join us on stage for a group photograph, please. सबको सेटअप किया सब कुछ ऑडियो
Take one.
A big round of applause for these students. Thank you all. An exciting part of this conference was the poster and oral presentation competition showcasing the innovative research of young minds. Our esteemed felicitators meticulously evaluated these entries and we are now delighted to announce the winners. I invite our distinguished felicitators for the oral presentations, Dr. Azhar Jabeen ma'am, Dr. Nidhi Agarwal ma'am and Dr. Javed Ahmed Sheikh sir to please come forward and join us on the stage.
So it's my privilege to announce prize for the world of first prize is awarded to Mr. Ross. He is a scholar from the Department of Biotechnology and the supervision of Professor Islami. And the second prize goes to Miss Unamre. Friends, we have a tie. It's Miss Jyoti Sharma and Miss Shazia.
छह महीने आप रखिए 6 महीने
Thank you so much. A heartfelt thanks to our esteemed facilitators for their invaluable time, expertise, and dedication in recognizing and honoring these exceptional contributions. I now request Dean Academics, Organizing Department of Biotechnology, please felicitate our esteemed felicitators with a token of appreciation. Thank you so much all. Now I request our distinguished felicitators of the poster presentations, Dr. Simon Majid ma'am, Dr. Jagati Nadan ma'am, and Dr. M. Iqbal Al Ansar to please come forward and join us on the stage. I will take the privilege on behalf of my fellow colleagues, Dr. Iqbal and Dr. Jagati Naran, to announce the awards for the posters. There were 50 posters, and the posters were evaluated by renowned judges, Dr. Niranjan Chakravarti, National Institute of Plant Genome Research, Dr. G.S. Tuteja, former additional DG ICMR, Professor Prashant Mishra from IIT Delhi, Dr. Pratima R. Solanki from Special Center for Nanoscience, JNU, Dr. Iqbal himself, and Dr. Saif Hamid from Amity University. It's time to now announce the awards. We had divided because it was really very difficult for the judges to really give away only 3 prizes. So what we had done, they had done rather, that the prizes, they were divided in 2 categories. One category on the basis of the range of marks out of 50. So 3 of them, they will be given trophies, and the next 3 of them will be given certificate of appreciation, but all of them are first, second and third prizes. So for the first category, the second prize is shared by 2 of our students, Anuradha Mishra and Aditya Goyal. Second prize in the same category goes to Manu Sharma. Manu Sharma from Jamia Hamza. Supervisor is Dr. Leiraj Ansari. Now the first prize in the same category. Nazish Akhtar. Nazish Akhtar from AMU. And this congratulations He's from Aligarh Muslim University. Now it's time to announce the awards for the second category. For the third prize, again, the 2 people, they share the third prize for certificate of appreciation. Again, the equal prizes. Nikita and Siddiqa Javed. Nikita and Siddiqa Javed. Siddiqa Javed is from AMU, and Nikita, she is from RCB. Nikita. Maybe I'll just tell. Nikita is. Where is Nikita? Second prize in the same category goes to Uruj Fatima and Adib Khan. Yes, sir. And the first prize in the same category goes to Furqan Chaudhary and Anupam Singh. Furqan Chaudhary and Anupam Singh. Anupam is not here, but the prize is it goes to Furqan Chaudhary and Anupam Singh. अपना कलेक्ट कर लें, बता दीजिएगा जो भी इंस्टीट्यूट से हैं. Again, I'd like to thank you, our esteemed felicitators, for their invaluable time, expertise, and dedication in recognizing and honoring these exceptional contributions. I now request the Dean Academics, Organizing Secretary, and Head of Department of Biotechnology to please felicitate our esteemed felicitators on appreciation. Yes, sir. It's Dr. Saino Vajna. Dr. Janu Karana. Dr. Thank you so much, all. This event would not have been possible without the visionary guidance of our organizing secretary and the relentless efforts and tireless dedication of our organizing team. Their hard work has ensured the smooth execution of this conference, and it is now time to express our heartfelt gratitude towards them. I request the academics, the Head Department of Biotechnology, and Saima ma'am to please present a token of appreciation first and foremost to the Professor M. Abdi, sir, the organizing secretary and the driving force behind this event. Sir, the membership, the instructor and management have been the cornerstone of this company. I request to present a memento to Dr. Zamira Parvati ma'am, Head Department of Biotechnology and one of the convener of this conference. She has been a constant pillar of support, ensuring that every aspect of this conference runs seamlessly. I am request to felicitate Dr. Saima Bajad ma'am, one of the convener of this conference, for her invaluable contributions, unwavering commitment to have greatly enriched Further, I request to present a memento to Dr. Janati Narayan ma'am, one of the convener of this conference, for her dedicated efforts, coordination, and coordinated input in making this conference. Now, I request to present a memento as a token of thanks to Dr. Javed Ahmed Sheikh, sir. I also request to felicitate Dr. Miraz Ahmed Ansari. Further, I request to present a memento to Dr. Javed Ahmed sir, the
I will request to present a memento to Dr. Monica Sarki.
Thank you all. Now I invite the backbone of this event, our hardworking organizing team. This team consists of three groups. Those who have been working tirelessly since the very beginning, those who have dedicated immense efforts during the conference days, and those who stepped into support in our various capacities. I request the first group, those who have been involved since the inception of this conference, Ms. Firdaus, Anupam, Anuradha, Saima, Madhura, Priya, Irfan, Rashmi, and community team from the contract for a good photograph. The committees and the have been instrumental in making this meeting successful.
चेयर के पास है
A big round of applause. Thank you so much. For our second group, those who worked exceptionally hard during the conference days, I request you all to kindly stand up in the audience as a mark of appreciation for your tireless efforts. I request Sakshi, Zaid, please stand up on your seats. Sakshi, Zaid, Ambar, Anand Nizam, Ramyan, Nigar, Abuzar, Adib, Aditya, Ubaid, Ehtisham, Stefan, Krishna, Uneda. Okay, come up on the dais for a group photograph. Krishna, Uneda, Manu, Ushi, Varun, Vakshita, Swati, Samitya, Sadiq, Anam, Jana, Bhupali, Harisha, Aman, Asad, Girika, Anam, Jayakasha, Sadaf, Sumaya, Arif, Monis, and Aisha. Please come up on the dais.
एक बार सेकंड का हुई नहीं मैं तो रुक गया
सर आगे भी टाइम मुझे थोड़ा साइड लाइन कर साथ में खिंचाएंगे आगे भी आ जाओ ना नीचे Thank you so much. And finally, to all the volunteers who contributed in any capacity, we extend our heartfelt gratitude. Let us all give a big round of applause. Every are in charge of structure. A big round of applause to the Department of Biotechnology, please. Now, I request Ms. Noor Fatima ma'am and Mr. Syed Naveed Kadri to please come up on the dais. Yeah, for their assessment and. Additionally, I extend my sincere gratitude to our department's technical staff whose invaluable contributions have ensured the smooth execution of this event. I request Mr. Rajat, Mr. Islam, Mr. Farad, come up on the diet for a group photograph. इंसान में हम सामने आ रहे हैं
जी जी मैं हूँ
सर्टिफिकेट है हाँ जी हाँ जी
सबसे मजबूत कड़ी सबसे मजबूत कड़ी
And please, a big round of applause to Srandaya and Dhanjaya and Vajaya. Efforts and unwavering dedications, your contributions have truly made this conference a resounding success. Now, I request Dean Academics, Professor S. Raisuddin, sir.
So good evening, everybody. So I see a lot of energy and enthusiasm in all the participants. So that testifies that workshop, this conference was a very resounding success. So I congratulate the organizing secretary, Professor Ardin Sir, and his very, very strong team of teachers, Dr. Saima Wadi, Dr. Umara Fafi, Dr. Maraj Ansari, Dr. Javed Sheikh. Javed Sheikh is rock star of the So there is another Javed Saab and the Monica Saifi, Jagati Naran, and all the supporting staff. I think the conference was for 3 days, but I understand because I was involved in the initial proposal stages. So in the month of September or August 2024, Professor Abidin started the preparation, and it is his vision and hard work and toiling efforts which we see that today it was a great success for 3 days and jam-packed the hall and all technical sessions and plenary lecture, keynote addresses. They were well attended. That shows that there was a lot of interest in this workshop. Department of Biotechnology itself is a big department in terms of PhD scholar, postdoctoral fellows, UG and PG students. So, but besides the department, there is a good participation from institutions like Illegal Muslim University, Regional Center for Biotechnology, and many others I don't exactly recall. So, at the outset, I'd like to congratulate the team of Biotechnology Department, the School of Chemical and Life Sciences, which made this program very successful and very interesting in terms of the scientific content, deliberations, and type. The level of the speakers was very high. I saw the first day there was former director of Central Institute of Medicinal and Aromatic Plant, Dr. Khanuja, and Deputy Director, the Additional Director General of Indian Council of Research, Dr. G.S. Toteja, and so many other speakers I can't remember and mention at this forum. So that shows that this workshop come conference was a success, and I hope that you all have taken some message, some kind of direction, some kind of inspiration from this workshop, and would like to utilize it for your career development, improvement of your file, and I saw that many young people, young research scholars won award in the oral for the oral presentation as well as for poster presentation. So I congratulate them all for this success and this achievement. And I don't need to emphasize about the importance of biotechnology. So biotechnology will be there uh, for all types of problems which human race faces, which, for example, the global challenges you mentioned, we are facing the challenges of global warming and other types of challenges, food security challenges. So in all these aspects, biotechnology, either animal biotechnology, plant biotechnology, environmental biotechnologies, or the agricultural biotechnologies, medical biotechnologies, they will be there to play some role. And since you are all young, you will flourish in your career. So choose a very good career for your life and contribute to the human welfare, environmental welfare, social welfare, agricultural welfare. As a biotechnologist, you can play. You can also start entrepreneurship, startups, which generally not very strong in our university, but still there is a lot of scope for establishing the biotechnology startups. For example, there is a biotechnology park in Lucknow. So the former director and founder director of that institute, Professor Prahlad K. Seth, was the first and director. So I visited one. So I saw a lot of young people like you in ages of 30, 20s have some kind of startup there and producing some product, some enzyme, some kind of product for industry or for pharma industry, agriculture industry, and whatnot, what on. So, for example, you will be surprised to know that in our department in the pharmaceutical biotechnology, there is one professor, Professor Vibhu Prasad Panda. He had one technology for enzymes, fungal enzymes. So it was bought by a Chinese company. They paid him 40 lakh rupees for that one technology. And then we have a policy in our university, 30% of grant only is retained by university. So 70% money which he got, that is 28 lakh rupees he got. So 40 lakh rupees technology he sold, so 28 lakh rupees he got in his own pocket. After paying some tax, definitely he must be having huge amount in his hand. So those kind of startups, you don't think, PhD is also one of the great and good objective of career. For example, you can become teachers. Many of you must be aspiring to become teachers like Dr. Professor Abdi, Professor Dr. Saimawadi, Dr. Faruqi, and others like the Sheikh, who is getting so much clapping in this hall. So you can become a job teacher. For that, you have to qualify NET examination. For PhD admission, NET examination has been compulsory for from this next academic year. So you have to qualify NET. I think you are aware. Those who are MSc and those who are already in PhD, they need not to worry. But for their job, maybe they have to qualify NET examination. So there is a lot of competition for biotechnology. Sometimes people say there are a lot of biotechnologists. For example, MIT University has produced 200 biotechnologists per year. And so sometimes there is overflow of biotechnologists, but you can go for entrepreneurship, some startup. If you don't find some startup in your university, at least go to the MIT Incubation Center. You will find, I think there was some award winners from MIT University. They will tell, I have visited their startups, so small startups based on biotechnology. So I think that career also, and Professor Abdi has already industry collaboration with many industries, like I can name one Ipika, which is one of the company which is listed on the the Sensex, I think America से Bombay Stock Exchange पे listing भी है इस company को, so big company, so he has transferred some technology to him, that company, so I think that may be one of the, and India provides, like there are many, on the first day, Dr. Sanjeev Kumar mentioned about the medium and small scale industry entrepreneurship grants. I think Sufi grant, Sufti, some grant he was mentioned to use that grant for taking, instead of become employer, give employment to so many. And biotechnology has limitless opportunities, I can say. I sometimes envy. I am toxicologist, so there is no much entrepreneurship opportunities in toxicology. Chemistry also, but biotechnology has a lot of these kind of biotechnologies, a lot of this opportunity waiting for you. A lot of Indian government's policies support entrepreneurship. So this conference was oriented towards that also. You might have heard from many industry guys, they spoke here about the biotechnological applications in various aspects. So once again, I congratulate you who came to Jamia and attended this conference and won prize also, many of them. And like to congratulate the team under Professor Abidin Saha, who has made this conference a success. It is 6:30, but still, I think 80% seats in this hall are full. So that again testifies that the conference was of great interest, not boring at all, and you look cheerful at this evening time. But that again shows that this conference does not need any kind of certification. It is a successful conference come workshop. So I hope that in near future again this department will come up with more such conferences and where you will be participating, gaining knowledge, interacting with big people. Listening to very renowned persons is also great opportunity, like Dr. Kanaja, Dr. Toteja. He is working in Jodhpur and they have 2,000 crore grant for entrepreneurships and all like that. So I think we might have spoken about that. I don't know, but Dr. Toteja, C.S. Toteja is working with the Rajasthan government for these kind of initiatives. So I like to again end with the note that take good message from Jamia Hamdard. Come again, those who are from outside of Jamia Hamdard. And those in Jamia Hamdard, go outside of Jamia Hamdard. Because it is not the zenith of the career which you should spend here. बाहर जाइए, दूसरी जगह जाइए. Go to the Regional Center for Biotechnology. The Translational Health Research Institute is there in Faridabad. And now why Faridabad, why NCR? Go to Bangalore, go to other places, Chennai, Baroda, Pune. A lot of biotechnology institutions and industry are there. So I wish all the best to you and good night. Thank you very much.
Now I request Professor N. Jati, sir, the organizing secretary of this event, please share the closing remarks.
So good afternoon to all of you. Actually, the Dean Academics, Professor Raisuddin, has already told about in detail about like the conference and then the take-home message that the conference has given you, and then how you can use the information that has been provided to you by the experts during these 3 days deliberations. So I hope that this conference will help the young researchers and also the scientists at early career stage and also the scientists. They can use all the information that is disseminated and then they can also, actually this platform has given them the opportunity to collaborate with the scientists working in their respective fields.
or even if they have some ideas to collaborate with the scientists in allied disciplines. So this conference was given a platform to them to interact with them. Hope that some of you have already discussed things and about the collaborations that may come up in recent days. Maybe some of our teachers, they may have MOUs with the institutions. Actually, some scientists, they have already discussed with me about the MOUs. So one of the institution, he has already submitted you the application for MOU. So I hope that you will expedite that, and then many more to come. I think during these 3 days we have discussed many areas in biotechnology, from plant biotechnology to animal biotechnology to microbial technology, and then also about the diagnostics so diagnostics you must have heard today itself, like how nanotechnology can help in having the diagnostic to detect metabolites and antimetabolites in different kinds of samples, including food, feed, and milk, and many other products. Actually, one of the experts from Jawaharlal Nehru University, she has already deliberated the product that they have developed as biosensors for this. And of course, in the area of plant biotechnology, I think you must have already informed about the kind of crop improvements that are being led by the scientists in different institutions. So now actually the emphasis is on making crops which are not going to yield better in this global climatic change in this scenario. But then also they are trying to develop crops having produce with better nutritional quality. So now focus is not only to feed people, but the focus is now more emphasis is on to provide nutritious food, so that the people should be healthy. And similarly, the major focus of the conference was that the sustainability. So actually, we already know that United Nations has set sustainability goals, and India is one of the signing for these goals to work upon. So we have already deliberated three of the sustainability aspects: one sustainable agriculture, sustainable health care system, and then sustainable environment. So actually, with the so these have already been deliberated, and informations which have been developed so far shared by the experts. Actually, I must thank to my team, to the teachers in the Department of Biotechnology, to the students, and the non-teaching employees of the department. They have really done a great work and helped me since the inception of this. So as our dean has already told, now he is dean academics, the highest post in the university. So in 2024, I have actually conceptualized about this conference, and then discussed, and we have this title for this biotechnology solutions for sustainable solutions. And I am really thankful to all of you. And actually, with these words, I thank you again. I thank you, all of you, and I am handing over as to the monitor, Dr. Mohan.
Now I invite Dr. Maya Talvi ma'am
in Department of Biotechnology for the forward vote. Very good evening to all of you. So it's my pleasure to stand before you all to formally present the vote of thanks and conclude the 3 days workshop on sustainable biotech solutions for global challenges. This conference was sponsored by the DDT and DST and also sponsored by many private companies. First and foremost, I would like to express my heartfelt gratitude to late Hakim Abdul Hamid Sahib for his visionary leadership in founding this esteemed university. We all are here because of his contributions. His pioneering efforts have contributed immensely to human welfare, and it's because of his vision that we stand here today. We deeply acknowledge and appreciate his extraordinary contributions to society. I extend our sincere gratitude to our Honorable Vice Chancellor, Professor M. Shah sir, for his unwavering support and encouragement. His deep interest in biotechnology, the environment, and sustainable development has been a constant source of motivation for the development of biotechnology, enabling its continuous growth and advancement. A special and heartfelt thank you to our esteemed chief guest, Dr. Sanjay Kumar, Chairman of ASRB, Department of Agriculture, Research and Education, Ministry of Agriculture and Farmers Welfare, Government of India. Despite his demanding schedule, he graced this session and with his presence and shared valuable insights. We extend our heartfelt gratitude to all our distinguished speakers for sharing their valuable expertise and thought-provoking insights. Their enlightening discussions and in-depth knowledge have greatly enriched our understanding and inspired meaningful conversations on biotechnology and sustainable development. I hope all of you have taken some lessons and it's a source of inspiration for all of the students to learn from these lectures. We extend our heartfelt gratitude to our respected registrar, Dr. M.A. Sikandar and his office, as well as our finance officer, Ashu sir, for their unwavering support in ensuring the success of this conference. Their assistance has been instrumental in making this event seamless. I also express my sincere gratitude to the Dean Academics, Professor S. Raisuddin sir, for his continuous support and guidance. His commitment to academic excellence and encouragement have played a crucial role in fostering thriving research and learning environment. Thank you, sir. A heartfelt appreciation to Professor Suhail Parvez. He was busy today, so he could not be with us today, this time. So our heartfelt appreciation for him, for his generosity, humility, and inspiring leadership. His constant encouragement and support have been a true source of motivation for all of us. A very special note of thanks for Professor M.Z. Abedin, Director of CESD, the organizing secretary of this conference. So, sir, without your guidance and support, our team would not be successful. You are a source of inspiration and always keep us on our toes so that we work hard and make the event successful. So thank you, sir, for your invaluable leadership and inspiration. We also sincerely thank the conveners, especially our senior faculty member, Dr. Saima Wajid. She has been she is the lifeline of our department.
every time anything is stuck, so we always just go to Dr. Simon, what is the solution? So, Dr. Simon, thank you so much for that. And Dr. Jagrati Narang, Dr. Jagrati Narang, thank you. And the co-conveners, Dr. Javed Ahmed, sir has already, Dr. A sir has already appreciated your Dr. Miraj and Dr. Monica Sapri. Yeah. A huge round of applause for Dr. Monica Sapri and Dr. Javed Ahmed for his active involvement, meticulous planning, and continuous engagement in making this conference. A very special thanks to Dr. Javed Ahmed in actually planning the workshop and very meticulously you have made the workshop manual. Everything was organized from you. So thank you. A special thanks for your contribution. Our deepest appreciation goes to our sponsors, DBT, DST Serve, NextGen, Edior, Takara, and Tag Biologists, Microlit, and Biorad for their generous support, which has been instrumental in bringing this conference to success. Our sincere thanks to the esteemed judges of the oral presentation, Dr. Azhar Jabin, Dr. Nidhi Agarwal, and Dr. Javed Ahmed Sheikh, and also the judges for poster presentations, Dr. Saima Wajid, Dr. Jagriti Narang, and Dr. Iqbal. For their time, so we are thankful that you spend your time, effort, and shared your expertise in evaluating the research work presented. We are also immensely grateful to all our dear participants whose active involvement and valuable contributions have enriched this conference. Your presence and engagement and active discussion, especially in asking the questions, that has kept this conference lively. A special acknowledgement to our dedicated volunteers whose tireless efforts behind the scenes ensured the smooth execution of this event. Their commitment and enthusiasm has been truly commendable. A big round of applause to Firdaus, Anupam, Priya, Anuradha, Furkan, Saina, Yelnaz, Yashvin, Madia, Samir, Amber, Anam Nizam. This shows the teamwork. All of you have really contributed. So I should go on. I should not miss any name. Oshin, Uneza, Manu, Aditya, Etisham, Rushna, Charik, Anna Mursaleen, Rupali, Arisha, Aman, Asad, Girika, Anam, Kekasha, Sadaf, Sumaya, Monis, Arif, Soumitra, Asad, and all the volunteers whose names have not been mentioned, but they worked very hard to make this event a success. So I apologize if I missed anyone's name. So we'd also like to extend our sincere gratitude to Miss Noor Fatima and Mr. Saeed Nakvi Kadri. Navid Kadri. Thank you, Navid and Naneem, for your efforts. You were also the backbone of our department. Our heartfelt thanks also go to technical support team and administrative staff whose seamless coordination and efforts have been crucial in managing logistic, audiovisual arrangements, and all other technical aspects. So their behind the scenes contributions ensure the smooth conduct of every session. So a big round of applause for Mr. Nizam, Mr. Islam, Mr. Imran, Mr. Wajid, Mr. Murad, Mr. Sajid, and all those who contributed to this conference. So my sincere thanks also to Professor Khurshid Ahmed Ansari and Dr. Salma Wajid, who in association with Poets from Urdu Academy, Delhi. Yesterday we had a big mishaya, and that is a contribution from Professor Mufid and Dr. Saima. So their efforts in curating a vibrant and engaging program added a special charm to the event, making it memorable experience for all. Finally, I extend my sincere thanks to every individual who contributed in any capacity to making this conference a success. Your support, your dedication, and hard work have been truly invaluable. Thank you all once again.
Thank you so much. Before we just conclude, a big round of applause to our UG students, the undergraduate students, dedicated with this, we officially conclude the lecture conference from workshop on sustainable biotech solutions for global challenges. I request everyone to please stand up for the next lecture. Thank you so much. We thank you all once again for being part of this extraordinary journey. May the learning from this conference inspire us to create impactful scientific solutions for a healthier and more sustainable world. Wishing you all success in your future endeavors. Thank you.
कॉपी कर लेंगे
बाद में देंगे ना तो इसके लिए अरे अरे अभी बाद में कर लेंगे
जाएगी मैम
ऐसे बंद कर दो
कॉपी कर ही ले
अरे यार तू ऐसा शाम कर लियो ना अपना हॉस्टल में
अबे इकरा क्या तो कर
अच्छा तो वो केबल निकाल
ये रखो
ये रखो हाथ में करेंगे चल
अबे कर ले 2 मिनट लगेंगे
बहुत कम
ये
करता
सारे
है
फोल्डर्स बैग डाल दो
तो इधर आ जा
सबको कट पेस्ट कर
कट कर लो
हाँ कट पेस्ट कर
ये सारी है ना
हाँ ये सब है
हाँ 63 आइटम बताओ
अबे तो और है अभी पूरे तो किए भी नहीं है
हाँ तो वो तो मुझे पता है
ये 2 मिनट बैठ पेन
ढक्कन का है इसका
पता नहीं इसका ढक्कन
फेंक दे से लीक हो रहा है
छोड़ काम है ये कैमरे किसी लेना
तो दोनों निजाम भाई को हाँ हाँ उन्हें देना
उन्होंने दिया कि ये मैम को देना
मैम को नहीं भाई निजाम भाई कैमरा किसे देना
कैमरा किसे देना कैमरा मुझे देना
हाँ तो और तो कुछ नहीं देना ना
नहीं सब ये सब तो अपनी पार्टी जाएगा
हाँ ये तो लड़की
फिर फोटो खिंचाते वहाँ कैमरे
वरना मैंने गया बैक कर
है और है रिप्लेस चल कोई ना स्किप दिस डिटेल्स कर दे
हो गई सारी कर दी
और है कॉन्फ्रेंस 2025 वाली नीचे वाली
ये तो वो है सिर्फ
हाँ उसे भी कॉपी कर
से डिलीट कर
संजय मिश्रा का भी कॉपी कट कर दी
अरे डिलीट कर यार इतना क्या रोता है तू बस खत्म देख दे
इकरा दी
पूरा बाहर हो गया